2-3, раз, два, три, проверка, раз, два, три.
Привет, привет. Как ты?
3, раз, два, три, проверка, раз, два, три.
Ты под аккаунтом Дмитрий Рудой? Под аккаунтом моим Дмитрий Рудой? Под этой почтой? А? Я, наверное, не буду, я, наверное, через YouTube буду сидеть. А?
Раз, два, три. Но меня не видно, смотри. Или закреплен там, или там закреплен, типа. Нет, а что это такое? Подожди, что я нажал? Включить оригинальный звук. У меня видео активно. А, у меня звук выключен. Вот. Раз, два, три. Проверка. Один. Так, ну ладно, что тогда? Если что, на крайняк буду просто... По идее, можно сделать вот так. Если там закрепить два раза, не раз, то два раза. Ой, это правый угол. Да, там есть закрепить. Вот. Есть закрепить, и все. И он будет
Это у тебя
закреплен.
оповещение или у меня?
Но я оттуда, я тебе не увижу, как закрепить. Вот так можно, повернуть.
Так, ну вот не видно.
Ну, вот так вот, да. Так. Ну, если не видно, да, все, это видно. Это мне что ли? Коллеги, доброе утро, слышно?
Это мы так. Так, это, наверное, лучше его.
Ты сможешь поперемещать?
Да, перемещаем. Только вы мне говорите, кто кого куда.
Good morning to all of you. I mean,
Hi, Santosh. Nice to see you again.
nice to see you too. How are you?
Good, good. Are you in your office?
Yes, I am.
You're allowed to go to office?
Yeah, now we can. But classes are online.
Yep, yep. We are not allowed. Oh, our university is locked down.
University is closed, the classes is online, but you can go to your office.
Прошу вас.
О, мне трансляция тоже. Это хорошо.
Мгм. Я знаю. Спасибо большое. Спасибо большое.
Да, 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 да.
Кто открывает? А где это сегодняшний реглан?
Большая печатная, либо вот такая, либо где можно делать пометки.
Да, да, да. И все включено.
Good morning, everyone. I hope you can hear me. It gives me great pleasure and honor to start today's conference. The preparation for the event was very challenging, as you may understand, but The pleasure is that we finally made it and we're here today. And not only today, but tomorrow also. And not only those of us who are here in this beautiful conference hall, but also many, many people all over the world who are also watching us, who are participating in this event actively, distantly from different countries. And without any further notice, I will present the chair and the founder of this conference, Professor Minzi Liang from Malaysia. This conference was started, if I'm not mistaken, in 2014, and there were a few events, and unfortunately the pandemic situation that emerged in recent years made it impossible for us to have our regular planned event, but we were working hard to keep this conference alive, and we're very happy working as an international team and to have it happen. So without any further ado, I give the floor to the founder and the chair of this conference, Professor Minzi Liang.
Hello and a very good morning to everyone. I wish to extend my greeting to Professor Besaria Meshki, Rector of Don State Technical University, Professor Michael Chikandas, Co-Chair of ICOM-2021, Professor Irina Donik, Vice President of Russian Academy of Sciences, Professor Irina Ryabchik, Deputy General Director for Research and Development of Lalaman, Russia, Professor Yuri Ivanov, Academician of Russian Academy of Sciences, Dr. Dmitry Rudoy, Organizing Committee Administrator, Organizing Committee Chairs and Co-Chairs, Honorary Committee Members, Secretariat and Members, Honorary and Distinguished Speakers, University teams from Don State Technical University, all our dear participants, both at site and attending virtually, members of the press, thank you so much for coming. Ladies and gentlemen, I wish to introduce the brief history of ICOM series. So the International Conference on Beneficial Microbes was first established in 2014 in Malaysia. And many people do not know where Malaysia is, so I have a brief map here. Malaysia is actually sandwiched between Thailand and Singapore. So these are some of the photos that we had when we first had our first ICOM in 2014 in Penang, Malaysia. We had our dinner at a revolving restaurant. The restaurant could turn 360 degrees, and our participants could have a very good view of half of the island. Penang is an island in northern Malaysia, and our university, University Science Malaysia, is the only national university in this island. In 2016, this conference was moved to Thailand in the beautiful city of Phuket. In 2018, we moved it back to Malaysia, in eastern part of Malaysia, in a city called Kuching. The dinner was also very special. It was on a boat. We chartered the whole boat, and everyone had a very nice dinner, river cruise, drinking. And I think this is the only photo I have of you, Michael. I wish to also stress that the ICOM conference is more like a family gathering. In addition to doing good science, all our invited speakers are like family. As we can see here, one of my distinguished keynote, Professor Zong, was helping us packing conference bags one day before the conference. So in 2014, we have 222 participants from 24 countries. Malaysia then was the host, and as you can see, we had the majority, 51% of the participants. The other 49% came from the other 23 countries. And the types of participation, majority we had oral presenters, then we had poster presenters and normal attendees, and at that time we had a very small percentage, 7% of exhibitors, because we had our sponsors and we had booths. There were 5 tracks at that time, so all related to beneficial microorganisms. We had agri and aquaculture, medical and health, food and nutraceuticals, omics and molecular and industrial perspectives. That's when our industrial collaborators and sponsors give insights of their own R&D. In 2016, in Phuket, Thailand, the participants rose a little bit to 226 from 27 countries. And at that time, we still had majority from oral presenters, followed by poster presenters. Industrial participation rose to 13%. We maintained most of the category of our tracks. In 2018, the participants rose a little bit to 231, but we lost in the number of countries. This time, only 17 countries participated. We have the academic, which covers both oral and posters. Then we have those from R&D institutions that presented their latest research updates. But from industries, we rose to 17%. And it was really a good insight at that time to see all these industrial players presenting their industrial R&D. So in terms of tracks, we have food microbiology, science and technology, aquaculture and environmental, medical and health, only 3. In 2020, ICOM 2020 was planned last year to be held in Taipei, Taiwan, end of May. However, because of the pandemic, this event was completely cancelled. And this time, this year, in 2021, having this event in Russia is record-breaking. We have never hit 300 participants, and this time, we had 378 participants from 87 countries. Too many to label, so I believe I just color the globe. And from the colors, we can see that we cover almost 3/4 of the world. So a well-greeted congratulations to our Russian team for making this a huge success. I just labeled the top 3 participants. So we have 37% from Russia, China 4%, and Egypt 3%. And indeed, although Russia is a host this time, participation is only 37%, which means the majority comes from all the other foreign countries, making this event a very international one. So this time, we have slightly different tracks. We have human probiotics, animal probiotics, and probiotics biotechnology. So the ICOM series has always developed to support young, budding scientists. We always have poster and oral competitions and awards to award our young scientists to present their work, encourage them to show off their R&D showcase, and at the same time, we give cash awards and certificates. So these are some of the examples from previous years. I can see that this time for ICOM 2021, this tradition is continued, and I'm very glad that our young scientists can benefit from this international event again. So, ladies and gentlemen, I wish to extend the biggest welcome to Russia at Don State Technical University, Rostov-on-Don. How I wish I could be there personally. I've seen photos from Professor Michael Chikandas. He's showing all these beautiful photos of Rostov-on-Don, making us very jealous. I hope to come and visit one day. Till then, everyone, have a great time. Have a great time contributing to science, doing great science, establishing new networks, and I hope the tradition of ICOM of being a family gathering will continue. Thank you.
Thank you, Minze. I wish you were there with us, but unfortunately there was some distance, and we were just discussing one matter yesterday and a few days before. We are hoping to organize the summer school here at the Don State Technical University once all these unfortunate events will finish. And we hope to see many of the international participants of the conference also participating in this event. I would like to give a floor to the rector of Don State Technical University, Professor Bessarion Mesky, and I'm very happy to use this opportunity also congratulating Dr. Mesky on election of the corresponding member of the Russian Academy for Education. This is a really very big honor, and this is an indication of Professor Mesky's achievements in education. Professor Mesky, the floor is yours.
Thank you. Дорогие друзья,
Dear friends,
уважаемый Михаил Леонидович,
dear Mikhail Leonidovich,
Миндзе,
Minte,
спасибо огромное за столь энергичную, великолепную информацию, которую мы сегодня от вас получили.
thank you so much for the wonderful and extended information that we got from you today regarding the history of the conference.
Сегодня, я думаю, что Донской гостехуниверситет может свой счет записать еще одну удачную, великолепную конференцию.
And I hope that today we'll be able to put another very successful conference into the books of our university.
Так как я посмотрел статистику сегодня, 2014 года до сегодняшнего дня, максимальное количество участников от 200 до 250 было, а сегодня 370 участников данной конференции.
Because as Honorable Minte has noticed, since 2014, the number of participants had hit the amazing 378 participants as compared to 2014.
Я прекрасно понимаю, что 2020 год, год ковидный, заставил сегодня нам думать и всем вместе собраться и обсуждать те проблемы, которые сегодня существуют.
I fully appreciated that 2020 wasn't an easy year for all of us, and it made us gather together and think about new ways of working.
Данная конференция сегодня будет рассматривать, и наши ученые, участники данной конференции будут обсуждать очень много вопросов, касающихся разных штаммов, микробов и все то, что сегодня нас окружает, и то, что является, может быть, и безопасной жизни всех, кто живет на Земле.
Today, our honorable members of the participants of the conference have gathered to discuss various significant microbes, strains, and everything which surrounds us, all the things which affect our safety, security, and general life welfare.
Сегодня данная конференция обязательно затронет вопросы, то, что касается медицины, пищевая промышленность, и комбикорма, аквакультуры, и полностью это безопасность любой страны.
Today, the conference topics will cover such important things as agriculture, aquaculture, food industry, pet foods, medicine, and other areas.
После мегагранта, который выиграл Донской гостехуниверситет, мы с гордостью можем сказать, что ученый с мировым именем Михаил Леонидович Чекиндас, который возглавил эту лабораторию, дал еще один толчок развития нашему университету и науки в этом плане.
I'm very happy to welcome Professor Michael Chekindas, who, since the Don State Technical University had won the mega grant, led the science school of our university, and we have already achieved quite a few big things.
Считаю, что данная конференция сегодня станет скрепом, наверное, объединяющим ученых, которые сегодня рассматривают данные вопросы, которые перед собой мы поставили. И мы должны здесь, при этом общении, большой синергии, решить те проблемы или хотя бы наметить планы решения данных вопросов, касающихся тех моментов, которые уже поставлены перед собой.
I strongly believe that today's conference will become a platform which will unite all the members of the conference all over the world, and it will become and which will lay significant groundwork for further research.
Каждое государство сегодня планирует долгосрочное социальное и экономическое развитие страны.
Each country in the world is in the process of planning long-term social economy development of the country.
Укрепление национальной безопасности каждой страны.
Strengthening of the country safety and of each country.
В этом огромную роль играет наука и образование.
And of course, science and education play the integral role in this planning.
Уверен, что сегодняшняя конференция с участием многих стран и ученых, которые здесь присутствуют с мировым именем, они дадут ответы на те вопросы, которые сегодня будут обязательно поставлены, не только обсуждать, и поставлены будут вопросы.
And I'm confident that all the scientists who gathered today will give answers to all the questions that were already put, and they will put the new questions and find the solutions as well.
Уважаемые коллеги, друзья, вы прекрасно знаете, что наш президент нашей страны Владимир Владимирович Путин объявил 21 год годом науки и образования и технологий.
Dear friends, you all know that the President of the Russian Federation, Vladimir Putin, has announced the year of 2021 as a year of science and technologies.
Я, как ректор, и вся команда нашего университета максимально приложат все усилия, что данная конференция прошла на самом высоком уровне в организационном плане и информационном плане.
So, I, as a rector, and the whole team of Don State Technical University will put the utmost effort into making sure that the conference is held at the highest possible level.
Желаю всем участникам данной конференции успехов, здоровья, хорошего настроения, что побыстрее, чтобы нашли вопросы, решение вопросов. То, что нас сегодня всех беспокоит, это ковид в первую очередь. И если будут новые вызовы, чтобы мы тоже быстрее приступили, решали этот вопрос сообща. Всем удачи. Спасибо за внимание.
So I would like to wish all the participants and organizers of ICOBM conference success, fruitful cooperation, constructive dialogue, and effective interaction in finding the solutions to such important questions as, for example, COVID situation now. And should there be any new challenges, let you find easy ways of solving those problems as well. Thank you so much and good luck.
Я хотел бы еще раз поблагодарить Михаила Леонидовича Чекиндас, профессора, и поздравить его решением ученого совета. Вчера Большой совет Донского гостехуниверситета присвоил почетное звание, почетный доктор Донского государственного технического университета. Это высокое звание, самое высокое звание, которое может сегодня присваивать Донской гостехуниверситет. Поздравляю и добрый час.
And I would also like to congratulate Professor Mikhail Leonidovich Chikindas on becoming the honorary doctor of Don State Technical University. This decision was taken yesterday by the Assembly of Don State Technical University. This is the highest possible rank that a scientist can get in our university, and I wish him all the best and a lot of new solutions to find.
Да. Еще раз хотел бы доктору Миндзи сказать о том, что Донской гостехуниверситет ждет вас. Мы вас приглашаем в наш Донской гостехуниверситет, как будет у вас возможность посетить и вместе обсудить те проблемы, которые будут интересны вам и России, и Донскому гостехуниверситету. Спасибо за все.
To invite Dr. Minze to come and actually visit our university when the situation allows. You're most welcome. I'm sure that together we'll be able to find new ways, we'll be able to discuss the most important issues, and you're very, very welcome. Thank you so much for organizing this conference.
Dear colleagues, friends, I would like to take a moment to introduce our guests, those who are presiding this event in no particular order, but of course I will start with Dr. Minze Leong, who is a professor at the University of Science Malaysia. No need to say much about these distinguished investigators, just one thing to mention. In 2020, she was listed in 2% of the top global scientists by the Stanford University. I think this is a true recognition of her achievements. Today with us are Dr. Vyacheslav Ilyin from the Institute of Medical and Biological Problems. Probably to say towards the keywords would be the microbiology and the space science. And of course, we know that there are many issues that are coming as challenges to those involved in space programs, and microbiota is important not only on the Mother Earth but also in the space. The next person is professor and corresponding member of the Russian Academy of Sciences, Dr. Alexander Suvorov. And some of you, but I hope most of you, know that the work of Dr. Suvorov is, for many years, is involved with probiotics and most recently with development of what we call live vaccines. So instead of getting a shot, taking a pill, we can probably drink yogurt or eat any of our regular meals and receive the positive effect from vitamins, probiotics to vaccines. So this is the true indication of what functional food can do to humans. Next to me, I don't think that our rector and Professor Maskey needs any introduction. I already mentioned that he was just recently elected as the corresponding member of the Russian Academy for Education. Next to Dr. Maskey is Dr. Dmitry Rudoy, who is the Dean of the Agricultural Engineering Faculty, and he's heavily involved in our event. His research covers engineering aspects of agri-technologies, but also the issues related to biological aspects of development of agriculture. Last but not least, Dr. Georgy Laptev. I think without any hesitation, I should say that the company he is representing, he's a director of Biotrof, and Biotrof is perhaps one of the biggest and most successful companies in the country, producing the food additives and the probiotics for agriculture. It is not just the business, it's also innovation, because Dr. Laptev is also heavily involved with education process at St. Petersburg Agricultural University. Thank you very much for your attention, and it is a particular pleasure to give the word to the Vice President of the Russian Academy of Sciences, Dr. Irina Donik. Now, what I want to say, and we're extremely grateful to Dr. Donik for finding time to be with us. You can imagine how much work the Vice President of the Academy of Sciences would have, but today is also her birthday. So I hope on behalf of all of us, I can congratulate Dr. Donik on her birthday and to wish many years of successful work in her position, and with great achievements to come many more in the future. Thank you very much, and without any further ado, I give the floor to Dr. Irina Donik.
Уважаемая Ирина Михайловна, Михаил Леонидович поприветствовал вас от лица Организационного комитета всех собравшихся здесь сегодня и всех вышедших на связь онлайн. Он крайне благодарен вам за то, что вы нашли возможность сегодня подключиться к конференции, поскольку он осознает вашу занятость, и тем более сегодня, в такой день, когда ваш день рождения, вы нашли время для того, чтобы поприветствовать гостей и участников конференции. Спасибо вам за это.
Спасибо.
Спасибо, уважаемые коллеги, Виссарион Чехович, уважаемый Михаил Леонидович. Слышно меня?
Да, слышно, слышно, великолепно. И мы вас видим, Ирина Михайловна, и хотим вас поздравить. Вы такая красивая вообще. Но я так думаю, что сегодня прекрасный день. У вас день рождения. Вот все, кто присутствует здесь, и 370 с лишним участников конференции, все приветствуют вас и поздравляют. Обнимаю вас и желаю всего самого доброго.
спасибо за добрые слова и поздравления. Я прошу прощения, что не могу лично присутствовать, но у нас сегодня экономический форум открывается в Санкт-Петербурге, и мне тоже еще предстоит там выступление, поэтому, уважаемые коллеги, позвольте вас дистанционно поприветствовать конференцию, пожелать хорошей работы. Мне кажется, что ту работу, которую выполняет Донской аграрный университет и все коллеги, участники этой конференции, делают очень важное дело, потому что тематика, она очень актуальна. И самое главное, что сейчас необходимо нам всем сплачивать свои ряды, особенно ряды ученых, и ученые должны заниматься вот этой так называемой научной дипломатией, в то время как в
Ирина Михайловна,
стране...
позвольте я быстро переведу, хорошо?
Ой, я прошу прощения, а то я забыла, что да, у нас же идет трансляция. Спасибо.
So thank you so much for all the warm words of greeting and all the warm words wishing me a very happy birthday. So today I would like to welcome everyone and to thank everyone to actually coming together in those difficult times to discuss acute topics which are so important, especially in the changing and challenging conditions of the current situation. It is crucially important that all the scientists, all the researchers work together in those times to find ways of handling all the challenges that the new era has presented.
Поэтому я желаю всем добра, здоровья и успешной работы, уважаемые коллеги. И думаю, что то, что мы делаем, это главное дело, это развитие науки, научного прогресса и дружбы между нами, учеными. Спасибо.
So thank you so much to everyone, and I wish you health, which is very important now. I wish you prosperity. I wish you fruitful cooperation between the countries. What we're doing together is very important. We're working on developing science, developing technologies and innovation. So I wish you a good day ahead. I wish you fruitful discussions, and thank you so much again.
Еще раз спасибо и удачной работы.
Thank you very much and good luck for today.
Спасибо. С днем рождения.
Спасибо, спасибо, спасибо.
Now we will have a short demonstration, the movie about the Don State Technical University. But before we start, I would like to also introduce our friends. As you can see on the banner, Rutgers State University, New Jersey is my home university. My another home university, I should say, because I feel Don State Technical University also my home. Lallemand is one of the leaders of the world in manufacturing probiotics for humans and animals, and two journals, Probiotics and Antimicrobial Proteins, the journal that I started some years ago, and I'm an editor-in-chief of this journal, and our sister journal, Beneficial Microbes, which was started about the same time, and we're going together hand by hand through all these years, serving as a platform for publications in the field of probiotics, health-promoting bacteria. So I would like to invite all the participants, should they have anything to share with the community in their achievements, in their results, in their work, to present their reports to these two journals. I'm not making any differences between these two. They're equally good. So you're very welcome to submit your research reports to these journals. And now we will have the short demonstration about the Don State Technical University.
I don't like being late. I have a lot to do and ambitious plans. If you wish to achieve a great deal, you should start in a place of vast dimension. If your goals are ambitious, it's important to find right people to start your journey with.
Our institute had ambitious plans from the very beginning. enthusiasts and professionals for creating future-oriented reality. Through all the changes and challenges, they managed to save it, strengthen, transform, so as to stay on top in a new, ambiguous, unpredictable and changing reality.
It is important to follow the leaders who think more one step ahead, those who know what the labor market will look like tomorrow, the day you start your career. I'm aimed at becoming such a keen expert in my field. It is a start, and then we will see.
Everyone matters here. Everyone who can become a part of the team and keep unique personality. Two pillars where our academic world stands on. Two principles that our university is built on.
Here you understand that the future is in your own hands. Work hard, take part in research, face real technological challenges.
Bring your ideas to life in the leading enterprises. Every project you take part in here works for you. It helps you to become more competent and stronger in the fields you have chosen.
Have you made up your mind how to surprise the world? Are you ready to try yourself as a business person? Here you will have support and turn your perspective idea into a real product. Get savvy in the most difficult areas. Make industrial prototypes. Here you understand the main thing. To make your dreams come true, you need deep knowledge. Here you can become strong and self-empowered. Here you can join the international family of young scientists. Make your name long before graduation. Prepare the ground for your future career. Here you're not alone. You're a part of our big team. You are with those who have wide knowledge and seek for new horizons. Happy birthday, days to you, the University of Top Priority.
I would like to give the floor again to the rector of the Don State Technical University, Professor Mesky, please.
Дорогие друзья, Лена Николаевна, у нас здесь присутствует профессор Пономарева, доктор наук, который тоже сделала очень много сегодня, то, что мы здесь собрались все вместе, и надеюсь, что наша благодарность и участие многих наших коллег по направлениям аквакультуры, конечно, заслуга ваша. Спасибо вам огромное. Благодарю вас за великолепную работу в нашем университете. Спасибо большое.
Dear Elena Nikolaevna, Professor Panamareva, the Doctor of Sciences, she's with us today, and she's done so much for in the area of aquaculture that, of course, her contribution is immense. The contribution to this conference, to the acknowledgement of our university at the international field. So thank you so much. We really do appreciate everything you do.
Дорогие друзья, дорогие коллеги, дорогие ученые, участники, наши международные друзья, которые участвуют в конференции, всем я желаю хорошей работы. Позвольте мне передать уже основное слово Михаилу Леонидовичу, профессору Чекиндас, и пусть он ведет, и мы продолжаем с вами работу. Я буду наблюдать через свой кабинет данной конференции. Позвольте мне туда передвинуться, и удачи вам всем. Всего доброго.
Dear friends, dear colleagues, dear participants, all the best of luck to you. And now I hand over to Professor Michael Chikindas, who will actually lead the whole conference. Unfortunately, I have to leave the conference today and go to my office. I'll join on Zoom. So all the best, good luck. Thank you for everything.
We'll continue with the greetings. I already mentioned Lalimand today, saying that this is one of the leaders, the world leaders in manufacturing of probiotics for humans and animals of agricultural importance and pets. But I wouldn't say enough about this company if I wouldn't mention that this company is not just about manufacturing, it's also about research. And their research and innovation are really truly amazing. They're leaders not only in production but also in research on health promoting microorganisms. These companies started in Canada, but now the company is present virtually all over the world, and in Russia here we also have the office of the company, and we're very happy to have today with us the Vice President of the, Deputy General Director for Research and Development of Lalimand, Russia, Irina Rabchik. And without any further ado, I give the floor to Irina for the welcoming of our conference.
Good day. I am glad to welcome you to International Conference on Beneficial Microbials. I think it's very important and interesting for specialties and scientific research, especially for young scientists. My name is Irina Rabchik. I am working as a Deputy of General Director for Scientific Research in Moscow office Lalimand Animal Nutrition in Russia. I am responsible for registration Lalimand's products in Russia and research them in the feeding animals and poultry in the farms or research center in Russia. Our company has a good relationship with the research institute, for example, All Russian Poultry Research and Technology and other agriculture institutes, research centers and laboratories. So, I would like to introduce you the company Lalimand Animal Nutrition. It's a global world leader in microbial fermentation using yeast and bacteria in animal nutrition. This company, founded in Montreal by Fred Lalimand at the end of the 19th century. Yeast has been a major of the business for nearly 100 years ago. The first plant in Montreal started to produce yeast in 1923. And now we have expanded over the years, and we now have 22 yeast and 8 bacterial production plants around the world. Canada, USA, UK, France, Denmark, Germany, Poland, Estonia, and other countries. Lalimand Animal Nutrition develops and markets products including probiotics. It is specific live yeast and bacteria to improve animals' health and digestive functions, bacteria enzyme for silage additive, and inactivated yeast including selenium-enriched yeast and antioxidant and mycotoxin solutions, and yeast for microflora balance. All Lalimand's products to improve an animal's health and safety on environment. So, I wish you a good day in this conference. Thank you for your attention.
Thank you, Irina, for your kind words and for being with us. I can imagine that being a Deputy General Director, your time is very busy, and that makes it very special for us to have you with us. We're now moving to the plenary session of the conference with the scientific presentations, and we start with a presentation by Dr. Minze Liang. I don't think I need to introduce this outstanding investigator anymore. Without any further ado, I give the floor to Dr. Liang.
Hello, and thank you everyone for joining my talk in this session today. Before we start, I wish to introduce a little bit of where I am, where I'm from. I'm Leong. I'm from University Science Malaysia. Malaysia is actually a small country that is sandwiched between Thailand and Singapore. And my university is located in a very small island called Penang in the northern part of Malaysia. So once we cross the bridge from the mainland to the island, that's the campus of our university, University Science Malaysia, or USM in short. We were established since 1969. Today, my talk will emphasize on three main aspects. The first, we're talking about smaller models. Then we will proceed to human, and we'll look at gut microbiota profiles. So let's look at smaller models. We had two. The first was flies, and the second was rats. We use a very small model of fruit flies called Drosophila melanogaster. It's very small. It is just the size of the tip of a pen. So what happens is that these fruit flies, these Drosophila, are not normal flies. They are actually mutants. They are mutated to represent an Alzheimer's disease model. So Alzheimer's disease is caused by several factors. One of it is the accumulation of amyloid beta peptide in the brain. And because of this, it causes high toxicity, high neurodegeneration, and finally apoptosis to neurons. So we have several parameters that we have adjusted to produce these mutant flies. We use these UAS flies, which already have the Alzheimer's disease gene, but we use a GAL4 promoter to actually exhibit this into the eyes so that we could actually see whether or not these flies have Alzheimer's disease or not. So we have the UAS flies that are mated with the GMR flies to produce these GMR GAL4 line flies. Now this is a very simple microscopic image where we have a positive control, the wild type, with very beautiful eyes, almost perfect in shapes. Whereas the Alzheimer mutant flies, or what we call the UAS GMR mutants, we can see some forms of deformation. Now this is just a very simple microscopy. Let's zoom in to scanning electron microscopy. So for normal flies, the eye structure is rather perfect, where they have a hexagonal ommatidia. Normally we see that as round, but they're not round, they're actually hexagonal. And on alternate angles of these ommatidia, we have bristles. They are like hair-like structures protruding out from each of these ommatidia hexagonal sides. When we look at the Alzheimer mutant flies, this is where we have the GAL4 promoter to exhibit this Alzheimer severity in the eyes. We can actually see that this almost perfect structure is already deformed. Most of the time, we could no longer see clear structures of the ommatidia. Also, some of the bristles are broken, they are malformed, and in certain cases, we can see holes or pores being formed on the eyes. Now, I would like to introduce one probiotic strain. It's called Lactobacillus plantarum DR7, or DR7 in short. So upon administration of this probiotic to these Alzheimer mutant flies, we could see it prevented the malformation of the eyes of these Alzheimer mutant flies. It is not perfect. We could still see some broken bristles. We can still see some hexagonal shapes that are not entirely hexagonal, but it's a lot better enhanced compared to the non-treated Alzheimer mutant flies. So we began to see some positive effects. Because of that, we proceeded next to using a slightly larger animal model, which are rats. For rats, we actually started with one very simple test. It's called the open field test, where we put the rats in boxes, and beyond those boxes are walls. Rats being very explorative and very adventurous animals, they tend to climb on the walls, scratch it, and try to see what's beyond those walls. However, if the rats are stressed or having anxiety or age with cognitive impairment, they tend to be less mobile, tend to stay in the middle, less to the side of the walls. So when we look at our data, this is an aging study. As brain degenerative problems, we have already associated that with aging. We have several groups of rats from our data. So we had young rats, we had aged rats that are equivalent to human of 60 years old. We have aged rats that have been treated with a control drug, which is statin, and aged rats that have been treated with probiotic Lactobacillus plantarum DR7. As we can see, those rats that have been treated or administered with DR7 showed less number of entries to the outer zone. We also know that as we age, the number of neuron branching reduces. We can see here, comparing between young and aged rats, the neurons, number of neurons branching reduces. So we attribute this to neuronal apoptosis, and we also attribute this to a decline in memory and cognitive abilities. Thus, we did another test, which is the Morris water maze. Here, animals are trained to swim, and inside the swimming pool, we will have a submerged platform. Rats were trained to reach that submerged platform. If they remember how to swim there, they would reach the platform faster. So here we have a video that I wish to share with you. So initially we have young rats that managed to find the platform rather fast as compared to old rats, which somehow lost a little bit of their memory, cognitive abilities, they lost their way, and they needed a longer time to reach the platform. Then we also have aged rats that were fed with the control drug statin, which relatively found the platform faster. We also have aged rats that were fed with probiotic DR7, which also found the platform faster, indicating that they have better memory. When we look at the data, indeed, aged rats needed the most higher latency time, that's what we call it, to reach the platform as compared to those aged ones that were fed with statin and aged ones that were fed with DR7. We also had another test called the t-test. So in this t-test, we put food in one of the arms of the T-maze, and rats were trained to remember which arm it was. Was it right or was it left? Then rats were allowed to run freely every time. How many times they actually succeeded in finding the food, which indicate that they remember which arm it was. So when we look at the percentage of correct arm, those rats that were fed with DR7 yielded a higher percentage, statistically significant, in our T-maze test. We also looked at the hippocampal properties after sacrificing the rats and dissecting them. So we look at inflammatory factors because neuronal branching apoptosis, even neuronal cells apoptosis, we associate that with inflammation. So we have the interleukin-1 beta, IFN gamma, and TNF alpha, which represented the pro-inflammatory cytokines. And as we can see, in many of these cytokines, rats that were aged but fed with statin, the controlled drug, or fed with statin and fed with Lactobacillus plantarum DR7, they actually showed a reduction in these pro-inflammatory cytokines. As this is an aging project, we also evaluated on some aging biomarkers such as P53 and P19. The gene expression of P53 was increased in aged rats. However, it was reduced in those aged rats that were fed with probiotic DR7. As we move along, we wish to examine some of the mechanisms, such as which neurotransmitters in the brain that was actually affected by the administration of DR7. I wish to note that in the brain there are 4 distinct neurotransmitters, one being GABA, dopamine, norepinephrine, and serotonin. When we evaluated the gene expression of hippocampus, for all the 3 neurotransmitters of GABA, dopamine, and norepinephrine, we could not see any significant changes due to the administration of probiotic DR7. However, when we look at the serotonin pathway, we saw something. Now, serotonin did not just exist in the brain. Serotonin is formed from the amino acid tryptophan. So tryptophan can be converted into kynurenine or serotonin, depending on which enzyme that's reactive. If the enzyme IDO is reactive, it will convert tryptophan to kynurenine. However, if the enzyme TPH is effective and reactive, it would convert tryptophan to serotonin. And we look at our data of IDO, the administration of DR7 suppressed the gene expression of IDO as compared to all control. But the gene expression of the enzyme TPH, which converts tryptophan to serotonin, was activated or upregulated upon the administration of DR7. Now, it is also important to note that serotonin is the happy neurotransmitter because those patients with a reduced serotonin level in the brain have been reported to have symptoms of mood swings, brain disorders, anxiety, and even depression. We next proceeded to a human clinical study. So for the human clinical study, we published that last year, and we use adults with mild stress, and our finding showed that DR7 could actually alleviate stress and anxiety, but not depression, in this randomized, double-blind, and placebo-controlled study. So the inclusion criteria was that the subjects must be moderately stressed. We used the PSS-10 questionnaire for this evaluation and screening. Upon that, we had successfully recruited 55 placebo and 56 DR7 groups for 12 weeks administration of DR7. Each day, each subject consumed 9 log CFU of DR7. When we look at some of the parameters, obviously from PSS, they were all stressed. That was the inclusion criteria for recruitment. However, throughout 4 weeks, 8 weeks, and 12 weeks, throughout that 3 months of assessment, no changes in total PSS scores. However, when we look at stress scores, we could see that DR7 exerted some effects as soon as 8 weeks in certain groups, especially the younger adults, which were 30 years and below, and overall subjects also 8 weeks. As for anxiety score, this was prevalent in all groups, regardless they are younger adults below 30 years old or older adults above 30 years old, and the effect was seen evident since week 8 or 2 months after the treatment of or administration of DR7. When we look at depression, there was no significant effects. So DR7 did not show any efficacy in alleviation of depression. We postulate that depression is a more severe case as compared to stress and anxiety, and thus DR7 may not be that effective. When we look at total DAS scores, we saw that the younger adults benefited more from the intervention of probiotic DR7 as compared to the older adults, which we did not see any significant. The younger adults, we could see some significance since week 8. Overall, in overall subjects, and since week 8, we could see a total reduction of total DAS scores. We subsequently evaluated some cognitive parameters. We used the Cogsted computerized battery test, where each subject was analyzed using a computer software to look at their memory and cognition abilities. So there are various types of memory and cognitive abilities. One of it will be such as visual learning, working memory, executive functions, and so on. So when we look at all these, we could see marginal effects for the administration of DR7, typically in those adults aged above 30 years old. However, statistical significance was not achieved. Statistical significance was achieved in other parts of cognitive abilities, such as social emotional cognition and also associate learning, where subjects above the age of 30 obviously benefited more from the intervention of probiotics. We also look at blood parameters. From our animal studies using rats, we could see that DR7 benefited the subjects from hippocampal anti-inflammatory parameters. So for humans, we evaluated on these inflammatory cytokines also in blood. So those subjects aged less than 30 with a more higher stress levels, we could see that the administration of DR7 reduced the concentration of cortisol, IFN gamma in blood, and increased interleukin-10, which is the anti-inflammatory cytokine in blood. Whereas those who are aged 30 with a higher anxiety level, we could see that the administration of DR7 benefited more in terms of pro-inflammatory cytokines such as IFN gamma, TNF alpha, and IL-1 beta. So in terms of all subjects in general, we could see some significant alleviation in blood of these inflammatory cytokines such as IFN gamma and TNF alpha for pro-inflammatory cytokines, and also an enhancement of DR7 against anti-inflammatory cytokines such as IL-10. And in all subjects, cortisol level, which is associated with stress, has been decreased upon the administration of DR7. We also look at blood gene expression of neurotransmitters genes. So from the rat studies, we could see that neurotransmitters mechanism were along serotonin. In terms of human, we evaluated the same thing using blood. So when we look at gene expression of neurotransmitters genes in the blood, this is an inverted data, meaning lower value is higher expression. We could see that for younger adult subjects, older adult subjects, and total overall subjects, we see a rather similar trend, where TPH2, the enzyme that converts tryptophan to serotonin, have been upregulated, whereas the enzymes of IDO and TDO, which converts tryptophan to kynurenine, have been downregulated. So we are looking at the same mechanism of pathway of effects from the administration of DR7 in both rats and also in humans, indicating that serotonin is an important parameter. For older adults and total overall adult, we also saw that the enzyme TH, which was responsible for the conversion in along the dopamine pathway, was also down-regulated. So upon stress, dopamine is converted to norepinephrine, and the enhancement or increase of epinephrine in the blood has been associated with increased stress and anxiety in many subjects or patients with brain disorders. Thus, in the human study, in addition to the serotonin pathway, we are also seeing a possibility of administration of DR7 exerting an effect along the dopamine pathway as well. So the conclusion from the human study and animal studies is that Lactobacillus plantarum DR7 probiotic alleviated stress and anxiety along gut-brain axis in both humans and animals via the serotonin and dopamine pathways. And in human, this was very prevalent in both the probiotic treatment group and the placebo group, where we saw not just the neurotransmitters pathways, but also blood cortisol levels, which was reduced by DR7, and also pro-inflammatory cytokines such as IFN gamma and TNF alpha, which was reduced, and an enhancement of anti-inflammatory cytokines in blood such as IL-10 and IL-4, in addition to memory and cognitive functions. We would also like to note that in addition to elevation of stress and anxiety, memory and cognitive parameters was also enhanced. The next aspect that we look at would be gut microbiota, which is an interesting aspect which we evaluated both in flies and also in human. First, let us look at flies. So when we look at the positive and negative control being mutant flies with Alzheimer's disease, we can see a very prevalent abundance of Wolbachia, which was almost zero in the positive control flies. However, those that were fed with DR7 probiotic and also another commercial probiotic that we get in Malaysia, let's label it Y, the abundance of Wolbachia was significantly reduced. And not just that, when we look at PCA plots, we could see that the shift of gut microbiota profiles was actually moving towards the positive control upon the administration of DR7. And this was better than the commercial control that we had in Malaysia. When we look at different abundance of bacterial taxa, we can also see that DR7 resembles more of the positive control than the negative control. And when we look at KGG profiling, we could see that Wolbachia was highly associated with several disorders of the brain, such as Parkinson, Huntington, and Alzheimer's. When we look at human, Lactobacillus DR7 actually modulated gut microbiota, and we managed to correlate that along the dopamine and serotonin pathways in stressed adults. So what we did was that, similarly to the previous study, which had a higher number of subjects, for fecal samples, we only managed to get 99 samples. So out of the 111 that we had, we managed to analyze 99 samples of fecal samples. And what we saw was that alpha diversity, which measures differences within samples, we evaluated using both the CHA1 index and also the Shannon index. So for the CHA1 index, it provides a measure of richness, while equally taking into account of frequent and rare OTUs. The Shannon diversity index, on the other hand, considers OTU's evenness, as it takes into account the frequency of each OTU as well. Both study groups, the placebo and also the DR7 groups, were comparable in terms of both richness and evenness at baseline, and we could see that across different taxonomic classes such as class, order, and genus levels. However, at the end of the intervention period of 12 weeks or 3 months, the placebo group displayed significantly lower richness as compared to DR7 at both class and order levels. So we managed to see some of these as indicated by the green arrows. This shows that the administration of DR7 prevented the reduction of within-group ecological diversity, which was evidently reduced in the placebo group over time. Next, we look at beta diversity, which measures differences between samples. We use the Bray-Curtis index, which considers both the occurrence, co-occurrence, and differential abundance of the OTUs. So compositional differences, again, were not observed at week 0. However, at the end of the intervention, significant differences were consistently observed between DR7 and placebo across different taxonomic levels again, such as class, order, and genus. Now, these indicated that amid stress within and between samples, microbial richness or ecology was changed or reduced, and the administration of DR7 has aided in the maintenance of such gut microbial ecology to prevent a shift in diversity amid stress. Let us start with phylum. Compared to DR7, subjects administered with placebo had a drop in phylum Bacteroidetes over 12 weeks, accompanied by an increase in abundance of Firmicutes. Now, these changes were also traced to subsequent taxonomic levels. Let us look at the next one, which is class. In the level of class, placebo showed a decreased Bacteroidea and increased Clostridia compared to DR7 after 12 weeks. Further down in the different level of order, we can see that placebo continued to show reduced abundance of Bacteroideals, accompanied by increased Clostridials as compared to DR7 after 12 weeks. A lower count of Bacteroidetes has been observed in patients with altered colonic movements, such as young children hospitalized for infectious diarrhea or patients with functional constipation. Now, this kind of differences was also further trickled down to the level of genus, where Bloschia and Rombolschia were increased in the placebo group at the end of the study as compared to DR7. Although Bloschia has been reported as a butyric producer that is beneficial for colonocytes, a high abundance of Bloschia has also been reported in patients with inflammatory pouchitis, non-alcoholic fatty liver disease, systemic lupus, and also breast cancers. Now, there are concerns where limitations have been raised because accuracy and use of higher taxonomic levels are reduced, such as phylum, to predict and correlate with diseases and biological functions. And these are attributed to the vast diversity of human gut microbiota along lower taxonomic levels. Thus, it is strongly noted that in our study, we show that these changes in gut microbiota upon the administration of probiotic DR7 was consistent along different lower taxonomic levels, all the way from phylum to class to order and also to genus. So as DR7 exerted brain health effects along the pathways of neurotransmitters, particularly serotonin and dopamine, thus now we wanted to correlate between these microbiota groups with genes that are involved along serotonin and dopamine pathway. So while changes in gut microbiota upon administration of DR7 was consistent along different taxonomic levels, the correlation of these changes with neurotransmitters genes also shows such a consistency in correlation. Now let us look at one by one. So the phylum Bacteroidetes showed a negative correlation with gene expression of DBH, which was consistent along lower taxonomic levels such as class Bacteroidea and order Bacteroideus. Now, DBH catalyzes the conversion of dopamine to norepinephrine. So it's an indication of increased stress as seen in the placebo group with a lower abundance of Bacteroidetes, Bacteroidea, and Bacteroideus. Bacteroidea and Bacteroideus, which were maintained upon the administration of DR7 compared to placebo, also showed positive correlation with TPH2. TPH2 converts tryptophan to serotonin in the brain, where an imbalanced level of serotonin has been reported in patients with psychological disorders, as mentioned earlier, such as mood swings and anxiety. As we move next, we can see a positive correlation was also observed between gene expression of DBH and taxonomic levels along the phylum Firmicutes, class Clostridia, order Clostrideus, and genera Bloschia and Robausia. So Bloschia has also a positive correlation with TDO, which competes with TPH for tryptophan in the brain for the conversion into kynurenine. So reduced level of serotonin and increased level of kynurenine is not actually good and has been shown in patients with anxiety and depressive disorders. So moving down to a different phylum, Delta Protobacteria, we see that its lower taxonomic level of order Desulfovibrilnales and genus of Bilophila, which were both increased in abundance upon administration of DR7, also showed negative correlations with DBH and positive correlation with TPH2. Meanwhile, for Actinobacteria, which showed a lesser increase in abundance upon administration of DR7 as compared to the placebo, also exhibited a negative correlation with TPH2. So all these indicated that the administration of probiotic Lactobacillus plantarum DR7 exerted gut microbiota changes which not only remain in gut but affected and positively or negatively but significantly correlated with neurotransmitters genes along the gut-brain axis. So there are many emerging studies in these years that have reported on the effects of gut microbiota on serotonin and dopamine levels, and also correlate those with psychological effects. However, to our best knowledge, this is the first study on probiotics to correlate between gut microbiota and genes that are related to serotonin and dopamine pathways, which are not only significant but also consistent across multiple taxonomic levels. So this ends my talk, and in summary, Lactobacillus plantarum DR7 showed gut-brain axis benefits in smaller models of flies and rats all the way to human. This strain has been patented in Malaysia, China, Korea, and also PCT. This strain is stored in depository in China and Korea. Whole genome has been sequenced, and human data is available on stress, memory, cognition, upper respiratory tract infection, and also gut health. So if any of you would like to collaborate with me to work further on probiotic strains along gut-brain axis, please visit my website. You can also contact me, and I welcome all levels of collaborations. And this is an aging study. I wish to thank every one of you, your time. The conclusion is I don't mind aging. We all shouldn't mind aging as long as we age healthily. So ladies and gentlemen, thank you very much.
I would like to thank Professor Minza Leong for breaking the ice and starting the scientific part of our conference. I can see many young people in the audience, but I'm pretty sure there are many more young people who are with us virtually. So please take a good home message. If you're in stress, if you are to approach your exams or tests or the final exams, certainly you may or should consider taking probiotics. What I also took as a general take-home message, without going deep into the details, and it was very interesting, that the probiotics should be considered as age-dependent type of the biologically functional applications available for the humans. So it's not like every probiotic works for everyone. We should really think carefully. I don't know if I can draw these kind of parallels. Some people say that certain things help them against the headache, and other people find that different kind of medicine help them. So probiotics, some of them work on the broader field of challenges, and some work on the specific fields of challenges. And again, this was also pretty clearly shown in this presentation. I was also very much impressed seeing how the investigators moved from these cute little flying objects. I'm of course trying to be humorous, from the fruit flies to the rats and finally to the humans, and we can see continuity of the research, the history and the connections, and how the investigators are going from the more primitive organisms to the more organized and finally to us humans. And we see the confirmation of each of the steps with moving up in the hierarchy of organization of the living creatures. As I mentioned before, we have some participants who are with us here in the audience, and some who are away but digitally connected. All of us can ask questions. Those who are watching us on YouTube, you can also write your questions. Please don't forget to indicate your name, affiliation, email address, and forward your questions to the organizing committee. If they will not be answered immediately, certainly we will try our best to deliver your questions to the speaker, and the speaker will also answer them. At this moment in time, I can see Min there with us. I would like to ask the audience if you have any questions to the presenter. Turn on.
Okay, thank you very much, Mike. Can you hear me?
Thank you very much for a great presentation. I feel a little bit shy asking you because it was so broad and fundamental research. So I have two little questions. First is related to the rat model. Did I get it correctly that statin wasn't good for the rats in these experiments? And that was the first one. And the second, how strain dependent is the effect of lactobacilli on these neuronal effects which you was monitoring?
Thank you, Professor Suvorov. I would say that it's my honor to finally meet you, although virtually. Just a quick check. Can you all hear me?
Yes, yes, I can.
Yes. I was blasting the YouTube link to many of my ex-students, and I've been using your name as a fishing bait to fish more audience. I like the fact that you're developing these edible vaccines. And anyway, back to your question. The first question on rats, no, actually statin worked. Statin as a drug control worked in reducing depression, anxiety, and stress in our animals. And even some of the factors, the lactobacillus DR7 was not that effective as compared to statin. So there are some of these data that we didn't publish. But in general, yes, statin did work. Your second question on strain specificity, it is not just strain, but it's also species. We had screened, I cannot remember the real number, but we have screened many strains, different species, and even within the same species. And not all would show all these kind of effects that we want. So when we first started with the fruit fly, as we can see with DR7, the composition of Wolbachia reduced tremendously. We didn't see that with many other lactobacillus strains, even with different species and within the same species. So based on that, we selected certain strains, and then we tested with rats, and that's how we ended up with humans.
And talking to this, are you planning to kind of go down to genomics and look at the specific gene patterns which are involved in this kind of phenomena? I'm afraid you didn't hear me.
We don't have the expertise right now. But if you have these expertise and you would like to collaborate, by all means, I welcome that collaboration. I was previously approaching several others, including Mike, so that we can proceed further along that line. And also proteomics, which I'm very interested in, but without that expertise.
I think it will be very nice to start collaboration. And that's the main meaning of this kind of conferences. So thanks for the organizers. It was nice talking to you. Thank you very much.
Thank you. My pleasure.
I would like to thank the presenter. If anybody else have questions from the audience, I don't see any questions. And those of you who are with us online, please, if you have questions, you can, as I said already, send them to the organizers, and we'll make sure to pass these questions to the presenters. We have a few more minutes, and I will just have one last, more fundamental type of a question. You mentioned that, and of course that's what we know, but I'm really happy to see it emphasized today, that the effect is strain dependent and the species dependent. So, and at the same time we see some movement towards the suggestions to consider certain species as probiotics or certain species as being grass. And of course, to be politically correct, I would like to ask your personal opinion on this matter, because and I am not hesitant to express my own opinion, saying that I think indeed we should consider every microorganism as an individual, putting it in quotes, and looking at its beneficial effect depending on a particular creature that we have in our hands. But I would like to hear your professional opinion, especially based on this really enormous work that you have done and presented today.
Thank you, Mike. Actually, I share the same sentiment, and I've been saying this over and over again. All of us here, whether attending physically or virtually, we are of the same genus and species. We are Homo sapiens, I hope. So, but we are all so different, and that's the conclusion of it. We are all the same genus and species, but we are different strains. So we all have different capabilities. And it is high time already that we really go for, along these pathways right now for personalized nutrition and personalized supplement, that we should not categorize all probiotics to be the same. There is no one size fits all. All of us, they are doing fundamental science, see that day in, day out in our lab. All these different strains, different species will have different capabilities, I would say. So like I've mentioned just now, when we are screening, even with a small fruit fly, even with the same species, we already see differences. So in the advocation of using certain new probiotics, certain putative probiotics to be included as probiotics and as grass, I think that goes with many regional regulatory confirmation and requirement, and that's needed. Because from the safety aspects, from the benefit aspects, that needs to be proven. And grass, for example, grass is not a blanket cover. Grass don't just go for one specific even species or strain. It goes by the end product. So there are a lot of things that we have to compare with and get sufficient data with before we confirm a certain strain as a beneficial probiotic.
Thank you, Minza. Thank you very much for your opinion. And gosh, I miss all our previous meetings where we were able to talk face to face, and I hope this time will come soon again. And I would like to again repeat the words of the rector of the Don State Technical University to welcome you here once all these things that are finished, dust is settled. We will have the summer school in Rostov, and then we will finally see each other and work together in flesh, so to speak. Thank you very much again for your participation and for being a president and the founder of this absolutely outstanding event. Thank you very much. I would like now.
Mike, and
thank
well
you.
done to our Russian team.
Thank you. I would like to give floor now to our next presenter, Dr. Ilyin.
So, distinguished colleagues, first of all, I should like to give a special thanks to Professor Chikindas for inviting me here. And I want to speak about the threats which expect the piloted space missions, traditional threats which is also exist while International Space Station flight, and expecting threats which expect us while fulfilling of interplanetary missions, and the means and measures to somehow to prevent the disorders which we can be faced with. So I work in the Institute for Biomedical Problems. I'm the head of the large department. Department actually of habitability, which consists of 5 laboratories. 2 of them are dedicated to microbial problems, and if 2 large collectives are devoted to this problem, so we can evaluate acuity of the problem. 1 is devoted to problems of environmental microbiology, and the other is laboratory of human microbial ecology, as we call it. So our institute was founded in 1964, just 3 years after the first flight of Gagarin, and therefore we together. So we are like a famous jazzman, Louis Armstrong says that I and jazz were born together. So our institute and the space flights, piloted space missions, also born about together. Let's start from traditional threats. So traditional threats are the same as we face with the clinics, maybe because it is a semi-closed objects like clinics. And here, but in spaceflight conditions, the habitability are mostly closed, and so we have seen here. Anyway, we can see here traditional threats, which is normal for closed collectives for a long period of time. It's first of all intensive microbial and plasmid metabolism exchange during acute adaptation and period. It is let's say it is the first 10-15 days of coexistence of the humans which are in the chamber, and they very actively exchange their microflora, and as a result we can faced with this formation of strains looking like nosocomial ones. So we can and more people are collected and in these closed habitats, so more more evidence that these strains can be formed, and they be formed without any selective measures like antibiotic admission or something like that. Then we have we have the other very serious problem, which mostly devoted to the environmental situation. So when we have the very acute, very heavy contamination level of the environmental subjects, like, for example, non-metallic constructions, cable net, and water supply system in certain conditions or something like that. And we have there a large niche of inhabitants, and this niche includes also human pathogens, like, for example, the pathogenic fungi, as well, like Aspergillus niger, Aspergillus flavus. And also we think this is the other very acute threat, as we say normally. It comes from the natural sources. Let's just suppose that in Baikonur it is just coming from the soil, from the wind, during the final preparation studies, and they, having been coming to the close objects, they have nothing to do either to die or to develop the other niches which is unnatural for them. And so they colonize the organic materials, which in non-metallic constructions and cable net, it's very acute problem. More acute for Mir station rather than International Space Stations, because we can see all the lessons which heavy lessons which we obtained from Mir station. We have tried to solve it while preparation of the ISS. So as a result of microbial and plasmid interchange, we have faced with pathogenicity potential of human microflora. So we can increasing of the strains which possess the aggressive enzymes like DNase, RNase, for example, proteinase, phosphatase, hemolysin containing strains, and as a result we are faced with increase of pathogenicity potential. And as a result, because together with lack of protective microflora, together with lack of immunity barrier, together with lack of the physiological features of covering tissues, we have free barriers weakened, and as a result we have, as we call it, colonization-resistant disorder syndrome, which is normal for the long-term isolation. In different it is connected with the space flight. It's connected with deep saturation diving, and the other things where the people spend in the confined objects. And new challenges also we can appear facing with interplanetary studies. For Martian mission, we can face with chemolithotropes which can settle human organism. If we have methane on the Martian surface, so we can face with metagenetic or metanotrophic microorganisms which can easily colonize different kinds of human biotopes. So it depends how they will behave afterwards. So we can see how the soil microorganisms behave on the environment of the space station. They change their trophical preferences very easily, and so how they work during the Martian mission, we don't know so far. And the other is Moon. This is the other story, because in the Moon we plan several expeditions. So the first expedition must, if we use the life support systems in certain levels. So let's thinking of the lunar bases. So anyway, the microbiota of the first mission will be anyway stored and left in the lunar bases. And what will happen with this microbiota having been subjected to the heavy radiation, to low magnetic fields? We are now studying it in our in vitro studies. We start these investigations in order to think what changes will obtain by microflora, which is natural for human beings, phylogenetically natural, but what changes they will obtain and how they will behave, having colonized the other organisms, so the organisms of coming crew, next crew. That is the essence of the problems. And the third and very interesting threat we have faced with having performed together with CNIMASH the space experiment test, when the cosmonaut during EVA is extravehicular activity will come out, taking sample from the outer surface of the ISS, and afterwards that very samples was subjected to testing, and we have like that, we have like it is on the picture, have done, and what we have found, we have founded different kinds of bacillus. Different kinds of bacillus species which form colonies. Actually, it's nothing to do with bacillus. It's absolutely innocent microorganism. But let's suppose about the threats. If there will be, if the trajectory of the flights will be more far from the Earth, so it is the evidence that maybe we will faced with some microorganisms which we don't know, cryptic microorganisms, how they will behave during EVA, extravehicular activity, after if the cosmonaut will take it by the spacesuit and return back to the living closed objects, and how they will behave there. That is also the very interesting problem. Meanwhile, they were very tolerant. We took this bacillus, one of these Bacillus pumulus. We use it for the interesting experiment called meteorite. So we put it in the hole of the stone, which we fixed in the outer surface of the biological space satellite. And leave it. It started and then returned. The other we have made about 200 inclusions there with different thermophilic microorganisms, and only this microorganism survived. Any other were born in the dense layers of atmosphere when this satellite returned back. So it is very tolerant microorganisms, and the problem is very acute. So yes, it's so this is the results of our experiment test, but it is prolonging, and we're expanding our investigations to taking. So we will possess our catchers just more out of the docks or the locks where the cosmonauts make their EVA, in order not to think about that this is the contaminants from the inner part of the ISS. So, and we for investigations of the activity of probiotics, because we elaborate certain means and measures for prevention of colonization of the humans with the microorganisms. So we make certain investigations in our block. It's the investigation center, which is connected with several locks. So there are several chambers for living, for working, for physical training, and this is devoted to this object is devoted to it is simulate Martian surface. So during certain time they come out, they go outside and go to this object and spend there for several weeks during the long-term studies like Sirius. 4 months experiment during the Mars 500, when they spent about 520 days during isolation and certain others. So this is other vision of this our unit, NEC, наземный экспериментальный комплекс. So, and this is some illustration. So this is to illustrate how easily the microflora of coming crew spent within the basic crew members. So it is 90 days isolation, where we have one basic crew consisting of three people and one visiting group, and one of the visiting group contained this E. coli strain. We marked it, and within 10 days it spread through all the isolated individuals. So, and the same story was during the end of this mission, when also the same situation took part. And this is illustrated the story of the spread of the strain, which is in connect of one of the volunteers, which spread on the 7th day among all of the volunteers. So it is just as it works as so it is the classic isolation without visiting group, and again we're faced with this story. So, and we have some visions which about the increasing of pathogenicity potential, and increasing of amount of positive probes, samples in confined habitats concerning alpha hemolytic streptococcus, for example, or just the other, it is Staphylococcus aureus again increased during the experimental period. So the other story is about the changes, increasing of pathogenic features within one and the same species, which we have isolated during one and the same experiment. So we can see that the Staphylococcus aureus pathogenicity increased here, and Clostridia as well. So the amount of E. coli which possess the hexanuclease activity increased 2 times within the acute isolation period. And the interesting story we have on the dental microflora of cosmonauts, where the Veillonella, which normally treated as protective microflora, is absolutely in all the flights is changed by the straight pathogens of dental system. It is Prevotella, Actinomyces, and Fusobacterium. After the flight, all of the biotopes were colonized by this microflora. Again, it is very interesting that the people who have very frequently participate in this experiment, so they are mostly tolerant to the changes of microflora than the newcomers. And here we have the volunteers, which microflora has weakened very much, and the microflora, intestinal microflora, higher respiratory tract microflora of the people who very frequently attend these experiments with long-term isolation, they remained on the very high level. So we have see here we have used here eubiotic index. So we investigated it to easify the calculation of the effect of probiotics. So it is the rate of changes. It's rate of positive changes of microflora to negative changes of microflora changes while obtaining of the probiotics. And if it is more than one, it is of course positive, and it is less than one, it is negative. And therefore, this way we are we're testing it. The same story is in intestinal tract, like high respiratory tract, within 17 days isolation period. So here we have what we have think about how we treat the colonial resistance syndrome, and this is our modest effects of different kinds of probiotics on microflora. So there are two kinds of investigations. The investigations of the new strains, new collection strains. It is mostly concerns the Carinobacterium pseudo diphtheriticum, which has a marvelous effect on the carriership of the Staphylococcus aureus, which consequently decreased to the real vanishing through the period of isolation, and increasing of Carinobacterial microflora instead. And the other concerns the investigations with Streptococcus salivarius, K12, which also lead to consequently decrease of Staphylococcus aureus in throat. Unfortunately, the impact of these microorganisms on dental system was were more modest. However, we expect better situation, but nevertheless. And this drawing concerned the consequently decrease of pathogenic microflora in intestine during the experiment Mars 500, when it is consequently decreased during the consumption of Enterococcus faecium, of auto strains of Enterococcus faecium, which the volunteers obtained within the first 60 days of isolation. We can see consequently decrease of this microflora even when after the consumption was stopped. And afterwards, afterwards it increased, of course. There was a real jump. And this stimulation afterwards to the very end of the experiment was caused by obtaining of absolutely probiotic drug which obtained from the drugstore. So yes. This is also and our modest attempt is to combine the autoprobiotics with the food products, to combine probiotics with sour milk product or with special fermentation drink made on the basis of Saccharomyces cerevisiae or Saccharomyces boulardii. And this is the result of our studies. This is 120 days isolation studies, and here the blue one is the group which obtained autoprobiotics with the fermentation drink containing Saccharomyces boulardii in concentration of 10⁻⁷. It's not bad in comparison with the control group, so we have our eubiotic index rather more effective than they have in the control group, which do not obtain the autoprobiotics based on the based on this fermentation drink, but they obtained the preparates from the obtained from the from commercial probiotics based on Linux studies. So we here can see that the amount of the opportunistic microorganisms are very low in the group which obtain these autoprobiotics based on the drink containing the Saccharomyces cerevisiae, and how much we have faced with the control group which didn't obtain this drug. Dry immersion studies formed the other group. It is very interesting group because it simulates the weightlessness. There you see the person which is laying on the basin on just floating on the basin, and this way we obtain the sort of simulation of the weightlessness. Here we also faced, for example, with increasing of Staphylococcus aureus. This is archived data, but nevertheless we have seen the overgrowth of Staphylococcus aureus in the throat for all the isolated people, and we have offered them the stripes. It is collagenic stripes enriched with their lactobacilli, and which we put just on the gut like that, and we have while dissolved this material, this liquid spread through all the mouth, and this way we got the decontamination, we decreased to the real vanishing of Staphylococcus aureus. So this we have. The conclusion is that we must work further with searching for new strains, searching for new mode of consumption of the strains, new food products. We have new coming experiments. One of them we expect in this autumn, and it will prolong for 8 months, and we look very much forward to the to fulfillment of what we have to do, because nowadays we will work as follows. So we will freeze the material which we obtained on the background, and afterwards we freeze to -80. So this way we simulate a sort of expedition on the outer planets, in which we have some disasters with their drug kit containing the probiotics from collection, let's say. And in this case, what to do? How to act? So to refreeze the fecal material or the other materials which we will store, including because we have also the women crew as well, so we put there the vaginal microflora and cervical canal microflora. And after refreezing, we will prepare the drug, maybe in the form of the tablets enriched with the microbial culture, and they will offer them to our volunteers on the final stage of these investigations. So this is our plans, and my report is over. Thank you very much for your attention.
Thank you, Dr. Ilyin. I will abuse my power to ask first questions, and please keep in mind we have just a few minutes left before the next presenter will start. I have a few questions. You mentioned that more than one types of microorganisms were found, and first thing that struck my attention was the spore-forming bacilli. And in some of your experiments, particularly number 35 and number 42, there were more than one species of bacilli. And we do know that these are microorganisms that have a natural ability to exchange genetic information, especially when they're going to sporulation and vegetation again after sporulation. Did you do the experiments, or you plan, or maybe you have some information? Was there any gene transfer mentioned in, or maybe you?
We have performed the studies of the plasmid mobilization and simple conjugation and transduction in the spaceflight conditions. Concerns Bacillus thuringiensis. It is not from that pool which obtained with this experiment with SNEIMAGE. And they have a slight increasing of frequency of mobilization and simple conjugation.
Yeah, that's indirectly but answers my question. So these should be considered as a challenge, a possible challenge.
Yes.
The answer is yes. And the second question is, you noticed the higher pathogenicity of the microorganisms in these particular environments. And higher pathogenicity to me is an indication of the microorganism to be prepared for the next step of invasion. Does it mean that what we see with the eukaryotic host is that some of the protection mechanisms become weaker, and the signal is sent to the microorganisms that they can, start taking this ecological niche again and deeper?
We have the destiny of this spike, which obtained during this first decade of isolation, is the following: it decreased. It decreased together with the indices of antibiotic resistance, which as well decreased, because nothing to do, because there are no selective
pressure.
Yes, pressure. Therefore, from generation to generation, the strain containing plasmids try to get rid of the plasmid, and we can face with the classic flights, space flights on Salyut 7 indicates the restoration of antibiotic susceptibility. That's it, and together with the associated genes with pathogenicity. But as soon as they they come out in the earth,
it's all restored.
Of course.
Thank you. And the very last one, quick question. We have to keep on time. With the eubiotic index, on the day 120. It goes to 2.5, and the control remains at the level of 1.0. And this is statistically significant.
Yes.
Can you interpret biological significance?
You mean in the first isolation period?
Yes.
Yeah? Because it dealt with our dream is to depress this very pike, which obtained in the first isolation period, yes, in the acute adaptation period, because this pike lead to auto-infection and cross-infection, and our idea is to suppress it by
as much as possible.
Yeah, by any means.
Okay. Thank you very much for your presentation, Dr. Lin.
Thank you.
I give a floor to our next presenter, Dr. Alexander Suvorov.
Good morning. It's still morning. First of all, I want to express my gratitude for ability. Different one, please. To talk at this wonderful conference, and I want to thank the organizers, yes, for the ability to present some of our ideas about personal microbiota and bacterial therapies. So it is partly philosophical, partly kind of a chance to share what we are doing in our Institute for Experimental Medicine in St. Petersburg. And going ahead of time, I just want to tell that we'll have a conference in October this year, maybe also devoted to beneficial microorganisms, and I will be happy to have you in St. Petersburg in our city. Okay, so today I wanted to talk on outer probiotics. Professor Lin was speaking a little bit about it, so it's easier for me, and just reminding you that microbiota opened to us out after appearance of the new technologies like new generation sequencing. Now we know that a microbiota of human beings is very broad. It is different in different parts of the human body. It's very individual. There is Professor Rumogam, who first discovered that microbiota of each human being is specific to this human being, that there are 3 enterotypes of gut microbiota of all the humans. And also now we know that microbiota is not developing immediately, and it kind of gets stable after 1,000 days of life. And after that period of time, we carry our set of bacteria in our bodies which help us to live and be healthy. And each one of us, and this is a study of Professor Budding from Holland, each person has highly individual microbiota pattern which allows him to kind of cope with all the problems of common life. I just want to show the picture of the article of Professor Blazer from New York. He is one of the most vigorous advisor for and supporter of ideas of manipulating of microbiota, showing that during the life period of time, the life is changing with a set of bacteria in our bodies, and when this change happens dramatically, we are getting in the pathological condition which is called dysbiosis, and it is extremely important to make a shift from unhealthy pattern to the healthy pattern. So, but at present times, we know that infections, which used to be the main cause of death of the human race, now we have other somatic diseases such as cancer diseases, heart diseases, immunopathological diseases, metabolic diseases. But when you look deeper, it turned out that all these diseases in reality start developing because of the changes of microbiota. And a lot of problems which we have in the common world are appearing due to this set of dysbiotic conditions. This can cause kind of neurological diseases, immunopathological disorders, and a lot of gastrointestinal problems. Basically, everything boils down to so-called low-grade inflammation in the gut with the appearance of a lot of pro-inflammatory signals. which help to develop the dysbiotic condition and is kind of manifested by production of certain pro-inflammatory cytokines, which can finally cause a lot of disease, including cancer, type 2 diabetes, and so on and so forth. So this low-grade inflammation can be in the biofilm of the gut microbiota or even inside human epithelium. People now know certain cytokine patterns which can distinguish between dysbiotic condition with prevalence of pro-inflammatory cytokine expression, or in health condition there is equilibrium between regulatory cytokines and pro-inflammatory cytokines. So there are a lot of signaling in the body caused by this low-grade inflammation, wrong bacteria in case of dysbiotic condition, and everything boils down to diseases in dysbiotic situation like gastrointestinal diseases, obesity, type 2 diabetes, type 1 diabetes, atherosclerosis, Crohn's disease, and so on and so forth. I don't have enough time talking about that. And now we know very well, and there was a first lecture which brilliantly showed that our brain condition also depends on condition of our microbiota. In implementing probiotics, you can influence the functioning of the brain. So-called gut-brain axis depends on the condition of our gut and our intestinal microbiota. The same with Alzheimer's disease, which is a disease of the elderly. And now we think, okay, now we know microbiota is important. Let's look at certain bacteria and we will find the golden button and say, okay, oh yes, Eubacteria is higher, Proteobacteria is lower, and now we know how to cure all the problems. No way, because if you look carefully just at this article, I don't have enough time for that, but you will see that different researchers, I'm sure all of them are decent people, are getting absolutely different results, and the final review just say, up to now, no study confirmed clearly that deposition and tau accumulation is related to ultra-particular microbe in the gut of Alzheimer's disease patient. It tells us that there is a lot of things to do before we will say, okay, you just get the sample of this bacteria and now you will be with great memory, healthy, and jumping like Valery Brumel, you know, if you remember old days of world champions of Russia. So now all the concept of contemporary medicine are changing. Before it was easy. We take antibiotic and treat all the infections. Now you kill pathogens, you cause dysbiosis condition, and you get a set of different diseases, including cardiovascular diseases and cancer diseases and diabetes and so on and so forth. So we need now to think of microbiota from the perspective of ecological situation, because only very little things now is more or less established, not for 100%, but more or less. At least we know that proteobacteria are usually related with pro-inflammatory signaling and so-called low-grade inflammation they produce. and the toxins which are supporting this low-grade inflammation in the gut. So if we look in the gut concentration of the gases, so you don't want too much oxygen in large intestine. It is very important. You don't want to have too much NO2. So there are certain kind of viruses which people are looking at, and a lot of things which we don't know. But what we don't know, we need to fight dysbiosis, and the most rational way of fighting dysbiosis is to restore the microbiota. So as long as dysbiosis is a trigger for the diseases, in order to fight the diseases, we need to do something. So this is wonderful lady McFarland, and she wrote in the article in 2014 that recovery of normal microbiota is defined as restoration of microbiota back to normal healthy baseline. And recovery can be different, can be complete or incomplete, but basically what you need to do is return microbiota to the base level. So this is another article from Mary Sanders on probiotics. If you add probiotic, the idea of probiotics is to make the dysbiotic condition shorter, or to lower down the pathological effects of dysbiotic condition, because the mechanisms by which probiotic can work are very multiple. So it's very hard to know and understand in all these different variants of actions of probiotic, how do they work. But again, what we see at the final stage is lowering down the pathogens, lowering down this low-grade inflammation, and restoring the normal microbiota. It can be done by different ways, like in our case, we work a lot with enterococcal probiotics, which produce a lot of bacteriocins. There will be a lecture on bacteriocins later on, which can do kill bacteria, or they can change this cytokine production level of in the intestines and the organism. And interleukin-10 is going up in different models. We already showed that, and it is important that pro-inflammatory cytokines after taking out the probiotics go down, and anti-inflammatory go up. Another approach is to do fecal transplantation. Now it is very popular approach. A lot of people do this kind of taking donor microbiota, giving it to person with dysbiotic condition, Clostridium difficile infection, something like that. And then you can kind of do so-called fecal microbial transplantation, FMT. There are a lot of different protocols how to do it. But you have to know that if you start manipulating with microbiota, it is filled with a lot of mines in it, because I just warn you that you shouldn't be too optimistic, because kind of usage of bacteria, because bacteria don't have personal sympathy for us as a human being. They need just to propagate and be kind of healthy inside our bodies. So they, in certain kind of conditions, and this is a list of different pathological effects of microbiota and different mechanisms by which a microbiotic can cause problems to the host. And in order to have this kind of ecological attitude for this subject, we started investigating another approach, which helps you with the knowledge that microbiota is highly individual and immune system is getting adapted to the immune system in, let's say, 1,000 days. Eventually there is so-called equilibrium, because personal microbiota and this kind of immunological response to this microbiota. So there appeared the idea of getting autoprobiotics. So what are autoprobiotics? They are bacteria which you take from your own organism. It can be fecal material, it can be skin microbiota, oral microbiota, vaginal microbiota. You can grow it outside of the human body. And what is important, you don't use it as it is, because even personal microbiota can harm the organism. But you do genetic analysis of this individual strains, and only after genetic testing, then you can grow the strain for each individual and use it for treatment of dysbiotic condition. This is a model on the rats. You can see that if you cause dysbiotic condition just by antibiotic, microbiota becomes very, very much destroyed. It becomes kind of very not diverse. So you need to diversify your microbiota after microbial treatment. And we use just 6 different variants of autoprobiotic based on different strains, different consortiums. So if you look at this picture, you will see that in blue, this is microbiota after dysbiotic condition. And if you use different kinds of autoprobiotic strain, can be bifidobacteria, lactobacilli, enterococci, mixtures of different bacteria, you can see that alpha diversity is coming back, and you can see that there is a change, like from antibiotic treatment, the composition of microbiota is very unique. If you don't do nothing, it starts changing after antibiotic, but can't return to normal condition, and different autoprobiotic can help you to return the microbiota to normal. So this is pretty much the same usage of different kind of autoprobiotics, like with lactobacilli, with enterococci or bifidobacteria. You can return to the normal condition. And returning to the animal model, we have a lot of different experiments to show, but I just wanted to show you the example which is related to the first lecture of the professor from Malaysia. And this is the rats with dysbiotic condition. In cross labyrinths, they are very passive, they don't move anywhere. We use autoprobiotic, and they start investigate the labyrinth, they start activity, so their brain functioning improves dramatically. And we can see a lot of positive effects on physiological activity. Finally, we thought, okay, if it helps to animals, why not to help people? And eventually we ended up to build up a first complex in the world which helped to take individual microbiota from human beings, analyze it with genetic testing. We have algorithms for different kinds of bacteria. We can store individual bacteria in the biobanks and then make so-called autoprobiotic products for the people in the powder form or in the milk fermented forms. And we can try to treat dysbiotic condition. We started with irritated bowel diseases and found out that we got fantastic results with the patients with IBS condition. The condition dramatically improved, not only in the gut, but the healthy level was improving. And then we started looking at different diseases. And it turned out that even in case of inflammatory diseases, we also get very good results on the condition of the patients. Even infiltrations in the lungs was dramatically improving. And if you look at the interleukins of the patients, you remember the animal model, we get exactly the same in humans. So we are animals, yes. And the quality of life of these patients was also improving. It turned out that autoprobiotic technology was extremely, and for me it was very kind of emotionally important, very important for cancer patients because they are suffering. They are suffering after chemotherapy, they are suffering after surgery, and one of the major causes of the problems after surgery is Clostridium difficile infection or this kind of gut inflammation, especially in case of colorectal cancer. But you can see you give autoprobiotic with personal bacteria which we take before the surgery and give them after the surgery, we can dramatically support their life. And we can dramatically decrease the number of pathogens and increase the number of health beneficial bacteria. So there is not time for it, but we can see that health beneficial bacteria pattern is dramatically improving. And autoprobiotic is not only for the gut. It's not only for colonal cancer or something like this. It's also, the strategy works for oral bacteria. You saw that in previous lecture of Professor Lin, but we have done the algorithm for oral treatment of chronic oral diseases like periodontitis. You also can isolate certain kind of bacteria outside, grow them outside of the body and treat the oral condition with autoprobiotics. So it's already prepared for patenting this kind of technology. So you can do very many things with autoprobiotics, like gallstone disease, chronic infection, vaginal infection. We could take vaginal microbiota, prepare autoprobiotic from vagina and treat vaginal infection. We have big study on Parkinson, irritable bowel syndrome, H. pylori infection, and cancer. Now we are in big project. Our institute is involved in so-called National Center of International Level for using autoprobiotic technology for treatment of metabolic diseases like type 2 diabetic diseases and cancer diseases. So, just by ending my talk, I just want to summarize what do we expect from autoprobiotics. We have to do more studies on different loci of human body like gynecology, dentistry, skin diseases. We have to make biobanks of human microbiota in order to quickly restore microbiota in case of necessity. And I hope that such center as we developed will be developed throughout our country, helping people. I think it is very important because you can do this kind of logistics of microbial therapy being really personalized. So you can address to biobank, you can prepare personal microbiota quickly, and perform the medical treatment of pathological condition. And finally, I want to thank you and say my cheers for healthy microbiota and microbiome. Thank you.
Thank you, Dr. Suvorov, for the excellent overview. I particularly appreciate the balance in presentation between the literature overview and how you gave us all the idea of what is happening in your research, in your field, and in your laboratories. We have very little time. If anybody from the audience want to ask questions, please, it is the time to ask a question. Yes, please, there is one.
Hello, my name is Alexey Vatlin. I am from Vavilov Institute of General Genetics. And I have only one question. Can you say about your approach to select strains from microbiota of the host? You said that you have some approach for selected specific strains for then use it for auto probiotics. And what is approach? You said some genetic approach, as I understand, right? Yes. Or what can you say about this? Thank you very much.
Yeah, thank you for your question. You can address our papers like we published in Frontiers Microbiology. It's available for anybody. But in short, I can tell you that it depends on the strains, kind of on the species which we are playing with. For example, we have a very good experience with enterococci, and some of enterococci, as we know, can cause infections. So in our case, we have genetic tests based on PCR and real-time PCR, which can help you to trace for certain pathogenicity factors. So we select several clones and analyze each one of them by PCR, and only those clones which don't express those virulence traits are selected for the further work and growth for the patient.
Thank you. Are there other questions, please? One more.
Can you hear me?
Yeah.
Can you hear me?
My name is Eli from Preparatory Faculty. I have just one question. Can a human survive a day or a week deprived of any intake of microbes?
I'm sorry, I can't hear you.
I said, can a human survive a day or a week deprived of any intake of microbes?
Can you hear?
Sorry, I can't hear you. Oh.
Yeah, it's hard. It's
hard to hear you.
Okay. Can a human survive a day or a week deprived of any intake of microbes?
The day or week of taking the microbes?
Deprived.
The what?
Deprived of any intake of microbes. Without. Just one day without microbe.
Oh, I see. Yes.
Your question is whether you can survive without microorganisms.
Yeah.
Yes, you can, but eventually we are surrounded by microorganisms, and eventually either you will get microorganisms. Let's say we can make a person sterile. It is kind of hard, but I suppose it is possible, but then you will be absolutely subjected to a lot of different pathological situations. For example, gnotobiotic animals, and you can make animals without any bacteria, like gnotobiotic. You can put them in the open world, they die very quickly because of infections. They have to be in certain sterile condition. In this case, you can kind of keep them alive. So bacteria are making a lot of things for us. It is very long story, but I mean, they produce vitamins, they produce a lot of metabolic products for us, like short-chain fatty acids. They fight pathogens for us. So basically, we are the complex of different organisms. We are not just human beings. We are human beings with our microbiota, and we can't do anything about it. So.
Thank you. We have one question. I hope we will be able to connect the international question from Dr. Santosh Tiwari. If we can turn on Zoom, please. And Santosh, I can see you.
Can you hear me, Mike?
Yes, I can hear you.
Thank you very much, and Dr. Suvarov, for presenting a very nice talk. Just I would like to interact that it is very nice to see you after Prague here. And just I wanted to ask you that what is the biosafety level of these autoprobiotics, especially the fecal matter transplant? Very quickly, if you can suggest us, what are the biosafety aspects they take care when they transfer this fecal matter transplant? Thank you.
This is one of the advantages of this technology, because if you do fecal transplant, you have a lot of stuff in it. You have viruses, you have eukaryotic cells with retroviruses, and tell me about it. In our case, we are selecting clones which are genetically analyzed and grown outside of the body. So, this technology was developing quite slowly, because being a medical doctor, by my education, I know that the major task for medical doctor is not to cause any harm to the patient. So my experience, and we have this technology functioning slowly but surely for about 10 years, we never expected and experience any side effects regarding the health condition. And this kind of studies were always supported by the authorities of Russia, and they were highly monitored, and the results were analyzed. So I think that this is the advantage of autoprobiotic personalized treatment relatively to fecal transplantation or any kind of transplantation or implementation of bacterial therapy.
Thank you, Dr. Suvarov, but don't leave the podium yet. We have also the question from Dr. Hasenberg, and I hope we will be able to connect. Yes, Dr. Hasenberg.
Thank you. Greetings from Vienna. Congratulations. Very nice idea, very nice talk, and it's a really exciting approach. May I ask you how you deal with strictly anaerobe bacteria? Is it difficult to handle them in your concept?
Thank you, Professor. It is nice to see you healthy in good condition. And I like Jena very much. It was the first university, international university I met. I visited and done some studies in CIMET. regarding the anaerobes, it's harder. We started doing that already. We have certain equipment for this kind of experiments. So basically, this is the answer why we started with easier models to begin with. But I think in the near future we will be able to tell you more. But in our model we have a so-called anaerobic consortium. I didn't have time to talk about that, but what we do, we can take the set of the microorganisms from the individual, grow them outside of his body in the anaerobic condition on the artificial media, and then we are doing some genetic testing, what we get after growing them. Usually we grow them, now we were analyzing it for a couple, actually 2-3 years already. So now our algorithm is growing them for 6 days, changing artificial media with certain additions. Then we test this anaerobic microbiota and provide them for the recipient of microbial therapy. It works quite well, but we want to explore it more.
Thank you.
Thank you, Dr. Suvorov. Thank you for your really inspiring presentation, as we can see by the number of questions also coming from across the world, Austria, India. Thank you very much. We have now a presentation from Professor Koen Venema, Maastricht University, the Netherlands, and he is also the chief editor of the journal Beneficial Microbes. I have a warm heart for this investigator. He and I spent 3 years in the Netherlands working at the same bench together, and we started these two journals pretty much at the same time. He is one of the lead scientists in the probiotics area in Europe, and I would say without hesitation in the world. And it is an honor and pleasure to have him presenting today. Thank you, and we have a presentation from Professor Venema.
Hello, welcome to my presentation on personalized prebiotics, opportunities and challenges. Let me start by explaining who I am. My name is Koen Venema. I have a chair in gut microbiology at Maastricht University, but I am also the initiator and organizer of a conference series on beneficial microbes, which obviously aligns with this international conference on beneficial microbes, where we started in 2008, had one conference in March, and the next one is going to be in October next year. Also, I am editor-in-chief of the journal Beneficial Microbes and founder and CEO of the consultancy company Beneficial Microbes Consultancy. And I trademarked the phrase beneficial microbes. So that's who I am. Let's switch to the science. As you may be well aware, the microbiota has been implicated in a number of different diseases that can be wrong with us. And clearly, since they are located in the gut, it's not surprising that they are related to diseases for colon cancer, inflammatory bowel disease, and irritable bowel syndrome, but also more distantly in the body, such as allergy of skin and lungs, or things like obesity with associated type 2 diabetes, cardiovascular disease, and things like that. But also autoimmune diseases like type 1 diabetes, and even how old we get. And intriguingly, a lot of different things that can be wrong with our brain, such as anxiety, stress, and depression, brain development and cognition, and some diseases of the brain like Parkinson's, Alzheimer's, and autism. Now you may be wondering what those two pictures of those rats in those bowls are for. Well, these are in fact little videos of a rat model for autism, and this research has been done by Derek McFabe in Canada. And what he did was inject a single metabolite that is produced by the gut microbiota directly in the brain of these rats to study their behavior. And on the left you see a control rat, on the right you see a autistic rat, and the control rat is happily exploring its environment as rats normally do, as you can see in the video, while the rat that has been injected with that single microbial metabolite shows repetitive behavior. It will keep on running in these circles until that metabolite has been dialyzed out of the brain or has been metabolized, and then it returns to more or less a normal rat, although there are phenotypic changes in the brain of that autistic rat. So you can appreciate how the gut microbiota also is involved in things like autism. So how to study the gut microbiota? Well, this is normally what we do in human individuals. We look at fecal samples. But then, what do we look at? Which microbes or metabolites are important in health and disease? And does the composition of the gut microbiota matter, or is there enormous functional overlap between different species? And does your enterotype or the number of genes that you have in your microbiota determine whether you are healthy or not? And is there a difference between people with, let's say, a different blood type? And we'll get back to that in a second. And should therefore prebiotics be personalized based on the presence or expression of host receptors, as these host receptors co-define microbiota composition? And I'll see if I can give at least my own opinion on that with some research that has been done over a couple of decades. It actually all started out with research by the group of Willem de Hosse at the Wageningen University, where they determined that the host genotype actually does affect the bacterial community in our GI tract, as you can see in this plot here. If on top here you take a single person but study the gut microbiota in time, so with temporal variation, you see that the similarity index that has been defined here is approximately 80. If you look at monozygotic twins, which are of course genetically identical, you see also that the similarity index is relatively high. Then if you look at partners that are married, you already see that that similarity index significantly drops. And but they still have the same households, they probably eat the same things, so that actually gives at least still some similarity in the gut microbiota. But then if you look at completely unrelated persons, you see that that similarity score even drops further. And if you compare humans to other primates, then there's almost no similarity left. So the host genotype actually does affect that bacterial community in the GI tract. Now, if we look at blood type, which can be characterized as ABO or ABH, depending on what typing scheme you like, then these oligosaccharides coat red blood cells, and the structure of these oligosaccharides actually determine whether you have blood type A, B, AB, or O or H, and the structure here is provided. So depending on which oligosaccharide is coating your red blood cells, you have a different blood type. But the same oligosaccharides that are on your red blood cells are also present on the surface of a number of mucosal epithelial surfaces, such as the respiratory, genitourinary tract, and the digestive tract. So since these are present there, they define perhaps gut microbiota composition by binding of these members of the gut microbiota to these receptors, or by the use of these oligosaccharides as a carbon source. And as the sugars are different in the different blood types, as indicated again here at the bottom, your microbiota composition may actually be different too if you have a different blood type. Now on top of that we have what is called secretor stage and Lewis factors that also define composition of oligosaccharides on all kinds of receptors. And the secretor stage is defined by the presence of a gene called FUT2, and the Lewis factor is depending on the presence of a gene called FUT3, which actually decorates oligosaccharides on receptors with galactose, N-acetylglucosamine, and fucose. Now, to make matters worse, if you have a particular sugar, depending on the presence or absence of these genes, you may have all kinds of coated oligosaccharides that are present on receptors that are expressed throughout the body, including the GI tract, and therefore that may define your gut microbiota. So in a study that we did already a couple of years ago, almost a decade ago, we were interested in isolation of blood type specific bifidobacteria to use them for personalized nutrition. So what we did is we took fecal samples from healthy volunteers, and those were run in a gut simulator, and I don't have time to explain that model in detail, but fecal samples were introduced in the gastric compartment of this model, and then samples were taken from the end of the small intestine, and we plated for surviving bifidobacteria to see whether there were differences in species and also in survival of these bifidobacteria that were dependent on blood group secretostatus. Now, the use of these saccharides as a carbon source from blood types is something that I will discuss in the remainder of this talk. Now, microorganisms use carbohydrate-active enzymes, or cazymes, to degrade carbohydrates. And there's a really beautiful database called the KZ database that has information on all of these genes that are from all microorganisms, bacteria, fungi, and whatnot, that are involved in degradation of these fibers. Now, I'd like to focus on glycosyl hydrolases here, but there are also glycosyl transferases, polysaccharide lyases, carbohydrate esterases, and also some auxiliary activities that are required to break down carbohydrates. And if you look at some carbohydrates, and this is just a simplistic view of the complex nature that carbohydrates are, then for instance here we have a pectin molecule with all kinds of different monosaccharides, with all kinds of different linkages between it, with branching, and already a number of different enzymes are required to break down this complex carbohydrate. So we need esterases, we need hydrolases to cleave the bonds between sugars. So microorganisms should really have a lot of these different enzymes. Now there are there are more than 130 different glycosyl hydrolase families which are based on amino acid sequence. At the time that I prepared this presentation, there were over 200,000 different modules in that KC database, and they can be grouped in clans on the basis of their three-dimensional structure. But one family can actually contain different enzyme activities. So I've show here, for instance, the glycosyl hydrolases clan of related families, where these families have numbers that identify them. Now, I've just gone into that KZIM database, and as an example, I clicked on glycosyl hydrolase family 3, where you see that there are actually a number of different known activities that are present within this family. So beta-glucosidase, xylan 1,4-beta xylidase activity, and all of the other activities that are listed here. Now, there are 7,116 different glycosyl hydrolase family 3 entries in the database, some for archaea, some for bacteria, some for eukaryotes and things like that. And I clicked on bacteria, and then you get a long list of different microorganisms in which this family 3 has been shown. And this is a wheel that shows actually some of these enzymes and where they have been found. Now, of course, we are interested in beneficial microbes, so I scrolled down to Bifidobacterium, and you can see that in Bifidobacterium adolescentis, the ATCC strain 15703, there are actually a number of genes present that belong to this glycosyl hydrolase 3 family. So if you click on that again, and here is family 3, you actually see that Bifidobacterium has a number of different glycosyl hydrolases within different families, on top of the transferases, the carbohydrate esterases, and carbohydrate binding module families. But it has 6 different genes for glycosyl hydrolase 3. And most of the time you can actually find that there are many families that contain a number of different genes that encode that family, although there are also a number of families that just have a single gene. So the list of proteins is provided in that database where known, but mind you, this is in silico prediction of a gene sequence. So where it says here that this is a beta xylosidase C, there is probably no biochemical evidence that this is actually a correct prediction of the activity of this particular gene. Now then, if you look at B theta or Bacteroides theta iota omicron, this particular strain, which is isolated from human feces, it actually has 268 different glycosyl hydrolases divided over 47 subclasses, and on top of that it also has some glycosyl transferases. So those are listed here, and I'm sure you can't read that anymore, but 268 different enzymes to break down complex fibers. So because of the large number of enzymes, it can break down numerous oligo and polysaccharides, including mucin. Now I list here another microorganism, a Lactobacillus casei, and that only contains 32 different glycosyl hydrolases. So that's an order of magnitude lower than B theta in only 14 subclasses. And I actually looked at the ratio between glycosyl hydrolases and transferases, which is approximately 3/1 in B theta and is approximately 1/1 in KCI. Now, because of the more simplistic and lower number of glycosyl hydrolases, it was easier to model that for LKCI. But for B theta, it is very difficult to determine the activities of all those different enzymes in silico, let alone in the lab. Now, what are the challenges with respect to the prediction of the function of these glycosyl hydrolases? I already alluded to that a little bit. First of all, these families are based on amino acid sequence similarity. This helps to reveal the evolutionary relationships between these enzymes, and it provides a convenient tool to derive mechanistic information. But it is very difficult to derive relationships between family membership and substrate specificity, as, for instance, already indicated by the known activities of enzymes within glycosyl hydrolase family 3, where all these different activities actually are present. So amino acid sequence similarity does not necessarily mean that you know the specificity of the enzyme. I forgot I had these animations here. Moreover, enzymes from different glycosyl hydrolase families can actually have the same predicted activity. So here we have enzymes from family 5, 8, 10, 11, 7, and 43, which all have xylanase activity. And even within a single enzyme, there may be two different activities, as is indicated here for a family 3 enzyme that has a beta glucosidase activity as well as a beta xylosidase activity. Now these have probably been tested experimentally, but it doesn't mean that this enzyme cannot have more than these two different activities. Probably simply because it hasn't been tested biochemically, we don't know whether or not that is the case, which I indicate here, so perhaps even more than two due to overlapping substrate specificity. Then we have family 5, which actually contains 53 subfamilies, as indicated here by this colorful wheel, with a very broad sequence diversity, which actually very much limits functional prediction of this family. Now, apart from challenges with respect to glycosyl hydrolase predicted function, we also see community specificity. So in this elegant paper in Cell from a couple of years back, the group here inoculated germ-free mice with communities that were isolated from a number of different origins, from humans, from zebrafish, termite guts, soil, and microbial mats. And they analyzed the glycosyl hydrolases in those communities that established in those germ-free mice. And even though the communities came from a completely different origin than from the mouse itself, they actually did see that there was a community specificity also with respect to glycosyl hydrolases that were present. They actually had a number of different samples from humans, from feces, from the tongue, and from the skin, and also there you can already see different glycosyl hydrolases that are present in those different communities, which is of course not surprising because every community probably gets exposed to different substrates that should be degraded, for which you need different enzymes. Then on top of that, there is actually genus and species specificity. Different species harbor different numbers of glycosyl hydrolases, glycosyl transferases, and pectin lyases. And for instance, species within Bacteroidetes that ranges from 15 to more than or approximately 450. So several orders of magnitude difference there. So clearly some of these bugs are geared better towards breaking down complex fibers, including mucin in our GI tract, than others. And this here shows some of the variation that you can see in this Bacteroidetes phylum. And again, this is probably too small for you to read, but you can clearly see the difference in size of these bars. And to make matters worse, there's even a strain specificity. Again, perhaps not surprising, but strain level identification is not something that we usually do on a large scale currently in next generation sequencing of gut microbiota samples, simply because we cannot get to that level of specificity without deep sequencing of those samples. So here in this +1 paper, the group of Bernard and Rissat actually looked at two different strains from Ruminococcus knapus that both can use a monosaccharide that is present in mucin, but only one can actually grow on mucin as a sole carbohydrate source. And that is because both strains actually have fucosidases in the family GH29 and 95 that are required, for instance here this is 95 and 29, that are required to break down mucin, but only one of the two has this GH33 sialidase that allows it to grow on mucin as a sole carbon source. So even within the species Ruminococcus cnavis, there is differences between different strains. Now, if you look at our own human genome, that encodes 97 glycosyl hydrolases. If you look at the human genome, there are estimates that this is more than 20,000 genes. So most of these genes are active on plant cell walls and animal glycans and present in different GH families. Then they are active on starch and glycogen, and the prevalence of these genes and their activities likely reflects the diet of the gut microbiota, or in other words, the fraction of the diet that we ingest that actually reaches the colon and that can then be used by the gut microbiota. So this large green circle is the number of enzymes for plant wall glycan degradation, then animal glycans, and here starch and glycogen, peptidoglycan, and here in blue it says other substrates. So people have also been interested in what they call the core caseome, related to research whether there is a core microbiome, in other words, microbes that are present in everybody. The idea existed that maybe there are some genes present in the gut microbiota that everybody needs based on the diet that we have. Now, unfortunately, there are only 8 stool samples that were taken along in this study, and the reason for that is that it's an enormous amount of work to characterize these samples, in which 318 enzymes were found, divided over 46 families, but only shown 19 here in the legend. And what the authors found there was that actinobacteria actually contributed approximately 30% of the genes that existed in the core, or in other words, were present in all these 8 stool samples. And Bacteroidetes only contributed approximately 5% to that core. However, you should realize that this does not mean that the bifidobacteria make up most of the microbiota composition and Bacteroides very little. In fact, that was very much the other way around when they looked at the microbiota composition. It just means that for Actinobacteria, for the phylum, some of these genes are actually present in a number of different individuals. Now, since this is a conference on beneficial microbes, I dug up some data on bifidobacteria where people looked at the response of bifidobacteria to prebiotics and then actually looked at genes that were involved in degradation of these prebiotics, again looking at glycosyl hydrolase families and here glycosyl transferases. Using proteomics, so not one of the most high throughput methods, but certainly one that is very specific and that you can investigate in a great amount of detail. And they found that some enzymes that were induced in bifidobacteria were actually specific for galacto-oligosaccharides. Others were specific for the human milk oligosaccharides that are present in breast milk, and yet again others that were specific for inulin or fructooligosaccharides. And from that, they modeled carbohydrate degradation by these bifidobacteria and the enzymes and transporters that are responsible for that. All right, that brings me to my take-home messages and points to keep in mind. And one of the first one is that individualized microbiota might be actually the reason for responder, non-responder effects that we usually observe in clinical trials. And you should also expect significant overlap in microbiota activity due to functional redundancy of the gut microbiota. So personalized prebiotics, as the title of this presentation mentions, might actually unfortunately be as specific as the current commercial prebiotics, although my belief is that if we do research on these glycosyl hydrolases and their specificity, we may find some more specific prebiotics that can be used to stimulate specific microbes in the gut microbiota. That may lead to different prebiotics for obese and lean individuals, but for that we still need to do a lot of research. Also, different prebiotics may be required for different blood groups because the gut microbiota of these people is different due to the presence of these oligosaccharides on receptors in the guts. Other points to keep in mind is that everybody has his or her own microbiota composition, which is responsible for health and disease. So in my opinion, personalized prebiotics are required to individualize the health of these individuals. And the biggest bottleneck towards personalized prebiotics is the prediction of these enzyme specificities for glycosyl hydrolases, but also other enzymes that are involved in fiber breakdown and their efficacy. So towards personalized probiotics, as I called this presentation, is a concept that holds promise, but requires a significant amount of more detailed information about the gut microbiota and its members, and particularly the enzymes that are involved in the breakdown of these fibers. So for more information, I've listed here my email address. With that, I'd like to thank you very much for your attention. And I hope that there is time to, in fact, answer some questions, even though this is an online conference. Thank you very much.
I would like to thank Professor Venema for his very interesting and in-depth presentation. I'm not sure if he is with us today because he mentioned that he will be busy at this day and time. If there will be any questions, please send them to organizing committee and we'll make sure to pass the questions to Professor Venema. Our next presenter is Professor Wilhelm Holzapfel, and I have a great pleasure introducing him and his presentation. We know each other for quite some years, and we got acquainted at several international meetings. I believe the first one was ICOM. So again, many thanks to Minze for introducing us to each other. So without any further ado, I give the floor to Professor Holzapfel and his presentation.
Hello, good afternoon or good evening. I'm not sure at which time it will be in Rostov-on-Don when my presentation is being shown, but it is evening here in Korea. And I just want to thank the organizers very much for the kind invitation to give this presentation at the ICOBM-2021 conference in Rostov-on-Don. We, myself and many others, were looking forward actually to participate physically, but as we all know, it was not possible. That's all will be there physically, and I'm grateful that we still have the online option of participating and presenting. Before I continue with my talk, I just want to point out that Puang is a city on the east coast of Korea, about just less than 500 nautical sea miles away from Vladivostok, which is the closest Russian border from here. So it's not very far to Russian border, but maybe still a far ways and a distance to Rostov. This is a beautiful campus. As you can see, in the springtime we have beautiful cherry blossoms and also the azaleas. And if you overlook the campus, you can see the Eastern Sea of Korea. And this is a little bit outside of the city of Pohang. The title of my presentation is Perspective for Beneficial Lactic Acid Bacteria, with focus on fermented foods. Now I will actually shorten my version of my presentation, realizing that I have to fit it in half an hour. First of all, I think we have to realize, and this is what I tell my students so many times, that we have to realize when we're dealing with microbes, doesn't matter in which context, always we have obviously an interaction with kind of different ecosystems, but we have to remember they don't read our textbooks. But they are masters of conversion, transformation, stabilization, and adaptation. In this way, they play a key role in so many processes on our planet and in our ecosystems, and also determining many aspects of conversion and recycling. But I think we can also say then when we deal with microorganisms in the context of interacting with, for instance, fermented foods or even in our intestinal tract, we always have the good and the bad, and they might be fighting each other, but then we have the humble, which actually to for my definition would refer to those microorganisms from which we do not know their real role in many kind of ecosystems. Some we have not studied sufficiently, we cannot even cultivate all of them, and so we have always, I would say, a mixed population with so many diverse interactions and also processes like we could call cross-feeding and also antagonism, synergy effects, and whatever. Now, the concept of biotherapy is actually derived from food fermentations, and that goes back many, many centuries in history. In fact, we can even go to the time before Christ when the Roman historian Pliny considered the administration of fermented milk, that it could be beneficial against gut infections. Also, the Greek medical scientist Galenos said that the consumption of yogurt is beneficial against gastrointestinal tract disorders, and so we can continue through history that the benefits of fermented foods have been appreciated and also promoted by means of specialists, even medical scientists in the earlier times. And so we still have, until present day, we have traditions that continue worldwide. And in most cultures, the fermented foods, traditional fermented foods, are actually considered very valuable. And so we have an example, which I will also give some information of from studies on the Maasai tradition, because until today the Maasai people have continue with an old tradition of consuming fermented milk in a traditional way, which are fermented in calabashes, and they consider this still as very healthy and also beneficial for protection against ailments such as diarrhea and constipation. And we know even until the 20th century we have great names in history like Mechnikov, also Vergio, who actually was the first to introduce the term or definition of a probiotic as opposed to antibiotics, and that was introduced in the early 50s when the realization came up that antibiotics also kill the good bacteria. So we have a very interesting development in history, starting from fermented foods until our present time, when we have better tools of investigating the interactions and the complexity in many food ecosystems. So we can say that until today we have still a highly valued valuation of traditional fermented foods in many cultures. They present very interesting study models, and for us, even in a modern context, they are still very interesting sources of what we can call strain prospecting or taking expeditions into the micro. So we know that fermented foods, and I don't have to repeat many of these benefits, they are very highly valued in most cultures, but also today we know much more about their role in restoration of gut microbial balance in different ways, and also understanding much more of underlying mechanisms. So the lactobacilli or lactic acid bacteria in general, they are usually dominating most food fermentations. Not all, but I think they are still very interesting for deeper studies in finding beneficial strains for application in what we could call today even biotherapy or therapeutic approaches. So we know that the lactic acid bacteria, they are maybe dominating even the probiotic market until today. But we find, and that is, the target for application of probiotics is mainly the human gut, or also for animal applications. But lactobacillus, representatives of the, I would say, the range sensu stricta, the former Lactobacillus genus, which is now actually subdivided in 23 new genera. These lactobacilli, they are not detectable in about 25% of human fecal samples. However, most of them we can consider passing through but having a beneficial effect or a mediating effect in an indirect role and in different ways, and that is why the study of beneficial activities in vivo in the context of probiotics and the human gut is not just a straightforward aspect or an issue, but I think it needs different kind of approaches and different models. So, but if you look at the development in the probiotics market here in the US, for instance, and the projections for the near future up to the year 2023, we can see that there's a clear continuous increase in the requirements or demands for probiotics, and that will include also finding new strains, maybe even new beneficial properties of strains or in combinations and in mixtures, which will be better than the probiotics that have been used so far. And you can see in the blue, it actually reflects the probiotic food and beverages market, which is still the by greater portion of the whole. And one other aspect is very interesting, and that is the paradigm shift we find in global sales of infant formula, where we had the conventional or traditional infant formula market, which is shown in blue, until 2019, which was dominating the market, but moving from the conventional to, I would say, a new approach or a new paradigm based on the fact that the realization has also now, I would say, penetrated through the whole industry, that the probiotics, I would say, supplementing the infant formula would have additional benefits. And we can see there's a so-called crossing over point in between the years 2019 and 20. And now, from 2020-21, that seems and is expected also an increase that will continue in the probiotic infant formula market. And that also underlines the importance that we should find better strains, more safe strains maybe, and that may be better applicable also to infants. Now I just want to give examples of what I would like to call a tale of two species, and also in this way emphasize the importance of interspecies diversity. The first species is Lactobacillus rhamnosus, which is now called Lacticacibacillus rhamnosus, and I want to compare here a strain which we isolated from Maasai milk, Maasai fermented milk, quite many years ago by a PhD student from Kenya, which we call the BFE strain, BFE 5264, and comparing it with a well-known commercial strain of Lactobacillus rhamnosus called LGG. The other example would be Lactobacillus plantarum or Lactobacillus plantarum, which we compare also with the LGG as a, I would say, a kind of a golden. Now, we know that the lactic acid bacteria, especially Lactobacilli, even the former genus, has a huge range of different species and also with different genomic sizes, from very small genome with very strict adaptation like the Lactobacillus sanfranciscensis, with a very small genome, up to the two largest, or some of the two largest genomes, which is represented in Lactobacillus rhamnosus and Lactobacillus plantarum. And I want to just refer to these species specifically in the further presentation. So, and here we have always to remind ourselves of the importance of intra-species diversity. That means not all strains of a particular species will do the same job and have the same exact features. And in this case, it's also reflected here in the, for instance, the example of LGG rhamnosus and the BFE strain, with a slightly different genomic size, a slightly larger size from the strain that was isolated from Maasai fermented milk as compared to the LGG originating from the human intestine. And I will also refer to HACO1 in the second part. Now, we also did a whole genome, full genome sequencing of Lactobacillus 5264, compared it with LGG. We found some differences here, and some of the major differences were actually reflected in the metabolic pathways. Of some of these pathways were not found in LGG, but were present in this Maasai strain, the BFE strain. So I think most of us know about the Maasai tribe in Eastern Africa, herdsmen, and they mainly consume fermented milks 3 times a day, actually, and they ferment this milk in calabashes, as you saw on the left, see it on the left-hand side. But very interesting is that these people have very low blood serum cholesterol levels, in spite of the fact that they consume full milk up to as a major diet and up to 3 times a day. So we were interested about these strains. Obviously, we isolated quite many different species and strains from the Maasai fermented milk, including Acidophilus and Plantarum and so on, but we also found some of these strains very interesting, and one strain of Lactobacillus rhamnosus was very interesting for us, and we started to do further studies on this strain after selection, and we actually first did some in vitro studies, and we were obviously intrigued by the question about the cholesterol reduction effect or hypocholesteric effect of the strain in vitro cell line studies. We used different cell lines like the THP1 cells, and in this case we also compared it with a Plantarum strain, which we isolated from kimchi. In another study, we used CACU2 cells, and what we found is actually that in this in vitro cell line study, we could show that there was a reduction in the cholesterol absorption in the CACU2 cells, and the mechanisms behind it appeared clearly to be based on the downregulation of NPC1L1 and also the upregulation of the LXR, which is responsible for the reflux of efflux of cholesterol in the epithelial. So this was followed up by in vitro studies on the cholesterol lowering effect. This was done by a former PhD student, Dr. So-Young Park. And again, and in this study, we compared this BFE strain from the Maasai with the very famous LGG strain, which was originally from. Now, in this study, in the mouse study, we use the C57BL6 male mice, and we had a low fat, a low cholesterol diet, high cholesterol diet, and then also one group received the high cholesterol plus the BFE strain, and the other the LGG strain, and also another group received the statin. And in this slide, you can just see a summary of the outcome of this in vivo study, by which we found that in vitro observations and results could be confirmed by the in vivo study by showing that cholesterol absorption was reduced based on the NPC1L1, and thereby improving cholesterol homeostasis. Also on the other side, by the increased cholesterol efflux based on the increase or the upregulation of the liver X receptor LXR by the strain of the Maasai as compared to LGG. I'm sorry, it's covered here with this kind of presentation. I'm not so used to it, so it moved on faster than I expected. So this is the LGG strain, the white bar here, and the one in the middle is actually the Maasai strain, where we can clearly see that the observations obtained in the cell line study could confirm here. Slide. I just want to remind about the heterogeneity of this species Lactobacillus plantarum, which is now called Lactobacillus plantarum. This is based on a former study and published by Kleinhammer and colleagues, where you can see a clear, huge diversity based on the first ring, the base deviation index, and also in the DNA variability, etc. What is interesting is that in Korea we have, I think, one of the most famous traditional fermented foods, which is meanwhile, I think, well known all over the world, which we call kimchi. And there are at least 300 different recipes, and that obviously is the basis also for finding different species and strains during the fermentation process. So we have a huge intra and intra-species diversity, but we know from studies so far and also many publications that some dominating species are actually Lactobacillus plantarum and Lactobacillus sakei. Now, we isolated one specific strain, which we found also after safety testing. We had actually a series of Lactobacillus plantarum strains, which we isolated in a government project, which we called White Kimchi Project, and several strains were plantarum and several others were sakei. And we found that one of the most promising for further studies was what we called HSCO1, and we studied this strain further following different selection studies in vitro, and also what we call the SSTP in vitro model, by which we compared the survival of strains by passing the upper intestinal tract, and we found that this strain showed very promising characteristics. So after this in a preliminary isolation and selection, we were interested in the impact of this strain on the gut microbiota. As you know, if we have I would say, disturbance of the stability in the gut microbiota, we call it dysbiosis, and we want to see if it would modulate the gut microbiota in a favorable. And so we again had a mouse study, the same kind of mice as before. Overall, in 8 weeks after 3 weeks of stabilization, we started the feeding. We had, in this case, a high-fat diet, and this is part of our studies and a general interest in what we call metabolic syndrome. And one aspect, obviously, is the obesity issue. And so in this case, we had a control, high-fat diet with just PBS, and then 2 feeding groups, one with the HCCO1 and one with the Rhamnosus LGG as a reference strain. And we analyzed the digesta and looked at the bacterial RNA to determine which are the active microbiota, and then also studied the cDNA by pyroseq. And we found actually, I must mention that we used 3 different levels of strains applications. We call it LP1, 2, and 3, and for the plantarum strain, and LG 1 and 2 and 3 also for 3 different levels of application of the LGG strain. We find small shifts in the population, but we find bigger differences obviously when we compare the different treatments here. We found that alubaculum was impacted in different ways by the LGG strain on the right hand side and the Lactobacillus plantarum in the middle, the black bar here. Also interesting is that we could finally confirm that the modulation of the gut microbiota was quite in a different way if we compare the high-fat diet with the high-fat diet where Lactobacillus plantarum was applied and also where the LGG was applied. So the modulation we consider was therefore in a species or even strain-specific experiment. The next question was about the alleviation of obesity and metabolic diseases and looking at particular biomarkers again in this feeding experiment. We had the same kind of approach and setup with a high-fat diet and also rhamnosus as a strain or as a standard and compare this with the impact of HSCO1. And what we found is that the whole body weight of the feeding period was significantly reduced in terms of the increase in body weight, but especially we found that the mesenteric fat mass was significantly lower in the group that received the LP, that the plantarum strain, and not significantly receiving the Lactobacillus rhamnosus LGG strain. Oh yeah, now I managed to go back. Yeah, I just wanted to point it out, with this kind of presentation you have to be alert what's going on here. So we have the plantarum strain that actually increased or upregulated the lipid oxidation biomarkers and downregulated significantly the lipogenesis biomarkers. Therefore, altogether, the administration of this plantarum strain changed expression levels of lipid metabolism associated markers. And in this way, one could clearly see that it had a very positive impact on reduction of obesity and also related issues to metabolic. Then we also looked at the mRNA expression levels in the mesenteric adipose tissue. And again, to summarize this, what is very interesting is that especially if you look at leptin and adiponectin that go in diverse directions, and when you take their ratio, they actually showed a very clear picture here, and that is that the adiponectin-lectin ratio significantly increased in the plantarum treated group, but not in the other groups. And in this way, the plantarum once again made a significant impact on obesity and I would say in the indirect way also for alleviating issues related to metabolic. So also we looked at adiponectin and leptin levels in the blood serum, and again we found the same tendency in the blood serum, so that the plantarum strain increased the serum adiponectin level and the leptin level was reduced. So in this case, the beneficial effects of the plantarum strain could again be confirmed, also as compared to the LGG. So when we look at the metabolic syndrome, we consider all kinds of aspects related to metabolic diseases as related or that will cause what we can call systemic chronic inflammation. And obviously the approach should be to reduce the pro-inflammatory responses and to increase or to promote anti-inflammatory responses. And this is what we tried also to find out in this experiment. And we found that, for instance, TNF-alpha, by the way, this is based on mRNA expression levels. This was reduced, and also relative IL-6, which is a pro-inflammatory cytokine, and also IL-12. And on the other hand, we found that the anti-inflammatory cytokine, interleukin-10, was significantly increased, and so also FOXP3 and also TGF-B. And in this way, the anti-inflammatory responses were promoted or increased or improved by feeding this plantarum strain. And this, to our understanding and to our hypothesis, this, by feeding this strain, the balance will be found back, or by counteracting pro-inflammatory response by the anti-inflammatory responses, also based on an impact on the gut microbiota. And so the question is, what are the underlying factors, or what other factors are still associated with these changes? And we also found, and this is obviously a condition that is associated with inflammatory conditions, and that is the tight junction protein biomarkers. That means also the leakage in the gut is also associated with several intestinal conditions. And we found in both cases that SO1, and in this case also oclidine, measuring both of them, they were significantly increased by the feeding of Lactobacillus plantarum. And this way we could show that the tight junction protein expression was also increased. So when we summarize altogether, is that by feeding this specific plantarum strain, we find a change in the gut microbiota by significantly beneficial modulation. Then the gut barrier function was improved. The immune response was reduced, and also inflammatory response was decreased. That means by also counteracting through the anti-inflammatory cytokines. And in this way, I think this strain has a good potential for alleviation or use in evaluation of. So in conclusion, we have very interesting models in our modern day time still, and I think we can still look for many fermented foods which provide such interesting study models, and by which we could also look deeper into particular strains and look at their particular functions. We need, obviously, not only the fermented foods, but also improved models by studying particular strains and their interaction, and for selection. For instance, in vitro and ex vivo study models, and in vivo. And obviously this is still a great challenge, because how do you select the best strain out of many strains within one species in a quick way, and without always having just to go to a final in vivo or even then clinical studies? Then we still don't know very much about some functions of metabolites, especially when we look at the complete fermented foods. We know that there are also pleiotropic effects, and these effects can be related not only to the particular organisms involved in a fermentation, but also in terms of their metabolites. And so one of the key metabolites that can be also of great importance is lactate. It does play a role in brain functions and in the immune system. Then obviously the issue about stress induction in a substrate, like for instance in fermented foods, we have a series of stress factors, and in this way some of our strains could be potentiated to be better adapted also to the intestinal tract. So, and we can obviously use a range of technical factors also for improving activity and functionality, also in terms of what I would like to call stress-related factors or stress response factors and improving functionality. Then obviously we have to remember about inter and interspecies diversity. A great challenge, as I see it, and I think maybe for young scientists especially, would be to take up the approach of finding fastidious strains, of studying them, and those which we so far concerned as unculturable, and to study their role not only in fermented foods but also in the intestinal tract. So I conclude to say that fermented foods still represent a rich source for present-day strain prospective. I just want to refer and gratefully mention the great financial support we received from the National Research Foundation of the Korean Ministry of Science. And this is just a glimpse of some of our team. And I want to thank you very much for your attention.
Thank you, Professor Holzappel, for your very interesting presentation. As always, I noticed several things that struck my attention. First of all, the fact that your study involved not just the microorganism of your interest, but also a reference strain. Certainly you know, and many of us know, how few papers published where the investigated microorganism is studied side by side with the reference, putting in quotes, generic probiotic. And I think this is particularly interesting. Another very interesting thing, and again, this is something that was already said today, is your mentioning that the effect is probably species, but more even probably strain specific. Another thing that I also noticed, and perhaps this is just my imagination and you can correct me, when you were talking about the pro-inflammatory and anti-inflammatory effects observed and how they change through the course of using certain probiotic, the studied strain, in your investigation, we saw some kind of a seesaw play where I immediately imagined that pro-inflammatory effects that are present, they also play a certain significant role in the overall health of the eukaryotic host. And to me, seeing the seesaw effect was a reminder of possible importance of pro-inflammatory factors. And of course, last but not least thing that I noticed is the very good collection of the biological markers, significant biological markers that your team selected to follow the entire story and to see how your studied microorganism performed and what exactly it is doing on the molecular level, and how this is translated on the level of the health of the eukaryotic organism. So these were really shining points of your presentation that I noticed, not the only ones of course, but there were many few else. I was told that you're with us today online, and I'm very happy that you're with our community during this meeting, and I would like to ask the audience, also those who are present in person and perhaps online, if there are any questions to the presenter, please raise your hand. No questions in the audience. Are there questions in from the network? No, no questions. So that was really very comprehensive presentation, and happens so that you know, some of the speeches leave no questions, not because they're not interesting, but because they're actually answering many questions, and because they're also providing a lot of food. Actually, as I was speaking, Dr. Suvorov has a question.
I think that it is not good when there is no questions after such a wonderful lecture. Thank you very much. It was nice to hear you. My question is very silly. Okay, you found the strain which is isolated from kimchi. What will be following? Are you thinking of making kimchi just with this strain? Because I would suggest that 300 different variants of kimchi carry different strains, and not just this one. Can you comment on that, or how much kimchi I need to eat to have a good health and wonderful memory?
Dr. Hausafel, are you with us? Because we see your icon saying that you are present, but we cannot. No, probably not. Probably not. So we will pass this question to Dr. Hausafel. And just to add a few pennies to this question, of course we're talking about functional food. Of course we're talking about the food that can deliver, positive effect to those who are consuming this food. So this is also very interesting and important direction in the science and applications of beneficial microbes. Sorry for using this term because I know that it was trademarked by Professor Vainema. We're giving now floor to our next presenter, Dr. Laptev, and his presentation will be in Russian. But what we're going to do is we're arranging translation. So once our conference is finished, the translation will be provided, and then on YouTube we'll have the option of Russian and the option of English language for this particular presentation. I believe most of the slides are still in English, so those of us who are not Russian-speaking people will be able to follow it. Thank you.
Thank you, Professor Chikindas. Our chairman, Dr. Minze Leong, said morning that most part of participants is from Russia. So also my English is not so fluent, so I prefer to speak Russian. Дорогие коллеги, настоящее, мой доклад посвящен изучению метаболических путей в рубце у крупного рогатого скота и экспрессию генов метаболизма на разных стадиях лактации. Вот это моя ученица Елена Александровна Йылдырым, фамилия турецкая, потому что вышла замуж за турка, привезла себе из Турции. Вот она защитила докторскую позапрошлом году, и она вот на фоне НЖС-секвенатора. И она является, я буду докладывать о результатах, которые получены большим коллективом, с ее участием Ларисы Ильиной, Евгения Бражника, Андрея Дубровина, Валентины Филипповой. На этом слайде корова, и у коровы до 100 литров занимает рубец, где происходит микробиологическая обработка корма. И он населен порядка 300-400 филотипов бактерий, более 30 видов грибов, простейших 40 видов, метаногенных археев, еще больше, чем здесь указано. И особенностью, если мы хотим, чтобы наши пробиотики широко применялись в сельском хозяйстве, в животноводстве, надо представлять себе, какое оно у нас. Вы теперь редко можно увидеть корову. Иногда бывают даже вопросы, куда делись коровы, почему мы их не видим. Но они стоят на фермах, на стойловом, на содержании, и за пределы не выходят. Они в основном галштино-фрисской породы, черно-пестрые. Они очень требовательны к корму. и рационы их, и они вообще-то доят. Вот, скажем, средний удой по Ленинградской области – это около 9 тысяч тонн в год. Вот, это больше, чем в Финляндии, или больше даже, чем в Германии, ну или на одном уровне примерно все это скачет. Ну и вот ведущие молочные хозяйства России, ну вот там очень высокий уровень удоев. Но беда заключается в том, что вот эта высокая удойность, вот, она привела к очень низкой продолжительности хозяйственного использования коров. Корова, высокопродуктивная корова живет вот хорошо, если 3 года, а то 2, и она не успевает даже окупиться. И вот есть, ну, есть разные версии, в том числе вот, я сторонник той, которая связана с микрофлорой рубца. Ну вот здесь показано на этом слайде, чем отличается энергетический баланс у коров. Это самый первый период лактации, вот когда это сухостой, когда корову не доят, потом она рожает и начинает стадия раздоя. И вот в это время происходит вынос энергии гораздо больше, чем корова потребляет. То есть она попадает в некий вот, период отрицательного энергетического баланса. Ну вот это наша лаборатория. Вот так мы берем рубцовую жидкость с помощью зонда. Вот и тем, у кого брали желудочный сок, знают, насколько это неприятная процедура. Ну вот нам приходится это делать у них. Ну вот здесь показаны данные о численности, значит, с помощью real-time ПЦР бактерий, грибов, архей. Мы очень долгое время занимались тем, что пытались найти связь между молочной продуктивностью и микрофлорой рубца. Здесь показано для разных бактерий, и для лактобацилл, для лахноспир, вот, упоминалась виолионелла, очень интересная бактерия, и фузобактерия, которая вообще-то, вот, хотя она является нормальным обитателем рубца и нормальным обитателем половых путей, но ее избыток приводит к развитию заболеваний. Вот я хочу сказать, в правой части слайда, значит, вот по каким причинам выбраковывают коров в хозяйствах. Вот в основном это низкая продуктивность, когда вдруг животное перестает доиться. Заболевание вымени, заболевание конечностей, но все это так или иначе заболевание, нарушение обмена веществ вследствие вот этого ужасного дисбактериоза. Значит, когда при увеличении доли концентрированных кормов и когда в рационе мало сена, ну вот человек одомашнил корову, но он не мог одомашнить ее микробиоту. И она действительно существует такая, какая, вот, чтобы переваривать сено, естественная корма, ну вот траву в естественной среде. Но так как вот высокопродуктивные коровы, у них зачастую в рационе больше 60% концентрированных кормов, то есть зернового происхождения, там очень много крахмала, и это приводит к увеличению содержания амилолитических бактерий. Повышается количество молочной кислоты, снижается pH, снижение доли, вот, причем это снижение pH приводит к снижению доли целлюлозолитических микроорганизмов, как грибов, так и бактерий, и дальнейшему подкислению. И вот в этой вот среди бактерий, способных к усвоению лактата, вот с одной стороны есть прекрасная виолионелла, которая переводит его в пропионовую кислоту, и это используется для синтеза жира. Но с другой стороны, вот эта самая нехорошая фузобактерия, которая способна прогрызать стенку рубца, попадать в кровь, разрушать печень. И все такое. Ну, понятно, что микрофлора рубца связана с микробиотой других органов коровы, с половыми путями, с копытами. Ну вот если это фузобактерия, которая их сильный протеолитик, вот, кто был на крупных комплексах, видел, что вот, там гидросмыв, там ходит скрепер, который перегоняет с места на место эту жижу. И зачастую вот они просто ходят по этим фузобактериям, что приводит к образованию язвы. Кроме того, вот такой показатель, как численность соматических клеток в молоке, это свидетельство о мастите. Такое молоко бракуется. Ну вот оно тоже связано с многими бактериями. Связано с наличием стафилококков в рубце. Ну, я хочу сказать, что вот если раньше мы отвечали на вопрос, вот, какие бактерии в каком количестве связаны с какими нарушениями, с какими рационами, значит, то вот сейчас появилась возможность, значит, перейти от вопроса, кто же обитает в рубце, к функциональному профилю, какой у них метаболический потенциал. И существуют вот возможности с помощью баз данных, той же КЕГ, Киотская энциклопедия генов и геномов, это воссоздание отдельных генов кластеров, связанных с синтезом ферментов, и воссоздание метаболических путей. Есть такая программа PICRAST-2, вот как этот летчик Руст, который много лет назад сел на Красной площади. Вот так же пишется. Ну вот можно, в принципе, ну вот просто от каких-то свойств микроорганизмов перейти к наличию у них определенных генов и синтезом определенных ферментов. Вот, ну вот здесь примерно все это написано. Реконструкцию генов и путей, включающих в себя деградацию, утилизацию полисахаридов, пептидов, крахмала, идентификацию генов и путей, включающих в себя синтез витаминов, метаболические пути трансформации ксенобиотиков и другие. Значит, ну вот здесь, причем есть возможность увязать это с потреблением корма, среднесуточным приростом или удоем. Ну вот здесь используется графа корреляции, и можно вот тоже вот вносить это. Ну вот первым шагом исследования является, естественно, значит, когда мы проводим, скажем, на NGS-секвенаторе изучение полного микробного состава содержимого рубца, после чего вот из этих результатов мы, используя программу PICRAST-2 и базу данных КЕГ, объединение генов бактерий в целостные метаболические пути. База данных PICRAST-2 представляет из себя биоинформационный программный комплекс, предназначенный для прогнозирования функционального содержания метагенома по маркерным генам, 16 СРНК и полным геномом. Ну вот здесь некоторые сведения о Киотской энциклопедии генов и геномов, которые все шире и чаще используются в исследованиях. Вот, и центром ресурса является KEG Pathway, вот, где диаграммы, называемые картами, соответствующих определенным сетям. Значит, ну вот мы, кроме того, вот то, что нам нарисует эта программа, а она может нарисовать все, что угодно, если ей дать волю, но у нас есть прекрасная возможность проверить, верифицировать вот это нарисованное нечеловеческим разумом, изучая экспрессию генов, вот, выделяя из рубца РНК, с помощью обратной транскрипции переводить ее в ДНК и смотреть, значит, вот, какие гены, потому что там кошмарное их количество, и кто из них экспрессировался, и какие метаболические пути так или иначе функционировали. Ну вот эти у нас финансировались грантом РФФИ эти исследования. Значит, в данном случае вот рубец. Вот есть такое заболевание кетоз, когда вследствие потребления некачественного силоса, ну вот напрочь меняется и микрофлора рубца, и сведения, образуются кетоновые тела, которые крайне вредны для животного. Ну вот здесь показаны результаты от НЖС-секвенирования. Вот эти самые, оказалось, что изменилось у коров с заболеванием кетоз, изменялась доля таксонов, связанных с нарушением энергетического баланса, в частности глюконеогенеза. Происходило повышение синтеза L-лактата, повышалось содержание бактероидез, исчезали некоторые виды, те, которые синтезируют пропионовую кислоту, а она как раз нужна для синтеза молочного жира. Возрастала численность не очень хороших бактерий. Ну вот здесь и данные, и статистически достоверные по сравнению функциональной аннотации метаболических путей сообщества рубца коров. Здесь показано, что там, где клиническая, что корова с кетозом, у нее больше, значит, происходила утилизация сахарозы, синтеза метахинонов, утилизация фукозы, биосинтез строительных блоков нулевого антигена, деградация разных соединений, деградация 4 аминобутаноата, деградация гекситолов, ферментация гексикола до лактата, синтез витамина В12 возрастал, и усиливалось гетероферментативное молочное брожение. Значит, я хочу сказать, что вот эти нулевой антиген, это скорее свойство, что там появлялись патогенные бактерии, это противостояние фагоцитозу, биосинтез бутандиола возрастал и деградация вот 4-бутаноата. Значит, вот я хочу сказать, что очень часто бродит такая теория, которую используют коммерсанты, продающие биопрепараты, ну, жулики, что где вот в результате применения тех же вот силосных заквасок есть D-лактат и есть L-лактат. Вот, и что якобы D-лактат, он не усваивается, а L-лактат, наоборот, стимулятор роста. Ну вот не именно подкисление связано именно с D-лактатом. И вот у нас появилась возможность исследовать, собственно говоря, есть, на самом деле есть бактерии, у которых есть гены D-лактата, есть D-лактат-дегидрогеназа, есть L-лактат-дегидрогеназа. И вот здесь как раз вот расчетные эти показаны. И вот результаты экспрессии анализа генов, ну, в целом подтвердили по D-лактат-дегидрогеназа, что у клинической коровы, что здоровой, то с диагнозом кетоз, а вот L-лактат как раз у здоровой коровы меньше синтезировался. Вот. Ну вот я хочу сказать, что мы поставили опыт по изучению экспрессии генов бактерий рубца, связанных на разных стадиях лактации. Вот я как раз рассказывал, значит, что есть сухостойная фаза, это за 30 суток до отела, новотельные, когда они вот, родили большого теленка, еще и доит по 40 литров в день. И происходит сверхъестественный вынос энергии, период раздоя, когда еще увеличивается, и в период стабилизации лактации, и период спаза лактации. И вот мы использовали НЖС-секвенирование плюс ПИКРАС-2 и анализ экспрессии генов при помощи ПЦР с обратной транскрипцией. Ну вот понятно, что удалось подтвердить, что в процессе вот, разных процессе вот, развития, как это сказать, в процессе фаз лактации меняются индексы Шеннона и другие индексы, показывающие серьезные изменения в микрофлоре. Но индекс Шеннона – это один из экологических индексов, который, ну, как бы показывает, как микроорганизмы распределены по всей этой вот, по всей численности бактерий. И это показывает, что действительно происходили глубокие физиологические изменения. Вот происходили изменения доли филумов. Например, актинобактерии как раз вот исчезали. Суперфилум веру микробиота в период раздоя, стабилизации лактации. В общем, происходило вот то, о чем я говорил, угнетение, переваривание клетчатки. И вот вы представьте себе, что происходит вот этот огромный вынос энергии, отрицательный баланс, энергетический баланс, и еще корова вдруг перестает переваривать клетчатку. Что ее ждет? Вот не только заболевание конечностей, не только нарушение обмена веществ, но и верная смерть. Вот. Ну вот здесь результаты анализа метаболических путей. И особенно вот мы обратили внимание и по литературным данным, вот было многие нарушения, вот у таких коров в состоянии кетоза, лактатного ацидоза, происходит нарушение как раз утилизации сахаров, цикла Крепса, вот этого трикарбоновых кислот. Ну вот здесь показано, что действительно происходило угнетение цикла Крепса, происходило нарушение синтеза микробиальных аминокислот аспартата, аспарагина, метионина, нарушение синтеза, ну, вообще нарушение глюконеогенеза. Вот оказалось, что экспрессия генов, синтеза жирных кислот, это то, что вот то, это энергетика рубца. Вот, собственно говоря, и молочные жиры, сахар, вот в рубце образуются летучие жирные кислоты. Больше там вот ничего не всасывается. Но именно это и есть углеводное питание коровы. Вот. И показано, что она вот как раз усиливалась. Значит, экспрессия фосфофруктокиназы, тоже биомаркер углеводного обмена, один из регуляторных ферментов гликолиза. В период лактации напряжение углеводного обмена в рубце и адаптация микроорганизмов к повышенным доступным источникам глюкозы, крахмала и моносахаридов. Экспрессия фосфоенол-пируват карбоксилазы. Это тоже фермент глюконеогенеза, метаболического пути, приводящего к образованию глюкозы из неуглеводных соединений. Следовательно, возможное усиление патогенеза в период лактации. Экспрессия редуктазы линолевой кислоты. Ген связан с метаболизмом жирных кислот, конъюгированной линовой кислотой. Период лактации синтеза жирных кислот происходит на фоне повышения доступных источников глюкозы. Усиление экспрессии этого гена связано с увеличением численности такой очень хорошей, полезной целлюлозолитической бактерии Butyrovibrio fibrina solvens. Экспрессия генов, вот как раз лактат-дегидрогеназы повышалась и D-лактат-дегидрогеназы в рубце у лактирующих коров по сравнению с сухостойными. Вот здесь это по разным стадиям лактации, это показано. Вот. Экспрессия генов цикла Крепса снижалась вот как раз у лактирующих коров по сравнению с сухостойными. Экспрессия метилмаланил-коа-мутазы, экспрессия гуанин-аминогидролазы. Это катаболизм, реакция катаболизма пуринов, угнетение синтеза микробного протеина в период раздоя и стабилизации лактазы. Ну вот здесь приведены выводы по данной работе, увеличение уровня экспрессии генов. Вот это Ганс Крепс и его цикл. Вот он his cycle. Значит, вот я просто хочу привести пример, раз конференция называется «Полезные микроорганизмы», как вот мы разработали еще, эти работы были начаты еще 40 лет назад, вот, и как мы их довели до конца. Это мой научный руководитель Лев Константинович Эрнст, академик. Сейчас его знают больше как папу Кости Эрнста, гендиректора Первого канала. Но он в течение лет, наверное, 20 был вице-президентом Россельхозакадемии как раз по животноводческим аспектам. И вот он поставил задачу выделить бактерии из рубца, которые повышали бы переваримость клетчатки. У нас была создана выделена целлюлозолитическая ассоциация. Сначала ассоциация, в ней было очень много компонентов, потом мы ее достаточно не спеша чистили. Но вот оказалось, что интересно, когда мы стали изучать целлюлазную активность этой бактерии, а нам помогал в этом такой Анатолий Клесов, член-корреспондент. Сейчас он больше известен скандальными публикациями по этой ДНК-генеалогии. Вот. Но когда-то он был очень хорошим энзимологом. И вот он показал, что целлюлазы находятся на внешней поверхности клетки, и они легко могут быть при покачивании, выходить в среду. То есть потом, вот уже в 90-е годы появился такой термин целлюлосома. У некоторых бактерий вот целлюлазы собраны в целлюлосомы, и там присутствует весь, значит, этот самый целлюлазный комплекс, и вот поэтому они могут просто вот полностью проводить работу от начала до конца, допустим, по растворению фильтровальной бумажки. Оказалось, что этот препарат, вот этот на основе, значит, практически работал на всех животных, на курах, на свиньях, на рыбах, на крупном рогатом скоте. Причем нам удалось превратить хищную рыбу-форель, которой необходима рыбная мука, обязательно в растительноядную. Это было заменено клетчаткой, очень сильно содержащим подсолнечниковым широтом. Ну вот, когда мы провели полногеномное секвенирование, оказалось, что это энтерококкус фециум целлюлозолитический. Там было много самых разных генов интересных. Вот сегодня много докладов, и дальше будут доклады про связь с мозгом, с нервной системой. Вот тоже синтез аминобутирата наотропа, передача нервных импульсов между клетками. Вот. Значит, ну и оказалось, что он действительно имеет антистрессовый. Он способен к формированию биопленок. Вот мы очень долго искали и продолжаем искать бактерии, которые бы разлагали микотоксины, потому что корма содержит как зерновые, так и травянистые, содержат большое количество микотоксинов. И вот искали, но вот нашли, и вот оказалось, что он хорошо разлагает и охротоксин главный, тот, который вот в силосе и в молоко попадает, и также вот такой микотоксин, в Ростове как-то неудобно это слово произносить, дезоксивалинол, сокращенно ДОН. Вот. Однажды даже автоматический переводчик как-то перевел его как микотоксин на Дону. Вот. Здесь показано, как мы искали эти самые разрушающие микотоксины. Здесь показано, что использование этого препарата, скармливание коровам, позволило снизить содержание, проходит афлатоксин через корову и попадает в молоко, и это молоко уже для детского питания не годится. А вот как раз мы с помощью вот этой технологии изучили, о которой я рассказывал, воздействие целобактерина на метаболические пути. Оказалось, что он очень сильно повышал синтез гликосилата, усиливал синтез мочевины, синтез аминобутирата, синтез пептиногликана, синтез пропионата, синтез сукцинил-КоА и синтез сукцината. Я хочу сказать, что все мы уже привыкли к этой мысли о том, что пробиотики имеют очень комплексное воздействие на многие процессы. Здесь, может быть, мы частично показали, почему это так. Происходила активация энергетического метаболизма, образование глюкозы и активация цикла Кребса. Активация превращения лантаина в мочевину, биосинтез пропионата, крайне важного для физиологии коровы. А доля этого препарата на рынке комбикормов. Скажем, в 2018 году около 7% комбикормов в России выпускалось с этим препаратом. Но я хочу сказать, что Марк Твен говорил, что есть ложь, наглая ложь и статистика, а особенно это относится к статистике Минсельхоза. Потому что если хозяйство не зарегистрировано в Минсельхозе, то его результаты не учитываются. Поэтому эти результаты по нашей доле, они очень сильно завышены, и они на самом деле скромнее. За эти все работы мы получили премию правительства в 2017 году. Мы в здании правительства на фоне кабинета Путина, там есть кабинет Путина, чтобы он мог прямо в правительстве кого-нибудь пропесочить. Это наша молодежь в прошлом году тоже получила премию правительства для молодых ученых. Тут две мои аспирантки и одна просто наша. Да, на фоне здания РАН. Из-за этого вируса не было такого. Затянулись вручения в Доме правительства, но так как наши девочки очень торопились, то им вручили в здании РАН. Спасибо за внимание.
Dear colleagues, we have time for one question. We're slightly behind our schedule. Are there any questions for the presenter? Yes, please. Yes, sure.
Микрофон, подождите.
Что передовая кормовая наука думает об использовании мочевины?
Спасибо большое за вопрос. Нет, она не такая уж и передовая. Но, безусловно, в моем понимании, при недостатке белка в рационе мочевину надо использовать. По очень простой причине. У коровы и вообще у жвачных животных существует очень сверхмощная система рециклизации мочевины. Когда мочевина вторично всасывается в тех же самых почках, попадает в кровь, подается на слюнные железы, диффундирует еще через стенку рубца. На стенке рубца сидят бактерии с уреазной активностью. И она очень эффективно используется. Я хочу сказать, что у других жвачных еще она иногда более эффективна. В частности, однажды мы охотились за лосями, и когда взяли содержимое рубца лося, оказалось, что там содержание, причем в тот период голодания, у нас была научная лицензия для научного отстрела, там в 10-100 раз количество и соединений мочевины, и белка превышало этих коров, которые якобы получают сбалансированное питание. Кроме того, нас спонсирует фирма «Лалиманд», так я понимаю конференцию, я сделаю им подарок. У нее есть очень сильный конкурент, фирма «Олтек», она даже сильнее представлена на кормовом рынке. У них есть такой препарат, называется, если не ошибаюсь, оптиген. Очень красивое название, модное, оптиген, но на самом деле это мочевина. Ну, вы знаете, надо в строгом соответствии с инструкцией. Вот ни в коем случае не допуская передозировок. Вот, скажем, хорошо это включать в какой-нибудь, ну, в миксере, перемешивать там, или, может быть, включать в премикс, в комбикорм, чтобы было равномерное поступление. Не так, чтобы там вот сыпать там и побольше. Спасибо. Да-да.
О биотрофе, да,
Да.
в препарате. В каких кормах использовался этот препарат?
Нет, ну вот, нет, наверное, о целобактерине, да?
Да.
Просто биотрофом у нас еще называются консерванты для кормов. Вот, если речь идет, у нас два целобактерина. Целобактерин плюс вот на основе энтерококка, Enterococcus faecium, и есть целобактерин Т на основе бациллы, и причем она, вот эта бацилла выдерживает и экспандирование, и гранулирование. Ну, вот, скажем, больше половины кормов для бройлеров проходит экспандирование и гранулирование, и там до выше 100 градусов поднимается температура в этом процессе. Значит, ну, у нас в основном нашими покупателями являются птицеводы, являются крупный рогатый скот вот в связи с этим, потому что действительно восстанавливает активность микрофлоры рубца. На свиноводстве мы представлены несколько хуже. Вот, включение в рыбные корма дает тоже очень позитивный эффект. Вот у нас, значит, ну, вы тоже знаете, что в России большая часть кормов, особенно вот в том регионе, где я работаю, это все заграничные корма, скандинавские, датские, и туда вложить какой-то пробиотик возможности нет. Но есть виды, тот же, по-моему, карп, где корма готовят в России.
Ну, сейчас не только для карпа, сейчас еще и для других объектов готовят.
Ну вот мы работали вместе с институтом Госньюорхом. Это есть такая, и она сейчас жива, хотя эти работы делали порядка 30 лет назад, Ирина Николаевна Остроумова, профессор. Вот и опыты с ней поставлены, что включение целобактерина действительно позволило превратить хищную рыбу форель в растительноядную.
А скажите,
Я
пожалуйста,
раньше показывал даже фотографию Франциска Осиского,
Какое
волка и теленка.
А какое количество добавляли? Потому что все-таки для лососевых характерно питание такое, корма на рыбной муке.
Ну, вы знаете, нет, вот в тех опытах, значит, добавляли количество, которое мне показалось очень большим, то есть даже нерентабельным. Вот, это были работы до 90-го года выполнены. Значит, и причем мы вели работу на старых штаммах. Вот именно тогда мы сообразили, вот, именно в связи с гранулированием рыбных кормов, что нам нужно искать термоустойчивые варианты, проходящие гранулирование.
Спасибо.
Thank you, and I would like to move to the next presenter. Before I will introduce the next presenter, I'd like to say that after we'll finish the session, we're moving to a different location. Everybody will be guided to the location, and we will have a lunch break, and after the lunch break there will be the session on human probiotics. So our next presenter, Professor Zong Deep, has a very challenging task to present before the lunch break. I'm trying to be humorous. We all know how the audience is becoming impatient in expectation for the food consumption, which is to follow the heart-brain word. We have a presentation by Professor Deep. You may remember today in the morning, Dr. Leong mentioned how he helped her to pack the gift bags for the participants of one of these conferences. And I can only humbly add to it that I was doing the same another day, helping to select certain things for the bags of the presenters. So Zong, I'm with you on these most challenging task. And without any further ado, I give the floor to Professor Deep presentation, Norwegian University of Life Sciences as Norway.
Good morning or good day to everyone. My name is Jon Deep. I'm a professor from the Norwegian University of Life Sciences. Before I present my talk, I would like to thank the organizing committee, especially Mike and Minji, who have invited me to present our work here today. I have been in the ICOM conferences a few times before, one in Thailand and the second time was in Malaysia. I must say that I have enjoyed every moment of the two conferences. I have learned a lot and made many friends. And so I'm really happy to be part of this community. And it's a great honor. And thanks a lot for that, Juminji and Mike. Now let me go back to my talk. The title of my talk is Bacteriocins, a great source of antimicrobials to fight against antibiotic resistance. This is the outline of my talk today. I will first give you a brief introduction of the bacteriocins, and then I will tell you where and how we find them in nature. And at the end of my talk, or the second part of my talk, I will show you how to use them to fight against antibiotic resistance. Antimicrobiopeptides are really common in nature. They are produced by all three domains of life. They are produced by eukaryotic cells, by archaea, and by bacteria. Here I show you a set of peptides produced by different organisms. You can see that some can be quite simple, just of an alpha helix, and some can be a bundle of alpha helices or a set of beta sheets, and some of them can be also really modified. These peptides, they are part of the innate immunity system, or the first-line defense against the invading pathogens. In terms of activity, those produced by eukaryotes seem to have broad spectrum activity, meaning that they often kill both gram-negative and gram-positive bacteria. This is quite different from those produced by bacteria. Antimicrobiopeptides produced by bacteria, they are normally referred to as bacteriocins. They are used as a means for competition for nutrients, but also for defense, to protect their ecological niche. Bacteriocins, they are gene encoded, meaning that you can find the sequence of the peptide within a gene. So they are formed by ribosomes. This is totally different from antibiotic peptides which are put together by multi-enzyme complexes. Most bacteriocins have narrow spectrum. They are active against just a few related species or genera, often those living in the same niche as the producer, but some can have broad spectrum activity as well. Classification is difficult sometimes, and still an ongoing debate because they are really diverse in terms of sequence and in terms of structure. But in general, you can divide them into three classes. Class 1, class 1, let me see, class 1, 2, and 3. Class 1 and 2, they are of small peptides, less than 10,000 kilodaltons. They are often heat-stable. While the class 3 bacteriocins, they are much bigger, larger than 10,000 kilodalton, and they are heat labile. Class 1 bacteriocins, they are heavily modified. And one example that you can see here with niacin, which contains many ring structure of lanthionine bridges. And for class 2 bacteriocin, they are either not modified at all, or they have just some limited modification. An example for that is pedocin A1, as we can see here, which has 2 disulfide bridges. Now let me take you to the next point of my outline. Where and how do we find the bacteriocins? For that, I can give you this simple answer. Where you find bacteria, you also find the bacteriocins. And then you ask me, but how do we find the bacteriocins themselves? For that, I need to show you this rather complicated picture. In principle, you can use any biological samples to do the screening. What in our case, we went to a local supermarket where they have different fruits and vegetables imported from different parts of the world. We bought somewhere 50 to 60 products, and we bring them home and chop them down into small pieces, and we add some water, and then we divided each sample into 2. One we didn't add any salt, and when one we add some salt, just to create two different environments. And we left the samples for 1 to 2 weeks in the storage room to let the bacteria grow and to fermentate. And after that, we just take the juice from each sample and then use that as a source for the screening. But for the screening itself, what we do that first we dilute the sample into different concentration so that we can hit the number 50 to 200 CFU per plate. And then over this layer, we add another layer, what we call it magic layer. This is just to keep the cells growing below the surface. Then we left the plate in the incubator overnight to let the cells grow into colonies to produce bacteriocins. And the day after, we add a layer of the indicator on the top. And then we just leave it again overnight in the incubator. And when you come back the day after, if you're lucky, you will see something like this, a plate with many colonies with inhibition zones around. And these are the candidates of bacteriocin producers. With this protocol, you can easily upscale to somewhere 1,000 colonies per sample. And you can easily do 10 to 20 samples a week, meaning that you can easily screen 10,000, 20,000 colonies a week. And this is really cost-effective and really powerful way to find bacteriocin producers. Here is just a sample where we have found some candidates from broccoli, from bitter lemon, and from milk. So what do we do after we have found the bacteriocin candidates? Yes, we can purify the bacteriocins. We look for genes behind the bacteriocin synthesis. Genes regulate the bacteriocin synthesis, and we also can find this receptor where the bacteriocin bind to kill the target cells, and the mode of action, how they find the target, how to kill the target cells, and the immunity mechanism, how the producer protect themselves. And we can do further study, like structural study, the structure of the bacteriocin, the structure of the receptor, how these two come together that eventually lead to the killing of the target cells. And the last part is the holy grail of bacteriocin research. all these are really interesting, but this is not the part of my topic today, so I'll leave it to next time. So now let me take you to the last part of my talk about how to use the bacteriocins to fight against antibiotics resistance. This is the picture we all have seen on the news, in the scientific journals, and on TV, that the pathogens, they become more and more resistant to most antibiotics, and the production of the new antibiotics is dropping dramatically over the years. And we also have learned that once we introduce a new antibiotic into the market, sooner or later we will have resistant bacteria. So we're kind of entering the evil circle that at the end of the day, the bacteria will win. So we need to think a different way to deal with this problem. So can bacteriocins be part of the solution to deal with this rather dreadful situation? Sharing some light in this tunnel. My answer is an optimistic yes. We have been working on a bacteriocin called galveston gas. It is a broad spectrum bacteriocins. It kills many pathogens, including Listeria and different species of enterococci, Enterococcus faecium and Enterococcus faecalis. And look at this one here, this isolate of Enterococcus faecium, an isolate from outbreak. This isolate is resistant to all antibiotics tested, from streptomycin, erythromycin, ampicillin, tetracycline, to even vancomycin. But yet, our bacteriocin can kill this pathogen, this isolate. Can this be the solution? The answer is both yes or no. The reason for that I will tell you in a second. What I'm going to tell you now is the story of two bacteriocins and one antibiotic that together we use to deal with MRSA, which is one of the major pathogens we have nowadays. This is based on two works recently published in a group. The first one on the left is to use this combination to deal with Staphylococcus biofilms, which is a layer that make the cells more resistant to antibiotics. The second paper is about to use this combination to deal with MRSA in skin infection in a mouse model. The two bacteriocins I was talking about are galveston gas and mycococcin P1. The first one produced by Lactococcus gavia, and the second one by Staphylococcus aureum. Both were identified by the screening assays I told you a few minutes ago. Galveston gas is the multipeptide bacteriocin, meaning that it is composed of several peptides. In this case, 3 peptides is in galveston gas: GOG-A, GOG-B, and GOG-C. And these 3 peptides they come together and form a functional unit that form pores on target cells. For mycococcin P1, it is a thiopeptide bacteriocin. It is heavily modified, and you can hardly see the amino acid, so the sequence of the amino acid itself in the final product, because it is so modified. Its mode of action is to inhibit protein biosynthesis. Both bacteriocins, they have a broad spectrum activity. They kill most gram-positive bacteria, including many pathogens. Among them are pathogenic species of Staphylococcus, Enterococcus, Streptococcus, and Listeria. Staphylococcus aureus is a pathogen. You normally find them in the open wounds. We have a collaboration with a group in India led by Dr. Aparna Srikantan. She's working with leprosy patients, and these people, they have open wounds in their hands and their feet, and normally these sites, they are infected by bacteria. And she did a screening, and here you can see that a list of bacteria she normally found in the wounds, and you can see on the top of the list Staphylococcus aureus is the dominating one. So together we decided that we will try to use bacteriocins, develop bacteriocins into drugs to treat these patients. And we aim at the Staphylococcus at the beginning. Now we have two bacteriocins with good activity. We have seen that both galvacin gas and mycotoxin P1 are quite effective for short incubation. But when you incubate the cells with these antimicrobials for longer time, 24 hours or 2 days or 3 days, you see resistant cells or adapted cells popping up in the middle of the zones. So this means that these peptides can not work for longer incubation. So that's why we need to seek for synergy. So what we did next is to try to seek synergy between galvacins and with all the antimicrobials, including a set of antibiotics, as you can see on the left. We see that, by the way, we use methicillin Staphylococcus aureus strains in this assay. So when we do the synergy assay, we see that galvacin gas together with mycotoxin P1, we see some synergy. Mycotoxin P1 is reduced from 2.5 to 0.16 microgram per ml. For galvacin gas, it is reduced from 32 microgram per ml down to 8 microgram per ml. And for the antibiotics, we saw that ampicillin, penicillin G, and streptomycin have good synergy with galvacin gas. We see that ampicillin, which is totally useless against this pathogen, the MIC is reduced from 2,500 microgram per ml to 16 microgram per ml. And for galvacin gas, it's reduced from 32 to 8. And there's similar results with penicillin G, and also for streptomycin. And as penicillin G is still widely used in medicine, we try to look further into this antibiotic. We try to see whether penicillin G has synergy with mycotoxin P1 or not, and it did indeed. As you can see here, the penicillin G, the MIC when it is alone is 2,500 microgram per ml, but in this combination it's reduced down to 16. And for mycotoxin P1, it is from 2.5 to 0.16 microgram per ml. So it is a lot. So since my penicillin G has synergy both galvacin gas and mycotoxin P1, and we try to all three together to see whether we can get stronger synergy, and we did, and we got it indeed. And as you can see here, we can further reduce the MIC values of all three components. And the good thing about this combination is that when we try to expose this combination to the cells, and we don't see much resistant cells or adaptive cells popping up in the middles of the inhibition zone. So meaning that this combination seems to be a viable combination to deal with Staphylococcus aureus. Normally, in the infection sites, you have a mixed culture, meaning that you have different bacteria living within the wound. And for this assay, we tried 3 different MRSA strains and 2 Enterococcus faecalis strains together as a mixture. And then we expose the mixture to different antibiotics and the formulation itself. Here you can see that the antibiotics, some is working, some is not working at all, some is quite reduced. But look at the formulation we have. It is still active. It's still really good in this assay. So it seems to be a very good candidate to develop further. And now we have the combinations that's working quite well. Let's try on biofilm. I mean, Staphylococcal biofilm is known to be involved in antibiotics resistance. It's the form layer. It is a layer that protects the antibiotics to come in to kill the cells inside. So how is the combinations in these settings? So this is the assay we did to see whether the formulation is working on biofilms or not. First, we grew the cells overnight to make good biofilms, and then we treat the biofilms with formulations. Before we use a kit, a biofilm viability kit, to detect the live and dead cells. In this kit, we have 2 components. One is cytonine, which detect live cells. It gives a green color. And we have a second component, which is a popidium iodide, which detects dead cells, and it gives a red color. And here's the confocal microscopic picture of the result. For the control, for the negative control, we just grow the cells and add the buffer only without the bacteriocin. You see that in all settings they grow at nicely. They all are green, more or less. And when we treated them with the bacteriocins or the different components, we here we have galvacin gas alone. We see that it kills, but you can see that several spots of green cells within the biofilm. So you have live cells within the biofilm. And here for mycotoxin P1, you see that it's not working at all. You see some spot of red cells, but the majority is green. And for the combination between galvacin S and mycotoxin P1, you see a lot of red cells, but still you can see some green cells within the biofilm. Penicillin G is not working at all. As you can see, most of the cells are green. And when we have all 3 components together, we see a dramatic reductions of the biofilm, and also more or less everything is red. You can see some green spot here, but it is more or less the artifacts, but not live cells. So the conclusion from this assay is that the formulation is working really well against the staphylococcal biofilms. Here's the scanning electron microscopy of the biofilms, treated and non-treated. On the left, you have the non-treated. You see that the cells look healthy, round, and they look just beautiful. But for the treated cells, you see a dramatic situation. The cells, they don't look healthy at all. They are squeezed, and also it looks like we have a decreased on the surface, likely something have been leaking out from the cells. Next, we want to try whether the formulation is working in an in vivo system or not. So we tried on a mouse model system. For this, we used Staphylococcus aureus MRSA strain called SENS36. This one has a luciferase gene in the genome. It generates photons, luminescence by itself. To the mice, for each mouse, we punch a hole on the back of the skin, and then we inoculate the bacteria at the concentration 2 times 10 to the 7, and then we just let the mice stay in the cage overnight so that the bacteria can grow, establish infection, and then we just do the treatment and collect the photons every day using the IRIS imaging camera. So we did the treatment and we collect the photons every day. This picture is from day 5, I think. So it has 4 treatments, 1 a day. And as you can see here, we have 4 different settings. On the top, we have untreated mice. So this just infected, and then we see that we have a lot of luminescence coming out from the mice. And for the second settings is with the vector. So this is just a buffer. You see that it has no antimicrobial effect. Still a lot luminescence coming out. And we also use a positive control and antibiotic fusidin, which is an antibiotic still using nowadays to treat skin infection. It's working against most mice, but one mouse seems to be not treated at all. There's a lot luminescence coming out. The reason for this is because, as you can see on the right here, fusidin generates a lot of mutants. And look at our formulation at the bottom. It's removed the luminescence beautifully from the wounds. More or less no luminescence at all. Now we want to know how many treatments do we need to get rid of the infection and to prevent the recurrence of the infection. For that, we did this experiment. It contains 3 different settings. 1 non-treated, 1 treated only 4 days, and then we left the mice untreated for 6 days later. And then 1 is the positive control, where we did the treatment every day. And for the non-treated, we see that the luminescence is slow at the very beginning, and it peaks at day 6, and it went down again. The reason it went down again is due to the innate immune system in mice. For the 4 days treatment, we see that the luminescence dropped down right away, and there is no recurrence at all after the last treatment. And for the 9 days treatment, I expected there is no luminescence at all over the entire experiment. And so to summarize what I have demonstrated so far, that we have managed to develop a therapeutic candidate containing 2 bacteriocins and 1 antibiotic that together form a synergetic formulation that is not only efficient but also viable against MRSA skin infection, at least in the in vivo system in mice that we have demonstrated. Before I stop, I would like to bring to you some take-home messages. Bacteriocins are indeed a great source of antimicrobials to fight against antibiotics resistance. That's for sure. bacteriocins, they are ubiquitous in nature. You can find them everywhere, just waiting for you to discover. But like antibiotics, the in vitro bacteriocins can give rise to resistance. So to prevent that, you need to combine several bacteriocins with different modes of action. You can even combine with old, less efficient antibiotics and make them alive again. And in that sense, you revitalize the old antibiotics. And finally, I would like to thank a few people. First, to my collaborators, Abana, Hanne, Tamara, Isabella, Pauline, and Harald, with whom we have done many nice work together in bacteriocins research. And also to the people in my group, especially to Kirill, who did most of the work on mice, and Christian on biofilms. I also would like to thank the funding bodies. Without them, I'm nothing. At least I couldn't do anything in my research. And lastly, but not the least, I would like to thank you all for listening. I'm sorry that I could not present my work directly to you. So if you have any questions, please write to me by emailing. With that, I say do свидания. Bye-bye.
Thank you, Dzongdip, for such a great presentation. I'm not sure if he is with us today. I don't see him on the screen. So as Dr. Dip mentioned, if you have any questions, please address them to the organizing committee. You should have email address for the committee, and they will make sure to pass the messages, the questions to Professor Dip. A small announcement now. We have a break, and this last presentation finalized the plenary session of our conference. After the lunch break, we're moving to the session on the human probiotics, and this session will start at 2:20 at the same location. Please disregard what you see right now in the program. We're moving out now. Excuse me. First, we will have a picture, and I guess weather is not really very good right now, so we will be on the ground floor of the main building in front of the posters, and in the Congress hall. And then we'll move for the lunch break. And after the lunch break at 2:20, those of you who are interested in human probiotics, please, you are welcome to return back to this hall, to this room. Thank you. Мы можем оставлять. Мы сейчас снимочек делаем, потом идем обедать.
Да, и потом все...
А потом возвращаемся все сюда.
Можно здесь оставить.
Да, я поэтому... Ну, я уберу.
Да, мне за что? Мне по маленькой кругу. Да.
Человеческая сессия потом начинается. Вы сидите.
Пойдемте на фото, пожалуйста, коллеги.
Один, раз, два, три. Раз, раз, два, три. Раз, два, три, проверка. Сейчас я тут выключу микрофон, все нормально. Это я чтобы не шел туда. Так, а здесь у нас идет
Добрый день, администраторы.
Так, сейчас, секунду. Слышно нас?
Да, вас
Добрый
слышно.
день.
Слышу вас. Меня слышно?
Да, все отлично, видно, слышно.
Видно, слышно. А можно я сейчас экран продемонстрирую, чтобы...
Да, да, можем.
Хорошо?
Да, хорошо.
Раз, два, три. Раз, два, три. А там восьмерка выключена. Раз, два, три. Работает. Значит, вот так вот. Раз, два, три. Раз, два, три, четыре, пять, шесть. Ну вот.
Так, слышно меня?
Раз, два, да, слышно. Слышно нас? Стереомикшер или на вход? Нас слышно? Скажите, пожалуйста. Нас слышно? Скажите, пожалуйста.
Да, вас слышно. Я сейчас вот переименовываюсь. Хочу все переименовался. Так, а видно меня? Теперь
Да,
мне
да,
надо,
вас видно, все хорошо.
так, теперь мне надо как бы демонстрацию экрана попробовать, да? Просто у меня здесь вот плохо с интернет, что-то там периодически начинается.
Ну, если что, можно отключить камеру, и он будет немножко поживее. Но это если прям вообще плохой интернет.
Я понял. Ну, если что, я понял. Так, отключить камеру, это просто я нажму кнопку, да, и все.
Да, да, в левом нижнем углу там выключить видео.
Остановить видео, да, и все.
Да.
И я...
Ну давайте попробуем с вами демонстрацию.
Давайте. Так, вы разрешите мне демонстрацию, что здесь пишет организатор отключил демонстрацию.
Раз, два, три. Раз, два, три, четыре, пять. А, восьмерка работает. Еще раз, включи. Раз, два, три, четыре, пять, шесть, семь. Он приглушенно работает. Раз, два, три. Главное, чтобы работало.
Hello, my name is Tatiana Khadykina. I am an associate professor at the Department of General Hygiene at the Erisman Institute of Public Health at the Sechenov First Moscow Medical University. The topic of our report is probiotics and the Hello, my name is Tatiana Khadykina. I am an associate. Introduction. Within the
from indigenous fermented food will be presented by Professor Santosh Kumar Tiwari. You are welcome.
Hello, everybody. First, I would like to thank the organizers for inviting me to present the kind of work we are doing in our laboratory, especially Professor Michael Chikindas and his entire team. At the same time, I congratulate the organizers for organizing such a wonderful event and the conference which actually addressed the message during this pandemic that how the nature-derived product and the beneficial microbes can play a very important role for the improvement of human health, nutrition, and also in the therapeutics. So the theme of the conference is very much related to the kind of work we are doing in our laboratory. And we were happy that we received the invitation, and I just agreed that yes, we will be happy to present the kind of work we are doing and address to the best of our capability that such kind of microbes can play a very important role. So I would like to share my presentation and then start.
Yes, please.
So my work is mostly the work we are doing in our laboratory, the source of lactic acid bacteria, their probiotic potential, and production of antimicrobials, especially the antimicrobial peptides like bacteriocins. So we have explored several fermented foods, and why we are going for the food, because you know, these fermented foods are a kind of a habitat, a natural habitat for such kind of probiotics which are found in the food. So if anything we have from the nature derived product which is available in our food and nutrition would be great source for the isolation of such probiotic bacteria and their application in the human health. There are a lot of literature you can find on the fermented foods and its application, its nutraceutical benefits, but the very least effect are available, least sources are available on the bacteriocin profiling of such kind of probiotics which are found in the fermented foods. So I would like to highlight some of the foods which are found not only in India, but throughout the world we are using such kind of fermented foods since dawn of the civilization. And few of them are very much of indigenous origin, means they belong to certain regions, and they are very much specifically cooked and prepared in such a way that there is a fermentation, and such fermentation leads to the growth of several microorganisms, like especially the lactic acid bacteria, yeast, which play actually a probiotics roles, and that is the source from where the human gut actually grasp all these probiotics from the kind of food we are taking. So at the same time, like if you look at the present situation, there is a lot of role of these microbes, and looking at the importance from my laboratory, that we have isolated several strains of lactic acid bacteria from the indigenous fermented foods and fermented foods which are available. There are several strains have been isolated from these foods I have eaten, like milk, dosa, cream, soya milk, vada, dokla, idli. So some of them are the vernacular names, but you can find very easily that such kind of foods like dosa and idli are the most common in the southern part of India and the southeast region of Asia. These are very much common, and I have mentioned recipe for the preparation of batter of dosa, and you can see that there is a involvement of some plant products like black gram, rice, chickpea, and fenugreek seeds. And when these flour of these plant seeds, they are incubated with in the water for overnight, and there is a fermentation. So our focus was to use such kind of food products which are known for the growth of certain microorganisms, and their health benefits are reported since dawn of the civilization. And we would like to prove that what kind of microbes they are present and what are their characteristic in terms of its probiotic potential, antimicrobial nature, and production of bacteriocins. So out of these stents, we have selected two stents which are showing the potential activity as antimicrobials, and along with this, they show a very good acidifying activity, which decrease the pH, and they show the bile salt tolerance, bile salt hydrolase activity, and cell surface hydrophobicity, and that actually leads to the auto-aggregation of these strains. So that actually prompted us to understand the probiotic property of such strains. And of course, along with these property, one of them was the antimicrobial activity. And we have used certain tests on the basis of biochemical and morphological tools for the identification of such kind of lactic acid bacteria, and found that one of the strain LD1 and belong to Lactobacillus, another LD3 which belongs to the Enterococcus genus. The same was confirmed using the molecular techniques, 16S rDNA sequence amplification, and it's a homology search through the database that suggests belonging to the class with the Lactobacillus plantarum, and another was the Enterococcus hairy. So these two strains were selected for further study. Of course, we have another one also, but I would like to highlight the importance of these two, along with the probiotic property, the bacteriocin production. So both the strains, when they grown, the production of bacteriocin was a growth associated, and when the strain was growing, at the same time, the antimicrobial production was found to be higher, and there is substantial decrease in the pH. That means due to the production of acid in the culture medium, and that has given some of the idea that such kind of strains which are found in the Dosa batter was Lactobacillus and plantarum and Enterococcus hairy, and they produce certain antimicrobials. Though it is not confirmed at this stage whether it is bacteriocin or not, but cell-free supernatant suggests that there is antimicrobial activity and the substantial drop in the pH was recorded. So with some of the idea that the cell-free supernatant was showing antimicrobial activity and it was heat stable, and when you treat with the proteolytic enzyme, the activity goes down, that has given some kind of assumption that these strains produces a kind of bacteriocin-like substances or bacteriocins. So we have purified from the cell-free supernatant using activity-guided chromatography methods, and also using the salt precipitation, ultrafiltration. So substantial, the information is available that the molecule is purified from the cell-free supernatant, and this was a kind of protein sequence using the Edmondig-Edison and terminal sequence. We have, though we don't have the complete sequence of this peptide, but even the N-terminal sequence does not belong to the sequences available in the database. That actually suggests the kind of the novelty or new compound which is present is still not reported in the database. So we were of the opinion that if such kind of bacteriocins which is available in the cell-free supernatant of this strain, Enterococcus, then we can characterize for further details and found that they are quite stable at the pH range from 2 to 6, but stability goes down after the 7, like at pH 8 and something like at pH 10 they don't say activity, but at pH 8 and 7 the activity goes down, but up to pH 6 it remains quite stable. And the stability at higher temperature is tremendously stable at up to 121 degrees centigrade and autoclaving condition, and also stable the boiling temperature, which is good for any industrial pharmaceutical application like food safety, food preservative, and so on. So the bacteriocin was quite stable at different, in the presence of different organic solvents, surfactants, and detergents. Its activity was not affected by enzymes like amylase lipid, but it has gone substantially down. And the activity was decreased in the presence of proteolytic enzymes when we use. It suggests the proteinaceous nature of the compound along with the sequences. So this information actually confirms the kind of molecule which has antimicrobial activity is probably bacteriocins. And trust me that we have tested this against not only lactic acid bacteria, but also against the other pathogens, which are both gram-positive and gram-negative, though it is very difficult to convince the scientific community that the probiotic bacteriocins are not effective against gram-neg. But we have substantial data to show you and justify that yes, such kind of bacteriocins which are isolated from these natural sources are effective not only against the related strains but against certain known pathogens like we have shown like Pseudomonas aeruginosa, Staphylococcus aureus, Enterobacter, E. coli, Salmonella, Gala, and Vibrio. So they are very fastidious pathogens. And if such bacteriocins is effective against such pathogen, that needs to be confirmed further. It can be a very good potential molecules, not only for the food safety, but for the several therapeutic applications. So we have seen the viability of such kind of stains after the staining, DOPI and PI, and shown that the red color is because of the dead cell and the blue color is the live cells. And when you treat the cells like M-luteus, we have shown that we have seen on the M-luteus when after the treatment, the viability has gone completely down, and the dead cells represented by the red cells. So that has shown that low concentration has few of the blue cells, but the higher concentration completely wipes this live cells and dead cells are there. The same experiment was repeated on the E. coli also and found similar results that the treated cells are dead, represented by the red and staining. At the same time, we are trying to confirm that whether this molecule is interacting with the membrane or not. So we have previously demonstrated the efflux of membrane potential and ATP, internal sodium and potassium concentration is released. Nucleic acids and DNA release that we have demonstrated, but one of the experiments where we do to demonstrate that when the treated cells are run on the IR spectrum, there is a disturbances of the spectrum near to the phospholipids and the nucleic acids. So this actually suggests that when the molecule is interacting with the cell, there is some disturbance in the IR spectrum near to the phospholipid range, and that actually suggests the molecule is interacting with the lipid, membrane lipid, and further the nucleic acid also. So this is a kind of assumption and speculation that the molecule is not only interacting with the membrane but also entering inside the cell and interacting maybe the cytosolic enzymes and the nucleic acid, of course, because we are getting such kind of disturbances and the nucleic acid range. So this is just a kind of experiment to show you that the bacteriocin which is reported is not only membrane acting but probably is going inside the cell, which is a very less report, and that so alludes that the molecule is entering inside the cell and killing the cytosolic effect. So the experiment was performed on the target cells on morphological behavior of the target cells using electron microscopy. In the transmission electron microscopy suggests that if you compare the untreated cells with the treated cells are very much ruptured, and there is a disturbances at the membrane level. So there is a membranous disturbances. So which actually confirms our data that it is active on the membrane. So the similar experiments like we perform on the E. coli also, and trust me that these are the real images. And if you compare the untreated cells, the treated cells are completely broken down. So see this one, and not only broken down, some of the cells is a complete dissipation of internal content. Cytosolic content comes out, bubbling. So these data suggest that the molecule when attacked is not only on the gram-positive microbacillus, but also a kind of one example we have shown with the E. coli that it is effective on the gram-negative also. So it shows the complete loss of viability along with the cell rupture and leading to the release of the cell content and cell dies. One of the experiments we have performed the importance of one of the important pathogen Staphylococcus aureus, and we have seen the loss of viability, killing of this pathogen not only in the medium but also a food model, like one of the food where it is most probably found to be contaminated there. And we tested in the milk model and found that when you grow these such kind of Staphylococcus aureus in the milk, after the treatment with the bacteriocins, both LD3 and one of the bacteriocins we have LD4, and when you compare the treatment, there is a loss of viability after the treatment. And it actually suggests that the bacteriocins are not only killing the cells under the laboratory condition in the medium, but also killing the cells in the food model, which is a very good example that this can be utilized as a food preservative. Another important one we have seen is the inhibition of Salmonella typhimurium. So this is one of the important foodborne pathogens, which actually causes several damages in the food and destroy. So we have seen the test in the fruit juices. When you contaminate the fruit juice and treat with these bacteriocins, there is a after the treatment there is a loss of viability count. If you see, this is the control one, and after the treatment there is substantial loss in both the case. Both the bacteriocins, there is a decrease in the viability. So these two experiments in the milk and the fruit juice suggest that the molecule is not only important for the academic purpose, but also can be applied for the various application in the food safety and therapeutics, also leading to the inhibition of several pathogenic bacteria. So recently we have reviewed this, that probiotics in general, that these probiotics are effective against the oral pathogens and not only killing the pathogens, but they inhibit the biofilm formation, which is a very critical requirement for the recent, by the recent clinicians, that inhibition of biofilm is one of the important aspects, because such kind of biofilms are creating a lot of problems while treatment with the different drugs which are not effective. So it's like direct killing of the cell, immune modulation, and inhibition of biofilm. Both all these are very important aspects for the inhibition of pathogenic bacteria and application of such kind of probiotics. Our recent reviews, this is a very good review by entire scientists from throughout the world, including Professor Michael Chikindas and Igor Popov from the Don State University. And they came while in last pandemic, last year, that we are doing this kind of review, that such kind of probiotics can inhibit the spread of this coronavirus or viruses in general from animal to human and from human to human spread can be stopped. And such kind of probiotics can be used as a therapeutics also for the inhibition. So there is no report on the coronavirus, but there are several viruses reports are available that such probiotics are inhibitory against this. So in general, what we are trying to understand through our work, that if we isolate the probiotic bacteria from the indigenous food and show the importance of these foods in the probiotic potential, And these probiotics may show several probiotic characteristics, including the antimicrobial activity. And focus of our laboratory was to understand the bacteriocins present in this lactic acid bacteria and their characteristic and their possible application in the food and the therapeutic if it is possible. So in the outset, like I would like to conclude that we have isolated these strains and shown the probiotic potential in terms of antimicrobial activity, bile salt hydroreactivity, cell surface hydrophobicity, and so on. So we have characterized the bacteriocins which are actually having a kind of novel features which can be applied for future applications. So I would like to thank my mentors and our collaborators, which are actually real-time helper to suggest the kind of work we are doing in our lab, and they're always ready to help Professor Michael Chikindas, Sanjay Patole, and Hussaini from Australia, and Professor Srivastava was my mentor at the PhD level. So most of the PhD students in our laboratory, they are highly dedicated. They perform the experiment, and so real credit goes to these students, and also the government funding agency. They supported our work for such kind of work on the probiotics and bacteriocins. Thank you very much for your attention.
Thank you very much, Dr. Tiwari, for your very interesting report. Who wants to ask the questions? Please, Professor Chikindas.
Good afternoon, Santosh. I'm very happy to see you. Sadly enough, only on a big distance, but happily with everything that we're together. I have a question, and perhaps this question will not be new to you, but I will appreciate you elaborating on this matter. As you may remember, and you certainly know from your own experience, that it was considered kind of a golden rule if the bacteriocin is isolated from gram-negative bacteria, it's active against gram-negative microorganisms. If it comes from gram-positives, it's active against gram-positives. And just maybe 10, 15 years ago, we've never heard about bacteriocins from one gram-staining group being active against another, and yet we see in your really nice presentation the bacteriocin that is working against both gram-positive and gram-negative microorganisms. What will be your take on that? And I know that you are studying the mode of action of bacteriocins. How do you think these bacteriocins that are active on both gram-negatives and gram-positives are different from others, and what makes them so unique? Thank you.
Thank you very much. Just quickly, am I audible to all of you or not?
We can hear you.
Can you hear me?
Yes,
yes.
Here.
Go ahead, please.
Thank you very much, and thanks all the organizers for giving me opportunity to present the work we are doing. Although it is a very initial level of study, but the kind of microorganisms like lactic bacteria we isolated from these indigenous fermented foods, and they were the basic inherent property of these lactic acid bacteria from these foods, that they were having a broad spectrum activity. So that has a breakthrough, that the knowledge what we have is the probiotic bacteriocin is active only against gram-positive. So we came with some conclusion that it is inhibitory to gram-negative also. Yes, it is very difficult to answer at this particular time, because the mode of action of this at molecular level is not enumerated much, but the kind of test we have performed is just like a physical disruption or cell killing or loss of viability,
is almost similar with the gram-positive and gram-negative, but we are looking at the mode of action of this bacteriocins in detail at molecular level. So unless we explore at molecular level the exact site of interaction of these bacteriocin with the target molecule in the cell, we'll be able to say. But our initial data suggests that it has a spectrum not only again gram-positive but gram-negative also.
Thank you. Who else wants to ask the questions? I have a question. What concentration of bacteriocin production bacteria which you find in these food products, what concentration did they reach in that very food products? Concentration is what amount per milliliter, for example?
Santosh, the question is, what is the concentration of these bacteriocin producer in the food?
Yes.
Have you ever identified the numbers in the food, or you just picked up it from the food sample?
See, we have isolated them from the food and purified in the laboratory conditions. So we have not detected the secretion of bacteriocins in food as such. The bacterium was isolated from the food and it was purified in the laboratory conditions and tested in the laboratory condition in vitro on different microorganisms we have seen. So yes, in the food it might be producing less amount or high, we are not sure.
Okay, then thank you very much again, and we move to the other presentation.
Okay, the other presenter will be a scientist from Russian Federation, Roman Yunis, a researcher from Vavilov Institute of General Genetics, Russian Academy of Science, role of PFNA operon of bifidobacteria in species-specific adaptation to host immune system. So, Roman, if you are here.
Hello?
I can see the presentation.
Hello?
Hello, hello.
Yes, can you hear me?
Yes.
Okay. Okay. Thank you. Thank you for the introduction. Thank you, Dr. Tiwari, for the interesting talk. I want to thank also Professor Michael Chikindas for inviting us to share our findings, and Dr. Vyacheslav Ilyin and Alexander Suvorov for hosting this session. I would like to apologize for my cough. I was unfortunate to catch a flu a couple of days ago. Let me start by introducing myself. So my name is Roman Yunis. I'm a researcher here at the Vavilov Institute of General Genetics. The work that I'm going to present was conducted by my colleagues Maria Alexeyeva, Ilya Dyakov, Marina Dishkova, Natalia Zakharevich, Venera Nizamedinova, and Dilara Anvarovna under the supervision of Professor Valery Danilenko. In my presentation, I will start by highlighting the importance of the gut microbiome as well as bifidobacteria for building a healthy immune system. And then I will present the results of our recent project dedicated to the fibronectin type 3 domains and the role in the evolution of bifidobacteria and host adaptation. So, as much as I'd hate to repeat how important is the gut microbiome for homeostasis, I mean, a lot has already been said today, but still it seems that there is no better time than times of a global health crisis to emphasize this. So to put it briefly, the gut microbiome is a condensed satellite organ that contains just as much, if not more cells than in our entire organism. So this might sound counterintuitive when in reality, I think the role, especially in the early stages of life of the microbiome, it cannot be overstated. And one fascinating facet of the gut microbiome is how it communicates with our own cells. Now, a great deal of attention is usually accorded to the relationship between the gut microbiome and the immune system, which starts to be established right after birth, if not even before that, as few recent studies suggest. And as we grow, the gut microbiome accompanies us throughout our lives. It shapes the immune system, and in return, it is maintained by it later in our lives. So today, the genus Bifidobacterium consists of 86 species and 13 subspecies, which amounts to, in total, to 99 taxa. And they are simply the prevalent genus in infants, and they can continue to be so until weaning, which is why the role in shaping the latter's immune system cannot be emphasized enough. So bifidobacterial colonization of 1-week-old newborns is associated with higher production of IL-5, IL-6, IL-13, TNF-alpha by the age of 3 years old, 3 years. So in contrast, intestine colonization by harmful species, other species such as Enterococcus, Staphylococcus aureus, or Clostridium species in early infancy results in lower production of IL-13, IL-5, and TNF-alpha at 3 years of age. So early life intestine colonization by bifidobacteria also affects T-cell maturation. Now, how do bifidobacteria regulate the immune system? They mainly do it either by directly interacting with immune cells, such as T regulatory cells, macrophages, T helper cells, and others, or via the epithelium. And as you can see on this slide, from these interaction usually ensues an upregulation or downregulation of expression of certain cytokines. So one of the major factors secreted by bacteria are extracellular polysaccharides. Yet the mechanisms and the genes that bifidobacteria evolved to be able to colonize a wide variety of hosts still remain poorly understood. So here I want to point out that when we talk about harnessing the health benefits of microbes, today there's a shift towards pharmabiotics with concrete mechanism of action and a targeted disease. And so what are pharmabiotics? This term was coined originally to address products containing live microorganisms with the intention to prevent or treat disease. And today, by pharmabiotics, it is meant any material with potential health benefit that can be mined from host microbes, dietary interactions in the gut. So there is a great interest in bioactive ingredients that could be mined from bifidobacteria. These could be extracellular proteins and peptides, extracellular polysaccharides, metabolites, and even DNA, since the DNA of bifidobacteria is known for its rich content of CpG motifs, and these were shown to exhibit immunomodulatory effects. So here are a few examples of compounds that are derived from bifidobacteria, and their effects are shown in different test systems. So as I mentioned before, here are a few examples of polysaccharides, pili, and other compounds. These are only just a few examples, of course. The actual compounds are much more numerous than that. And now I will switch to our contribution to this field. So what is the PFNA cluster or operon? Just a few words about the background of the study. And our study was grounded in the assumption that bifidobacteria must have inevitably developed species-specific genes that enable them to communicate with their hosts. So we set out to identify the genes involved in species-specific interaction with the host immune system. And we finally ran against a species-specific gene encoding a serine threonine protein kinase, PKB2, and it was found in most species of the genus Bifidobacterium. Now we know from literature that serine threonine protein kinases can play a significant role in a wide range of cellular processes. And we also found out that PKB2 gene is part of a gene cluster designated PFNA, which is illustrated on this slide. the PFNA cluster is present in most bifidobacterial species, and usually it contains 5 to 8 genes depending on the species. The most common form or structure of the operon is 5 genes. And so the PFNA cluster exhibited also high divergence between species, and transcriptional analysis revealed that these genes are transcribed together in Bifidobacterium longum GT15, meaning they function as an operon. And another key gene of this cluster is the FN3 gene, which encodes a protein containing 2 type 3 fibronectin domains that include cytokine receptor motifs. Now, generally speaking, fibronectin binding proteins enhance the ability of bacteria to colonize their host. And also, this cluster or operon contained a transglutaminide, a microbial transglutaminase, which are known to be responsible for a variety of post-translational modifications of proteins. We also conducted phosphorylation analysis that showed that PKB2 phosphorylates the proteins highlighted here on this slide in bold. So the PFNA cluster was detected in 71 out of 86 species of bifidobacteria, which typically inhabit various ecological niches, including the GIT of mammals, birds, and insects. And the genes of the PFNA cluster, which vary between 5 and 8 genes, depending on the species, are usually transcribed in one direction. As you can see in the first group, where Bifidobacterium angulatum is part of this group. And the second group contain usually between 6 and 7 genes, and its structure was slightly different. Some genes were transcribed in the other direction. And there was also a third group containing the whole 8 genes of the cluster. And the final group was the fourth group, which is not shown in the slide, and where the cluster was absent altogether. And so here I wanted to show you the structure of STPKs in the species Bifidobacterium adolescentis and Bifidobacterium angulatum. This region has a catalytic kinase and a transmembrane helix. STPKs are known for their role in signal transduction. So now this is shown on figure A, and we also calculated the likelihood that certain parts of the PFNA operon were favored by evolution, and found out that the gene encoding a microbial transglutaminase experienced positive selection, which implied it is important for the survival of the species and must therefore be also implicated in host microbial interaction. This is shown on picture B. So what's interesting is that the PFNA operon contained a fibronectin type 3 containing two domains similar to those of humans. And each of these domains has a cytokine cytokine binding motif as part of the structure. And the presence of these domains suggests that these are sensory regions that allow the bacteria to detect cytokines in their environment and react accordingly. So the FN3 domains were relatively similar between human-dwelling bacteria, or more specifically gut-dwelling bacteria, which I will show you on the on the next slide. So this is here the first FN3 domain and the second FN3 domain. Now the first FN3 domain was relatively conserved among the species. Usually there's always a tryptophan and serine in the beginning. Then there's here a random amino acid followed by other amino acids that are more variable in this region. Now the second FN3 domain was more variable than the second one. And based on the second FN3 domain, we actually classified the bacterial species, which I will show you later. And here you can see the majority of these species contain rather conservative motifs. So here you can see how different species have different motifs of the FN3 domain. And the second FN3 domain contained a unique also cytokine receptor characterized by a different sequence for, in the case of Bifidobacterium ungulatum, which is interesting because it is not very characteristic of humans. And so it was likely less similar to other species which inhabit the human intestines. So here you can see four classes of the 6 classes that we actually divided the bifidobacteria into. And to test the hypothesis that FN3 can indeed bind cytokines, we set up an in vitro experiment. And after several rounds of optimization, we were finally able to show that FN3 binds specifically to TNF-alpha. It was not able to bind to neither of the other cytokines that were tested, IL-1, IL-6, or IL-10. And we inferred from this experiment that this protein is possibly involved in immune system regulation. And so today, a number of monoclonal antibodies-based drugs targeting TNF-alpha are undergoing clinical trials. Here, I just mentioned a few of them. And so the technology of neutralization of TNF-alpha using monoclonal antibodies is more or less well developed. And research is currently underway to create cytokine binding proteins based on the 10th domain of the human FN3 protein to neutralize TNF-alpha. Now, in our opinion, fibronectin-containing proteins derived from bifidobacteria are a robust alternative for creating anti-TNF-alpha drugs. And modern FN3 derivatives are already in development due to their advantage over antibodies, which is mainly lack of complexity. And these are called monobodies. So I want to conclude this presentation by emphasizing its main points. A unique cluster of genes, PFNA, involved in species-specific interaction with the host organism was discovered for the first time in human-derived bifidobacteria. And the FN3 protein exhibited high interspecies divergence and sequence variability of cytokine binding motifs. One of the products of the PFNA cluster, the FN3 protein fragment containing fibronectin domains, was found to be able to selectively bind to the tumor necrosis factor TNF-alpha. And it is likely that FN3 domains of various species of bifidobacteria are capable of interaction with various pro and anti-inflammatory cytokines. So the FN3 domains of bifidobacteria are promising therapeutic agents, which are potentially capable of regulating cytokine levels in the host organism and countering inflammation that lies at the heart of many diseases, including COVID-19. So thank you for your attention, and I will gladly answer any questions you have if they come up.
Thank you very much for a very nice and clear presentation. Before other people is ready with questions, I would like to ask you 2 questions. First of all, did you ever measure the affinity of this very domain to TNF-alpha?
Well, I will show you. I will go back to the slide. I don't know if you can see the slide. Yes, and thank you for your question. What we did is we set up this experiment where we had a construction based on. We actually attached antibodies to the surface of polystyrene plates, and then we isolated the protein, the FN3 protein, and had it was bound to the antibody, and this is basically how we showed that TNF-alpha binds to the protein. But this is basically what the whole experiment was. It was sufficient enough to suggest that there's some kind of affinity between TNF-alpha and FN3 protein. Yeah.
Yeah, thank you. But usually affinity can be estimated by numbers, but you have kind of not quantitatively made assay. Did you ever think of inactivation of this gene in the genome of bifidobacteria in order to be sure that it's really is doing that?
Yes, you're totally right. I mean, this is just a preliminary study, so we just came upon these interesting findings, and our next step would be, of course, to validate these results and see whether there's there's some kind of to see if there's definitely a affinity between these proteins, and perhaps also genetically engineer the protein or just modify it and see how will that affect its binding to TNF-alpha. So this will be the next step of the study.
All right. Any other question? Just short one, because we're lagging behind time. All right, Roman. Thank you very much. It was very interesting.
Thank you.
Thank you. We are moving forward with the presentation Low Carb Diet and Microbiome, to be presented by Professor Igor Bychkov from Rostov State Medical University. You are welcome.
Thank you. Just a minute. Okay, respected chairman, respected Congress participants, let me as clinical dietitian share my thoughts regarding low carb diets and their effect on microbiome and health. I'm sorry. There are several types of low-carb diets classified according to the degree of carbs restriction and dietary fiber. An average normal need for carbohydrates and dietary fiber is 300 grams a day and 25 grams a day accordingly. But some types of low-carb diets, for example, keto carnivore, there is a very strict restriction of carbs from 15 grams a day to total absence of carbs. These patients eat only meat, fat, some fish, and some eggs. Properly ketogenic diet, restriction of carbohydrates to 15-20 grams a day, and other diets like modified Atkins diet, restriction to 50 grams a day. A low carbs diet, paleo style, Mediterranean style, with more liberal restriction to 100 grams of carbs a day. Diet change and nutritional changes can be due to various reasons. Some people prefer meat diet, others want to be vegan. The seasonality of food is an important factor that is currently being leveled due to agricultural technologies. Age. Older people are more likely to prefer carb food. Also, it's not right choice. Everyone is familiar with religious fasts, in particular the Orthodox fast with exclusion of all animal products. Dental problems force people to eat more finely chopped food and often ignore the solid constituents of plants. In my practice, if a patient has gut problems, I immediately prohibit him to eat all plants, and this will be very good for him. Ketone bodies, it's a result of ketogenesis during ketogenic diet. Ketone bodies have many functions. You can see these functions. But first, ketone bodies are energetic substrates which are oxidized in heart, brain, and muscle during ketotic states. Also, it controls mitochondrial metabolism and energetics, reduce oxidative stress, Ketone bodies act as signaling molecules, modulate inflammation and immune cell function. And the state of ketosis has versatile effect on the body, and this is not surprising since ketosis has been a physiological state of human body since ancient times. In fact, in ancient times, life required regular adaptation to changing conditions. Our ancestors did not always have access to food and periodically starved when food was scarce. The man was constantly in ketosis, fasting ketosis or nutritional ketosis. Easy transition to ketosis and provision of body's energy needs through ketones ensures the metabolic flexibility. Modern agro-technologies have deprived mankind to be in ketosis, and this is bad. Some people do not experience ketosis in their entire lives. There are many investigations about low-carb diets, but really we have to understand that very often the term high-fat diets is simply mean a modern diet that is high in both fat and carbohydrates. Accordingly, the obtained results cannot be interpreted correctly. Look at this slide, and you can see that high-fat diet is 46% of carbohydrate, but normal level in high-fat diet carbohydrate 5%, maximum 10%, and fat should be up to 70% of total energy. Actually, patients are now informed via internet. They are worried about fiber absence in their diet. And the first question any patient asks doctor after having been proposed a keto diet: what about my gut? My best friend hiccups. And my answer, never mind. There will be new friends, new microbes, new friends, and diet can shift the gut microbiota. Took a look at this beautiful work. Changing your diet can change your gut microflora very quickly. What we see in practice is when, after excluding FODMAP, so-called FODMAP food, bloating passes within 2 to 3 days, stool improves, and persons can fasten his belt, which he couldn't do for years before. This slide, we see how quickly the microflora diversity changes in response to dietary changes. So the real question is, how is it our bodies have the metabolic flexibility to cope with food availability, but our gut microbiome doesn't have the same metabolic flexibility? Did our ancestors really develop unhealthy microbiome every time when starchy carbs were low? We can say that in the body, there is not only metabolic flexibility, but also microbial flexibility. And we can influence by diet in the direction we need. And about microbial flexibility, that is the question about what is normal microbiome. Normal when, normal whom, normal yesterday, normal now, normal 20 minutes before dinner or 2 hours after dinner, and others. Here's some interesting work about anti-inflammatory benefits of keto diet. Keto diet reduces bifidobacteria. Is this bad? We don't know. But ketogenic diet reduces bifidobacteria, but increase Akkermansia and Parabacteroides. The result in reduces levels of in intestinal pro-inflammatory Th17 cells. Well, what happens in the intestines of those who prefer plant foods? Phytobacteria have special enzymes, for example, inulinase, and can metabolize fiber into short-chain acids, acetate, propionate, butyrate. The main one is butyrate, especially for colonocytes. Here is the main butyrate effect. On intestinal level is ion absorption, cell proliferation, differentiation, intestinal barrier function, immune regulation, oxidative stress, and extract intestinal level, insulin sensitivity, cholesterol synthesis, energy expenditure, ammonia scavenger, and others. And that is the question: does keto reduce the production of butyrate? Yes, it does. And what then? Ketone bodies and isobutyrate can replace butyrate. There are three molecules that can replace butyrate: isobutyrate, acetoacetate, and beta-hydroxybutyrate. Isobutyrate is a metabolite of protein fermentation that is typically produced at lower level than butyrate. When butyrate is less abundant, isobutyrate can be taken up from the gut lumen by gut epithelial cells and metabolized for energy. Moreover, isobutyrate can stimulate the same receptors as butyrate in the gut, so-called G protein coupled receptors and GPR 41, 43, and others, and stimulate mucus secretion, antimicrobial peptide release, and immune regulation. And while isobutyrate may be produced at lower levels on a moderate high protein diet, then butyrate would be produced on a high carb diet. Isobutyrate has been shown to be a more potent stimulator of GPR41, one of the primary receptors for butyrate, than butyrate itself. Ketones may help overcome impaired butyrate intake. The ability to use ketones instead of butyrate may not seem advantageous until you consider that many people with inflamed guts have mucosal damage, where butyrate uptake is impaired. If somebody has a healthy microbiome and gut mucosa, butyrate is sufficient for gut's needs. No ketones needed. But if patient has ulcerative colitis or other mucosal damage where butyrate uptake is impaired, or he has gut dysbiosis characterized by a lack of butyrate producers, or he is on restrictive diet such as a low FODMAP diet, resulting in reduced butyrate production. It may be wise to try therapeutic nutritional ketosis to support gut epithelial cell metabolism. And this is, to my mind, the main advantage of fat diet, of high-fat diet, ketogenic diet, to support gut metabolism without fiber. No evidence that a good practice keto diet is detrimental to health. Keto-induced changes in the gut microbiota and gut barrier may even be the reason we see so many benefits from ketosis. Ketogenic diet is not right choice for every patient, but it's really necessary for those who suffer from obesity, autoimmune diseases, cancer, neurodegeneration, and gut inflammation. Thanks for attention.
Thank you very much. Thank you very much, Professor Bычков. Who wants to ask the questions? Questions, please.
Okay, I will ask a very short question, which maybe I didn't understand correctly. So your opinion is that keto diet is basically can supplement Mediterranean diet, or can be as efficient as other variant diet with fruits and vegetables?
Ketogenic diet is a strict protocol for severe diseases such as, for example, autoimmune diseases, allergy, cancer, and others. And Mediterranean lifestyle diet is for healthy persons who want to be in good condition, to be healthy.
So, so it is more therapeutic diet. Is it correct?
More what?
More therapeutic diet, according to what
Keto diet? Yes, it's therapeutic diet. Yes.
All right. Thank you very much.
Thank you very much again. And we move forward to the next presentation, reduction of conditionally pathogenic bacteria in gut microflora in children with functional digestive disorders after treatment with autoprobiotics and industrial and enterococcus strains, to be presented by Dr. Konstantin Yermolenko from the Institute for Experimental Medicine. You're welcome.
Thank you very much for your interpretation. Thank you for opportunity to take part in this wonderful meeting. And I want to start with the words that I'm unfortunately not the senior researcher of Institute of Experimental Medicine. I'm a researcher of Pediatric Research and Clinical Center of Infectious Diseases, but Institute of Experimental Medicine is the main source of ideas for our topic, and the institute with whom we had a wonderful collaboration. And I want to thank my colleagues, Natalia Gonchar, Alexander Suvorov, Galina Alyokhina, who helped in this work, who made the most of this work. And it's a great chance for me to present the data which was which we had in this wonderful collaboration. First, I want to start with very provocative thing to say is that not all probiotics are always effective. In some cases, probiotics can lead not to stabilization of microbiota, but can even worsen the dysbiosis. And sometimes widely used strains of probiotics, in contrast to indigenous bacteria, they can cause some harm to microbiota itself. As we see here, there is a different types of microbiota, and in different people, we can see different effects made by different strains of probiotics used in our medical practice and used in everyday treatment in children and in patient adults. We can see that according to different probiotic strains we use, we can have different effects on immune system, different effects on morphology of enteron and colon. And of course, different probiotic strains act differently against different commensal bacteria and different strains of other symbiotic bacteria. For some reasons, it is good. In some conditions, it can really be effective. In some cases, it can harm colon and enteron and lead to some bad consequences. Of course, if we speak about future life, we can speak about personal therapy, and one of the best solutions for probiotic therapy in future will be personalized probiotic therapy, the design of novel experimental trials, which will include only probiotics which will be administered according to microbiota type in a patient, according to his specialty and his peculiarities. One of the solutions with which we already have now is autoprobiotics. This is the strains of normal microbiota isolated from specific patient, specific individual, and these drugs can be used for treatment of different conditions. There is a big data on different probiotic strains which can be isolated and can be affected. And of course, if we speak about advantages, they are very common. We can see that it is a long-term effect. Microbiota, indigenous microbiota, has very high chances to stay in intestine for a long time. And of course, there are some difficulty, especially in how to prepare it, how to make it safe for patients. And of course, we need some kind of biobank for every patient to store this microbiota, as it could be effective in special conditions which cannot be in the time when this autoprobiotics was made. There are different types of autoprobiotics. We know that even now we have both monostrain, multistrain, and even consortia of autoprobiotics, which can be used in different conditions and can be very effective. And one of the conditions in which it can be especially effective is different kinds of functional gastrointestinal disorders. We know that microbiota changes are one of the main part of gastrointestinal disorders pathogenicity, according to Douglas Drossman conception, who said that different factors like genetics, culture, food, what we eat, different kinds of infections, immune dysfunctions, and gastrointestinal infections, they can lead to different consequences like visceral hypersensitivity, and as a result we can have different signs of functional gastrointestinal disorders. Visceral hypersensitivity, one of the most important pathogenicity pattern in these patients, is very closely connected to intestinal dysbiosis. And we can see that normal microbiota, it makes a lot of efforts to stop visceral hypersensitivity, to block immunopathogenical activity by regulatory T cells, by dendritic cells, and opposite to this, when we have dysbiosis, we have a lot of B cell producing immunoglobulin G, we see local inflammation, and of course we have immune dysregulation. As a result, we see different kinds of functional disorders. One of the most important of them is irritable bowel syndrome, and it affects children up to 3 years old, and is one of the most common reasons for long abdominal pain, long diarrhea or constipation, and is one of the most important problems in gastroenterology. There are some minor forms like functional constipation, functional diarrhea, and some others, but mostly we speak about irritable bowel syndrome. Of course, probiotics are a good drug of choice for treatment of irritable bowel syndrome, but previous studies showed us that not all probiotics show very good positive effects in treatment of irritable bowel syndrome. And of course, the choice of probiotic strains is a very difficult question for every particular patient, for every doctor. And of course, we need to find some data which will help us. There is a good study made by Professor Mahoney who showed us that Bifidobacterium infantis can have positive effect on treatment of irritable bowel syndrome in elderly patients. We have a lot of studies which show that there are some potential new candidates like Prevotella copri, Akkermansia muciniphila, and some inulin producing prebiotics, which have shown in some clinical trials good effects in different kinds of functional gastrointestinal disorders. Of course, we can speak about Faecalibacterium prausnitzii, one of probiotic strains which can have very good results, but we don't have a lot of probiotics which can have this strain and which is very effective in long-term effects. Another possibility for treatment of irritable bowel syndrome is the fecal microbiota transplantation. We have a work by Professor Vlasov who showed that it is very effective, but on a very small group of patients. And of course, we cannot forget about the possibility, danger for our small patient who can be affected by some retroviruses which can be transmitted with FMT and some other cases of some other microbiota members who can be very dangerous for small patients. And one of the potential drugs of choice for a small patient, for children, is Enterococcus faecium, mostly because it can produce bacteriocins which are studied well and which can be a very important factor for inhibition of semi-pathogenous flora and stabilizing of microbiota all over. Of course, Enterococcus faecium also has an ability to produce some micronutrients and vitamins and can improve motility. And of course, there is no difficulties in cultivation that makes it very good for production. We know that up to now there is a good technology how to colonize typical Enterococcus faecium, how to make pure culture of this microorganism, and how to select apathogenic Enterococcus faecium for our patient. And up to now, we have a lot of patents made by Alexander Suvorov, by Vladimir Ilyich Simonenkov, who had wonderful results in adults. Unfortunately, up to now, we don't have a lot of data about effects of autoprobiotics of Enterococcus faecium in children because of some peculiarities which are common in young children. And our aim was to identify the features of the influence of both autoprobiotics and Enterococcus faecium on clinical and laboratory parameters in children with functional gastrointestinal disorders. We had a group of 33 children which were controlled and studied in Clinical Research Center for Infectious Diseases in St. Petersburg in 2020 to 2021, and several studies were taking place for every patient, both study of FETSES, coprological and real-time PCR, including colonoflora primers. Both groups were common. One of them received autoprobiotic strain, and 18 children received industrial probiotic strain Enterococcus faecium. We included patients elder than 3 years without any prior symptoms before inclusion, no signs of inflammation in gut, and of course no comorbid somatic or neurological and endocrine pathology. All groups didn't receive probiotics for a long time before they were included in study. If the person didn't agree for the study or there were some side effects, it was they were included from the patients. 70% had irritable syndrome, and for some of them we had a functional dyspepsia or functional diarrhea. Some patients got atopia or chronic infectious diseases which could affect their visceral hypersensitivity. And after the treatment, most of patients had positive dynamics. Only one patient in first group and 2 patients in the second group had some symptoms which followed after administration of probiotic or autoprobiotic, and most of patients have very good positive dynamics. If we speak about study of FETSES, most of patients had dynamics. It was seen in the amount of detrite and some leukocyte in FETSES, which didn't take place in patients who received both probiotics and autoprobiotics after treatment. We saw some significant changes in microbiota before and after treatment with probiotic and autoprobiotic. As we see the red bars, it is the average amount of different microbiota strains in patients who received autoprobiotics, green one for those who received enterococci. And we see that the most probable effect was seen in the increase of symbiotic bifidobacterium and Faecalibacterium prausnitzii in patients who received autoprobiotics. Another thing that was seen in both groups is that the level of Faecalibacterium prausnitzii was much higher in patients who received autoprobiotics. It was much, the average level was much higher than in group 1. And of course, it is very important to say that opportunistic bacteria
Konstantin, not much time.
Yes, the growth of opportunistic bacteria decreased rapidly after the consumption both of Enterococcus faecium and autoprobiotic. It is also important to say that autoprobiotics had very significant effects on anaerobic imbalance, and that effect was most profound in patients who received autoprobiotics. So we can say that both Enterococcus faecium and autoprobiotics are very effective in treatment of children with functional gastrointestinal diseases. Due to the lack of time, I will not read the conclusion. For the second time, you can see it on your screens, and I will give the word to our other speakers. Thank you.
Thank you very much for your very interesting presentation. Who wants to ask the questions? Any questions? Everything is clear. Thank you very much. Thank you very much,
Thank you.
Dr. Yermolenko, for your very interesting presentation. And we are moving forward to presentation health benefits of probiotics and postbiotics on gastrointestinal health, to be presented by Professor Sae-Hoon Kim from Korea University. You are very welcome.
Hi, I am Sae-Hoon Kim from Korea University. The title of the talk today is Health Benefits of Probiotics and Postbiotics on Gastrointestinal Health. I'd like to thank the organizing committee to invite me on this great event. I've been working on this lactic acid bacteria and their functions for more than 30 years now, and it feels like that these days are best of all. It seems like blooming up. It's because of that blooming of the understanding of gut microbiota. As you all know that the human body has quite a bit of a microbiota, and 95% of them are residing in the gut. So human gut is home to 100 trillion microbial cells, and having a genome size of at least 10 to 100 fold greater than human itself. And most of them are still unculturable, where we cannot understand too much of it up to now, but the technique is available to analyze the genome these days more better and cheaply. So much of information are coming out and will be available, so that we are more interested in the gut microbiota these days, which makes this probiotic business blooming also. As we know that the gut microbiota surely affect the well-being of the gut itself, gastrointestinal tract itself, but it doesn't stop there. It will go beyond the gut to the brain and bones and livers and almost every place of the body. It affects it and being affected by. So this gut microbiome is has an effect on the health of other parts of the body, as well as is being also affected by whole body conditions. So it's not one-way communication, but two-way communication. So to regulate or modulate the gut microbiome, you may need a lot of conditions to be changed. But there's something that we cannot do anything about it, except a few of them, including having more sunlight, exercise, and having better sleep, and so on and so forth, which, even if we changed it, you may see the good result in million years. You have to wait before you see the good result. So there's only a few things like a proper diet and to cause against the antibiotics and others are the only way to directly modulate or regulate gut microbiome. So that's why the probiotics or prebiotics are being a hot issue these days, because they can directly modulate or regulate gut microbiome. So the including probiotics, postbiotics, and prebiotics, the one that can regulate and modulate. These days they're doing fecal microbiota transplant. To do that, they get healthy feces into the intestine of the unhealthy individual, make it changes and regulate the microbiota and improve the condition of the diseased individual. And in here, probiotics are the living microorganisms that benefit the host health when administered in adequate amount. And the prebiotics are the substrate to be utilized by the probiotics or the host microorganisms, so that it can affect whole array of function of gut microbiota. The term postbiotics are rather recent one. It is not live microorganism, but the fragments or portion of the the probiotic microorganism, as well as the metabolites of the microorganism, and then some traditional fermented food or the fermented natural product may be included as in postbiotics. So I'm talking about the effect of probiotics and postbiotics on the gastrointestinal health. As you know that gastrointestine is having an array of epithelial cells, and then in between these cells having a space, intracellular space, that is tightly regulated by so-called tight junction proteins, so that not much of the content in the lumen can pass through this space of the epithelial cells. But something happened that the changes in the microbial composition, so-called dysbiosis, induced more inflammation and make it more permeable in between these two cells, lessening the structure of the tight junction proteins, and more of toxic material can pass through that cause more inflammation on the gut, but it's not stopped there, but this is delivered systemically through the blood system to the liver and other parts of the body to cause more trouble with that. And the stress is another causative agent to damage on colons and as well as other parts of the body. So we want to use probiotics and postbiotics to regulate the gut microbiota so that it keeps this epithelia healthy to be good tight junction, not to allow the permeation of the toxic material from the lumen to the body, systemic body of the gut. Colorectal disease is many types of diseases is that, and here I want to cover 3 types of diseases. One is irritable bowel syndrome, that including the abdominal pain, cramping, and bloating, as well as constipation and diarrhea, and it seems like it keeps increasing and expected to be increased more. So they categorized these constipation type IBS-C, diarrhea type IBS-D, and mixed type, both diarrhea and constipation happen alternatively. And then even more serious condition is inflammatory bowel disease. It's actually having severe inflammation on the gut, so it causes diarrhea, rectal bleeding, and severe abdominal pain and others. Types of IBD so far is either in ulcerative colitis or Crohn's disease. Ulcerative colitis having inflammation in a certain point of the colon is more likely to the end of descending colon, but the Crohn's disease is having inflammation all around the gastrointestinal tract, not specific position. So, and then they kept increasing and seems like stay there, but it's another headache for the gut health. And then it comes to the cancer, because if the inflammation goes on and continues, it is more likely develop to the cancer, in colorectal cancer. So we want to check whether this gut microbiota regulation having effect on this cancer also. So we want to find out by modulating gut microbiota by probiotics or postbiotics, whether they have effect on the irritable bowel syndrome. First, what we used is the probiotic strain of the Bacillus and Lactococcus species, we use not just alone the microorganism, but using with the chocolate on the induced constipation model of mice. And when it is induced, the constipation, the feces is a very much of a constipation phase. But when we use the probiotics in a chocolate, it recovers, seems like it recovers much to the normal state as compared to the controls. And here we found out that since we treated with the probiotics, it seems like the serum cholesterol level was lowered, even though we didn't expect too much of in this characteristic. And the intestinal motility increased by the treatment of the chocolate with the probiotics, and then permeability through the tight junction area is much lessened by the treatment of the probiotics with the chocolate. And colon length was increased by the treatment. And then the tight junction proteins, claudin, GO1, occludin, all increased by the treatment of that, the probiotics with the chocolate. And inflammatory markers like IL-8 was increased by the treatment of the inducing constipation, and it decreased it. And then MUC2 is a mucin producing gene increase, so that it may produce more of a mucin to protect the permeability. And then the gut microbiota change is observed. The Firmicutes is increased a bit, while the Bacteroidetes remained the same. And then because we treated the Lactobacillus and Bifidobacterium, it increased it, and while the Enterobacteria CEA is decreased. So we did this formula, the probiotics with the chocolate. Using that, we did the human trial for 4 weeks, and we feed about 90 individuals with the chocolate probiotics. We found out that the Bristol stool score improved fairly well, as well as Lactobacillus and Plantarum goes up because we fed them. We didn't see much of inflammatory signals in the blood. We did pretty much the same treatment, the human trial, but this time with the yogurt with the different probiotics, and about 50 individuals are being tested for that, and found out that total anaerobic bacteria was decreased, and then the lactobacilli we treated was increased, while the Clostridium difficile and E. coli is decreased, while increasing the level of lactobacillus. And we didn't expect too much of changes in the inflammatory cytokine like IL-12 and TNF-alpha, but it seems like increased a little bit, and we have to see why is that and for further. But the abdominal pain and the constipation frequency were significantly reduced by the treatment of this probiotic yogurt. And then one of the causative reason for the IBS is stress. So we induced the stress to the mouse model and see whether if there are changes in the IBS syndromes. So we do the chronic, unpredictable chronic stress onto the animals and treated with either probiotic strains or fermented product of this probiotic. As you see here, this is a normal length, the colon length of the normal mouse, and then when it is stressed, the colon length is shortened, while the treatment of the probiotic or fermented
Thank you very much for your presentation. It is recorded, so there will no living discussion, and we move to the second presentation. Is probiotics as food additive can affect the weight gain of poultry chickens. To be presented by Professor Alexander Svorov from the Institute of Experimental Medicine. You're welcome.
Dear colleagues, dear head of the meeting, I decided to present this lecture out of schedule just because of the results, which I found very interesting, and I think as long as here we have a lot of people interested in agricultural aspects of usage of microorganisms, I thought that the thing we found worth listening. So I will try to be really quick because we are lagging behind time. I just want to say that this is the product of collaboration of Russian and Chinese group of scientists. We managed to organize our institute together with the Institute of Agriculture in Shandong province, Sinan, so-called Russian-Chinese Center of Microbial Ecology. And we performed the experiment with probiotics fed to the chicken in order to see how probiotic can compete with kind of getting stuck. Okay. So as I said, the research objective was to see how if you add antibiotic like ernomycin is allowed in China to be added to the chicken food, whether it can be supplemented with probiotic. So we decided to use a probiotic which we know very well, which we studied very much in our department, selected about 25 years ago. I've done whole genome sequencing, and we use this probiotic in Russia as a food additive, as a health beneficial food for quite some time. There are several products on the Russian market, and we know that it is health beneficial and safe, and it has very many interesting features. And for this purpose, what we were targeting is antimicrobial activity of this very strain, Ifesium L3, because this very strain carry genes of 5 different bacteriocins, which is quite rare even for Enterococci. This is an example of the operon with 2 different bacteriocins in the strain, and we know that this very strain in the organism work as a strong anti-inflammatory agent, and which is very helpful in case of the inflammation. So we know also many things like usage of this probiotic for the purpose of treatment of neurological conditions like multiple sclerosis. This is an experiment when we are treating animals with multiple sclerosis on the model. And presently, when we have these epidemics of coronavirus, we use this very strain as a basic strain for introduction of the gene of S protein of coronavirus genome of this very probiotic in order to get new probiotic-based vaccine against coronavirus. But this is a different story. This very story is related to chicken. This is few things which I just mentioned, like clinical applications and its efficiency against H. pylori infections, chronic liver infections, Parkinson's disease, multiple sclerosis, and as I said, now we want to make it as a vaccine. But here we have done the experiment in Chinese ground in a very well-equipped laboratory with ideal selected animals, which were broiler chicken, which had the diet, starter diet, which at day 21 was switched with a grower diet. And in both cases, probiotic group was getting 10 to the 7th colony-forming unit of Enterococci in their food, which is money-wise is extremely inexpensive. So these are the groups under the study, and you can see that here was a kind of starter diet which was switched to the grower diet in each group. And basically we have the group of control, group of antibiotic, and group which got Ifesium L3. Very simple. And when we looked what was going on at first, probiotic group was lagging behind the other groups. Even antibiotic groups was doing the best. But wait a little. After 39th day, we have dramatic increase in probiotic group relatively to antibiotic and group of control, and it was statistically relevant. So, and all these weight parameters, which I'm not very well informed before, you can see that carcass weight increased, chicken weight increased, body weight after slaughter increased, and feed to meat ratio dramatically decreased. It means that you can provide less food and get more meat. That's what we are looking for. As long as it was experiment, we were interested in what's going on with biochemical parameters like cholesterol level, lipase level, high density, low density lipids, which you usually test for different animal studies and human studies as well. We also tested for immunoglobulins, but we didn't see anything significant. But with this cholesterol level and blood parameters in the biochemistry, we saw dramatic changes as well. And what was extremely interesting, that high low density lipids, which are really dangerous, was growing in antibiotic group dramatically relatively to other groups, and you can see that probiotic group didn't work as well. So talking of diet, the lecture before this one was very kind of complementary to our data, because we saw that after switching from the starter diet to the grower diet, we saw that microbiota in all three groups did change. And it switched from one group on the enterotype level to another group. And we can see that, as we saw it on the rat models before, that antibiotic group was also sitting in the same very place, which shows that basically getting antibiotic is kind of depleting the number of bacteria, is basically causing the damage to your microbiota. If you look at the alpha diversity of this chicken, we have done all the genome analysis. You can see a lot of different species. I'm not mentioning that, but you can see that the numbers of different species is growing, which means that alpha diversity is growing. And what was important for us, as I was mentioning before, that proteobacteria is one of the most important indicators of kind of bad condition of the microbiota. So you can see that in case of antibiotic, you get significantly different increase of proteobacteria in the gut of the chicken. So what we can see that by the day 39, we have increase of proteobacteria in antibiotic group and gradual growth of Lactospiraceae, Bacteroides fragilis, and proteobacteria. And this is the result of our analysis, which we have done employing Python study analysis of the microbiota, because sometimes you get too many data and it is very hard to kind of comprehend what's going on in reality. So we took 4 main by the abundance species or groups of microorganisms, which were main microorganisms in the chicken's intestines, which were Bacteroides fragilis, which were proteobacteria, Fecalibacteria, which we consider beneficial, and Lactobacillus. And if you put it all together, in comparison with Bacteroides fragilis, what you can see that in group A, which is just control group, you can see that eventually you have a decrease of proteobacteria, which is normal, and slight increase in Fecalibacteria, with Lactobacillus with little bit decrease in Lactobacillus in the intestines. But look at the antibiotic group, which is group B. You have dramatic change in Fecalibacteria, decrease of this Firmicute group, and increase in proteobacteria relatively to this indicator group, Bacillus fragilis. But if you look at probiotic, you have an opposite situation. You have increase of Faecalibacteria and decrease of Proteobacteria, which means that basically you get this weight gain, changes in lipids, which are beneficial, basically because of this switch in the composition and arrangement of bacteria in the intestines of the chicken. So you use, if you use the same test for correlation of this species with biochemical parameters, you get pretty much the same. In the group of probiotic, you get decrease of low-density lipids and significant decrease of cholesterol, basically. And in group of antibiotic, what a surprise, you get an increase of low-density lipids. So basically, you feed the animals with antibiotic, you get worse results regarding the weight gain, and you get worse quality of food, basically, which was exciting for me. That's why I was talking about this. So we get the weight gain of the animals by the 39th day of life for 100 grams at the average. And we see the significant differences in the content of lipids and blood parameters in the cecum. Thank you very much for your attention.
Thank you very much, Professor Svorov, for your very interesting presentation. Who wants to ask the questions? Please.
Конверсия корма. И что можно сказать о нем?
Вы имеете в виду замену корма?
Ну, скажем,
когда
ваша, скажем, группа с антибиотиком или группа с пробиотиком, скажем, меньше съела корма. Вот на единицу привеса сколько было затрачено корма?
Конечно,
вот
да. Yes, we tested this parameter. I just didn't show it, but it will be published. I hope we will present the publication in near future.
Please, Professor Chikindas.
Thank you for the very interesting presentation, and we hope to see your paper published in the very near future. A question. I presume that the antibiotic used as a reference was one of those that are or were used as growth promoters in poultry. Is that correct?
I was looking in the literature on this antibiotic. So this very antibiotic, it is supposed to be growth promoter, and it does provide a little bit of growth and for the chickens, and it is the only one which is still allowed in China.
All right.
That's why it was used in this experiment, because now they are lowering down on antibiotics, but still use it.
And the related question, okay, the story looks like a miracle, that you were so lucky to find this, beautiful strain that works. And yet I remember you were talking about, let's put it from the time being in quotes, an algorithm for selection of the best probiotic candidates. In this regard, can you say anything, if there is anything known at this moment in time, what exactly is so special about this strain? Because it goes back in history many years, decades, and we're supposed to know it. It's like, it's been used for humans, and now suddenly you find new application, And indeed, a very important thing, we're talking about healthy food, and you're able to reduce cholesterol and to have, not to have these negative effects as we observe in the case of antibiotic feeding. In your opinion, what is so special about your strain?
It's highly antagonistic. So for this, what do you expect from antibiotic? You expect to kill pathogens, pathogenic bacteria. So in this very scenario, you want to find something which is highly antagonistic and can do the work of antibiotic as a replacement for antibiotics. That was our logic behind this experiment.
Thank you. Thank you.
Thank you.
Just one announcement. As a co-chair of the entire event, please blame me, but I suggest to perhaps maybe skip the coffee break if you think we should have 5 minutes here just to catch the breath, but then to start again the second session in 5 minutes because we're behind of our schedule as it was planned. So I think with this we are closing the first session of the human probiotics. I would like to thank the co-chairs for handling this event. It is still very interesting and keeps us alive and awakened. 5 minutes to catch the breath, and we're back at 4:30. Thank you.
So please take your seat. We are starting the second session dedicated to human probiotics, and with presentation effects of Enterococcus faecium probiotic intake in patients with type 2 diabetes. To be presented by Dr. Anna Tsapieva from the Institute for Experimental Medicine. You are welcome.
Hello. Okay. Dear colleagues, thanks for hosting us here, and I'm very happy and proud to tell you about our work. So today I'll tell you about the effects of IFIDs and probiotics on the patients with type 2 diabetes. So as all you know, type 2 diabetes is a metabolic disorder which is characterized by high blood sugar, which is caused by the insufficient insulin. I'm sorry, I'm nervous. That's new for me to present and to be somewhere offline. So it's insufficient insulin secretion and insulin resistance, both. And people with type 2 diabetes, they have, how to say, it's low quality of life. And probiotics could help them to feel themselves better. And as you know, a lot of reasons why we should take care of our intestinal microbiota, because imbalance can trigger a lot of serious diseases, and type 2 diabetes is one of them. So altered intestinal ecology could lead to low-grade inflammation, which can be the reason of rapid progression of the insulin resistance. So here comes the probiotics, which are needed to help people with different diseases, and in particular type 2 diabetes, to take care of the intestinal microbiota. And a lots of work during recent years have been dedicated to the link between the intestinal microbiota, type 2 diabetes, and probiotic consumption. So as you know, as Professor Suvorov said, I'll skip this, sorry. And as you know, Enterococcus faecium strain is a strain which we use for the long time in our lab. And here are some publications for the last 10 or 15 even years. And it is the strain which is very effective for the lots of different diseases. And as we very closely related already to this strain, so we used it for the patients with type 2 diabetes when they had a big work with some doctors from other institutions in St. Petersburg. So here are the results from our colleagues about the maltose activity of the enterocytes after the consumption of probiotics. Here we wanted to say about the potential of Enterococcus faecium L3 strain. So you can see that in green column that if the strain can degrade the maltose, then the epithelium don't need to do it. So here is the results, also the maltose activity in the colon, in the different parts of the colon. And you can see that if probiotic Ifeциum is consumed by the patients, the maltase activity is lower than the control group or in the group of other probiotic which don't have maltase activity. So it was just a small introduction again to Enterococcus faecium L3 strain and why we used it in our study. So this study design is 23 patients with type 2 diabetes. They received Enterococcus faecium L3 strain probiotic for 20 days. And before and after the treatment, blood and fecal samples were taken. Blood were tested for biochemical parameters changes, and fecal samples were tested for microbiota alteration by real-time PCR. And during the study, patients filled in the gastrointestinal diary. So what we saw after the study, we didn't see any miracle, but still we saw that how to use here. Here is the only parameter in this study which were significant, and this is C-reactive protein. And we saw that the concentration of C-reactive protein in the blood of the patients decrease. And that is very important for the patients with type 2 diabetes because we can tell about the lowest in inflammation. So we were happy to see that. Here is microbiota parameters shift before and after the probiotic therapy. Here we saw that here we saw the decrease of some opportunistic pathogens such as Escherichia coli and Staphylococcus aureus, and increase in a trend to increase in Faecalibacterium prausnitzii and decrease in Bacteroides species. So for the patients with type 2 diabetes, it is known that they have high levels of Bacteroides species in their colon. So then we saw that after the treatment of Enterococcus faecium L3 strain, the number of Bacteroides goes down. That was interesting for us, despite that Bacteroides species is not a parthistic pathogen. It is a normal microbiota of the human. So nevertheless, that is an interesting information for this study. And here are the results. I've been shocked. So we seen the positive effects of probiotic therapy on the severity of gastrointestinal complaints. I didn't say it during my talk, but all the patients filled in the gastrointestinal diary, and so the level of the pain in the gut, dyspepsia, and diarrhea or consumption. They all the patients filled in the diary, and all the patients didn't have any side effects, but they all said about the good well-being, and so they were happy to take the probiotic. And we saw the decrease in level of C-reactive protein in the blood, and other parameters were had insignificant changes. We saw the significant decrease in opportunistic bacteria in the intestinal microbiota. We saw the decrease of Bacteroides fragilis level, and we saw upward tendency in intestinal lactic acid bacteria level. Here are the authors of this study. I presented just a little part of it, which were dedicated to the Enterococcus faecium L3 consumption, but the whole study included the autoprobiotics as well. And thank you for your attention again.
Thank you very much for your very interesting presentation, Dr. Sapieva. Who wants to ask the questions? You are welcome, Dr. Chikindis. Professor Chikinus.
Thank you for your very interesting presentation. It's not like a question, it's more perhaps as a suggestion and discussion, just a quick note. The type 2 diabetes predisposition is a growing issue with a better understanding of the prehistory, especially when it starts in younger children. All right, and we can identify nowadays the families where we can trace the predisposition to type 2 diabetes. And I think this type of research may really expand to study how the probiotics may help in children who are predisposed to type 2 diabetes. Whether these kind of probiotics can help them to stay in the healthy status and not to develop type 2 diabetes. Of course, diet is another issue. And another thing is just a note of addition, you know, to plant some seeds in your brain if you wish. And because I know that the group of Dr. Suvorov is heavily involved in work with Chinese scientists, the magic word CHENPI, C-H-E-N-P-I, a handful of data published on CHENPI, which is the Chinese orange peel, effect on the diabetes in different groups. Well, CHENPI is considered to be a kind of a miracle in China, you know, the silver bullet for many types of health disorders. But in addition to other things, a significant number of reports were published on its effect on diabetes. And because nowadays probiotics are merging, going hand by hand with phytobiotics, you may consider also taking this path for a future investigation, which is absolutely very interesting and intriguing. Thank you.
Thanks a lot.
Who else want to ask questions? I should like to make a short question. You mentioned, maybe I missed something, but you mentioned in one of your results about tremendous decrease of opportunic microorganisms. You mentioned, I have seen it concerning Staphylococcus aureus. It's very good result, but maybe some other, also some other representatives of opportunic bacteria also decrease.
Yes, E. coli.
E. coli.
As well.
Pathogenic, yeah, lac minus.
In general.
In general.
Yes.
Okay, thank you. Thank you very much. If no questions, so we move further to presentation: genes, bacteria, methanogenic signature of humans gut microbiota as a biomarker of depressive conditions, to be presented by Dr. Alexey Kovtun from Vavilov Institute of General Genetics, Russian Academy of Science. You are welcome.
Hello, my name is Alexey Kovtun, and today's topic is genes, bacteria, metagenomic signature of humans' gut microbiota as a biomarker of depressive condition. Currently, microbiota is viewed as an independent neuroendocrine organ that is connected with host organism via bidirectional symbiotic gut microbiota axis. The interactions between the microbiota, gut, and nervous system, including brain, occur via neuroendocrine, immune, and metabolic pathways. The human's gut microbiota is capable of producing hundreds of metabolites that can directly affect most systems and organs, including nervous system and brain. According to the latest data, gut microbiota also plays an important role in the development of psychic and cognitive disorders. The normal state of the gut microbiota can be described with stable core taxonomic components. Any disturbances in the composition of microbiota during the first stages of the disease may affect the composition of its primary core taxa. And as a result, abundance of bacterial genes will be altered as well, including those involved in production of some key beneficial metabolites. Resonations in bacterial composition of gut microbiota correlate with various psychic and cognitive disorders such as depression, as it has been shown in some recent research. Few words about depression. It is nowadays viewed as a global threat to mental health, and it is ranked fourth among the causes of disability worldwide, affecting every fifth person in developed countries, according to the statistics. Major depressive disorder is a complex and heterogeneous psychiatric disorder that is revealed in patients based on their narrative, and only it. And over the last decades, many literature reviews have scrutinized the relationship between the gut microbiota and depression. However, it remains imperative to identify the biomarkers correlating with the symptoms of the depression. So in our ongoing research, supported by the Russian Science Foundation, we try to detect changes in the taxonomic composition and functional properties of human gut microbiota that correlates with depression. For that purpose, we developed the reference catalog of amino acid sequences of homologs for bacterial enzymes. It involves information and degradation of compounds with neuroactive potential. We conduct metagenomic analysis of gut microbiota of patients with depression and healthy controls, and try to determine biomarkers of gut microbiota that correlates with the depressed condition. Currently, our cohort consists of 29 metagenomes obtained from 14 patients with depression and 15 healthy volunteers. All the samples were sequenced Illumina High-Cyclics 10 sequencer. So we obtained the whole metagenomes. One of the main stages of our research was the revision of our previously developed catalog that's already included bacterial enzymes involved in formation of neuroactive metabolites used by gut bacteria. We added more enzymes involved in metabolism of compounds which correlate with depression. And thus, our revised catalog overall includes more than 1,000 minute sequences for 101 enzymes. In our research, we operate with a feature called metagenomic signature. It is an object that combines both taxonomic and functional properties of gut microbiota, and basically it's a simple matrix which describes which genes originate from which bacteria in gut microbiota and with what relative abundance. The algorithm for detection of these genomic signatures is as follows. First, we detect some open reading frames and annotate them using BLAST and our developed catalog. Next, we classify the origin of these open reading frames and finally combine them into metagenomic signature and count their relative abundances using Gregson, Perl, and R. And we also count some statistics to determine some significant changes between the groups. As a result of the analysis, we have obtained more than a dozen signature pairs during speeches that are statistically significantly decreased in patients with depression. After that, we conducted a more traditional taxonomic analysis using method plan 3, and we discovered that the upper diversity on the species level is slightly decreasing patients with depression, though not significantly. And more importantly, we have discovered that one of the species called Ficalobacterium proutnitzi is statistically significantly decreased in patients with depression. It is interesting because we have discovered that several genes from these species have been also decreased in patients with depression, which can be connected. On the current stage of our research, we have revised our existing catalog and included more enzymes involved in formation degradation of compounds with new reactive potential. All for the one simple cause, detection of metagenomic signatures of gut microbiota in patients with depression. We have conducted metagenomic analysis and identified 15 signature pairs gene species in the group of patients that have been statistically significantly decreased. And in conclusion, I'd like to add that the obtained data of comparative genomic analysis correlates with published data and indicate disturbance in the composition and functionality of the gut microbiota in people with depression. For the first time, the data on metagenomic signatures of gut microbiota of patients with depression were obtained. And these data will be used for further comparative genomic analysis of the gut microbiota for selection of biomarker genes composition. The paired signatures as a universal marker will also give us an opportunity for studying various characteristics of gut microbiota. Neuromodulatory, immunomodulatory, its antioxidant potential, and many others. And as I have already mentioned, this research is an ongoing one, and in the end, we hope that our results can further be used for the development of methods for correction of gut microbiota of patients with depression, including the use of prebiotics and postbiotics. Thank you for your attention, and thanks to all the participants of our project that
Thank you very much.
These results.
Yes, thank you very much for your very interesting presentation. Who wants to ask the questions? Who wants to ask the questions? Nobody wants. Everything is clear. Thank you very much again, and we move forward to the next presentation. Is changes in the intestinal microbiota of patients with inflammatory intestinal diseases after healthy intestinal microflora transfer, to be presented by Professor Vitaly Morozov from the Institute of Chemical Biology and Fundamental Medicine, the Siberian branch of Russian Academy of Science. You are welcome.
Добрый день, глубоко уважаемый председатель, уважаемые коллеги. Пока мне еще не подан знак о включении переводчиков, поскольку доклад будет на русском языке.
research of a part of a work, of a bigger work. The development and the widespread use of NGS, next generation sequencing technologies, characterize microbial communities, including human intestinal microbiota. At present, human intestinal microbiota is considered as a dynamic system under the influence of the host organism and external factors. It is known that the main components of the normal human intestinal microbiota are representatives of primitive and Bacteroidetes. Although the core group of particular bacterial species does not coincide in different healthy bodies. It should be taken into account that different variants of the normal microbiota are able to provide the stable functioning of this microbial community. It is believed that the main functions of intestinal microbiota include not only digestion, but also direct protection against undesirable agents, immune system effect, and proliferation and differentiation of epithelial cells control. Today, there has been much talk on the disorders of the intestinal microbiota occurring under the influence of external or internal factors, which can lead to pathological conditions, including not only inflammatory and oncological diseases of the gastrointestinal tract, but also disorders of the immune system, type 2 diabetes, vascular diseases, and even brain disorders. Possible ways to influence the intestinal microbiota are the use of antibiotics, probiotics, and prebiotics, as well as fecal transplantation, or FT, the transplantation of intestinal microbiota from a healthy donor into the patient's intestinal tract. It is believed that in our time, the procedure was first performed in 1958 on a patient with enterocolitis. And only recently, this method has been successfully used to normalize the intestinal microbiota in various diseases, including relapsing Clostridium difficile infections, metabolic syndrome, ulcerative colitis, and other chronic inflammatory intestinal diseases, and even autism. Moreover, clinical tests in the United States proved possible effect in 80% of cases with recurrent Clostridium difficile infection. And now it is officially recommended to use fecal transplantation after the second or third episode of this disease. With other diseases, the results are not so good. Foreign researchers write about 30–40% of permanent improvement in ulcerative colitis. Almost the same percentage is for temporary improvement. However, the FT method is currently used in many countries. There are different ways of performing FT. The donor microbiota is injected during colonoscopy. In different types of sensing, when using enemas, the microbiota can also be injected with pearl capsules. It is important to know that there are still no generally accepted standards for conducting FT, and therefore success depends on developments and techniques in each particular clinic. In addition, despite the apparent evidence, the mechanism of the positive effect of FT on the intestinal microbiota of patients with various diseases are only clearing up. In addition to the obvious mechanisms, the reproduction of useful bacteria and the displacement of pathogenic microorganisms, an important role plays the impact on the patient's immune system and injection of antimicrobial peptides and other molecules with regulatory functions. In Russia, FT was first used in Novosibirsk in the Center of New Medical Technologies of Chemical Biology and Fundamental Medicine Institute, Siberian Department of Russian Academy of Science. Later, FT was also used in Moscow, St. Petersburg, and Yekaterinburg. In our center, this method is used to treat pseudomembranous colitis caused by Clostridium difficile, ulcerative colitis, and some other chronic inflammatory bowel diseases. So, ulcerative colitis, the pathogenesis of this disease is not yet known for sure. It is believed that it can be mediated by disorders of the intestinal microbiota, genetic predisposition, environmental factors, etc. The effectiveness of FT in ulcerative colitis varies among different researchers, reporting from 20% to 92%. The official randomized clinical testing demonstrated stable remission in 25-27% of cases, which, however, was statistically significantly higher than the placebo effect. According to our data, the effectiveness of FT in the CNMT is higher. These The study uses feces samples from 20 patients, 57 years old, diagnosed with UC. The diagnosis was confirmed on the results of the level of fecal calprotectin study. The data of febrile colonoscopy and histological examination of biopsies taken from different parts of the colon and ileum. Samples in which Clostridium difficile was detected by routine methods were not included in the study. All patients provided informed consent to the ongoing study and anonymous data processing. Samples were collected 1 to 2 days before FT and 7 to 12 days after FT. The donors were young, healthy volunteers, 20 to 39 years old, without chronic diseases, who have not had an injection or have not been hospitalized for at least the last 2 months. And donors were examined, including general and biochemical blood tests, as well as blood ELISA for Giardia, Toxocara, Opisthochiasis, Ascaris, Trichinella. In addition, the standard test systems were used to confirm the absence of syphilis pathogen, HIV-1 and HIV-2, hepatitis B and C. Also, routine methods were used to analyze feces for dysbiosis and for the absence of pathogenic microflora, Clostridium difficile, Campylobacter jejuni, Salmonella spp, Shigella spp, enteroinvasive Escherichia coli, Cryptosporidium spp, Cyclosporus spp, Giardia spp, Isospora spp, rotaviruses A, novaviruses 1 and 2, and adenoviruses F, as well as helminths and other eggs. The aim of our study is to evaluate the change in the intestinal microbiome in patients with ulcerative colitis after a single FT. It is believed that on average, the biodiversity of the microbiota in healthy people is greater than in various pathologies, including diseases of the gastrointestinal tract and UC in particular. Therefore, with successful treatment, the bacterial biodiversity in the post-FT samples should increase. We assessed whether the biodiversity in the samples has changed after the FT. Indeed, the Shannon index, which reflects this biodiversity, is large in sample sequences from patients after the FT procedure than in samples from patients before the treatment. Although the difference is statistically insignificant, we see an increasing trend. It should be taken into account that we see this difference after only one FT procedure. Normally, they are carried out several times, and the effect should be increased. The second independent method for assessing the change in biodiversity is a pairwise comparison of samples before and after FT by the principal coordinate method. This comparison showed that 15 out of 20 patients after FT had increased biodiversity of the bacterial microbiome and 2 main coordinate black arrows. 4 more patients increased biodiversity after FT, at least in the area of 1 main coordinate, green arrows. And only 1 patient after FT increased in microbiota biodiversity. It is known that the microbiota of healthy people consists of permanent and transient species of more than 17 types, including firmicutes, bacteroidetes, protobacteria, actinobacteria, fusobacteria, and virumicrobia. The results obtained correlate with the data of other researchers, indicating a reduced biodiversity of the intestinal microbiota in UC. Thus, the studied samples from patients in average showed significantly fewer firmicute sequences, which is well agreed with the observations of other authors. And after FT, the number of firmicutes did not significantly increase. Firmicutes were mainly represented by Clostridium, mostly non-pathogenic, and Bacillus classes. It is important that after FT, the species composition of lactobacilli increased significantly, which is undoubtedly quite positive, since lactobacilli is not only involved in the breakdown of lactose and other carbohydrates, but also are antagonists against pathogenic microorganisms, displacing them from the microbial community of the human intestine. The proportion of sequences belonging to the bacteroidetes types in our patients with UC was not reduced, which differs from the observation of other researchers. And after FT, the share of bacteroidetes as well as firmicutes practically didn't change. This type was mainly represented by the sequences of bacteroidetes SPP and Prevotella SPP. And after FT, the proportion of bacteroid sequences decreased from the average of 19.7% to 9.6%. And the proportion of sequences belonging to the genus Prevotella increased significantly from the average of 3.7% to 14.6%. It is known that most of the bacteroids that live in the human intestine are able to decompose a variety of plant polysaccharides, and the intestinal microbiota of the inhabitants of Western countries is dominated by bacteroides, SPP, and in the population of countries with predominantly plant-based diet, Prevotella SPP dominates. Our patients before as well as after FT had Prevotella prevailed, same as residents of Southeast Asia. It can be seen that after FT, the number of representatives of Veromicrobia and actinobacteria and protobacteria approached the norm, and considering protobacteria, normalization occurred due to the decrease in the occurrence of pathogenic and opportunist species. Regarding opportunistic and pathogenic protobacteria in patients with UC before FT, more than 9% of Acinetobacter SPP, about 1% of Klebsiella pneumonia were found in single samples and noted in other studies. Also, Proteorhabditis and Stenotrophomonas multiphilia were found in single samples, the proportion of which didn't exceed 0.1%. It should be noted that after FT, the proportion of the above-mentioned sequences of pathogenic protobacteria in the corresponding sample significantly decreased, sometimes to more than 0. Or less than 0.02%. In addition to the sequences of pathogenic protobacteria in some samples from patients before FT, sequences of pathogenic representatives of firmicutes were found. Thus, 9 samples had C. difficile sequences, in proportion of which exceeded 0.5%. The increased occurrence of C. difficile in the intestinal microbiota of patients with UC was noted earlier. The sequences of Staphylococcus aureus 0.1% to 0.9%, Streptococcus and Enterococcus about 0.1% were also found in some samples. As in the case of pathogenic protobacteria, the proportion of these sequences after FT in the corresponding samples decreased dramatically. The exception was samples with C. difficile. Only in the corresponding sample, 3 out of 9 patients had the proportion of C. difficile sequences decreased after FT to 0.1%. Thus, the study of the biodiversity and taxonomic composition of the 16th sRNA gene fragment sequences associated with the intestinal microbiota was made for 20 UC patients before and after FT. The results of the study showed that a single FT procedure resulted in an increase in the average sequence biodiversity in samples obtained after FT, if compared to samples collected before FT. The proportion of lactobacilli was increased, their species composition has extended. The proportion of sequences of pathogenic and opportunist representatives of the firmicutes and protobacteria types has sharply decreased. Can you tell us the time, the dynamics in time?
Лечение, так сказать, патогенной микрофлоры, любое лечение позитивное.
Клиническая оценка наблюдаемой динамики производилась в ходе лечения. Данные, касающиеся риботипирования образцов, секвенирования образцов, они, соответственно, были за 2-3 дня до фекотрансплантации и за 7, в среднем где-то через 7 дней после выполнения трансплантации. Образцы, которые можно было бы...
2 days before the FT and 7 days after FT. Next time we shall consider more proliferate time period. Have you did the research of intoxication of this transplantation?
Limocylactobacillus fermentum, U21 genes and proteins that potentially determined it. To be presented by Dr. Alexey Vatlin from Vavilov Institute for General Genetics, Russian Academy of Science. You're welcome.
Thank you very much. I'm glad to. My name is Alexey Vatlin. I'm from Vavilov Institute of General Genetics, laboratory of genetic of microorganism, and I'm glad to present you my report about immunomodulatory activity of Limocylactobacillus fermentum, U21 genes and proteins that potentially determine it. So the study of the microbiome of the human intestinal tract is a global interdisciplinary area research in world science of the last decade. Recent data convincingly prove the possibility of selected according to certain criteria strains of lactobacilli and bifidobacteria selectively modulate the body's immune response and have antioxidant activity. Protein and peptide extracellular components of lacto and bifidobacteria potentially responsible for the immunomodulatory and oxidant activity and oxidant potential. Metagenomic studies of the intestinal microbiota have allowed the development of basic approaches for identification in the necessary strains of commensal bacteria with specified parameters. Modern knowledge about the structure and functions of the microbiota as well as the integration of data with previously obtained molecular genetic, transcriptomic, proteomic, metabolic, the results of the study of commensal bacteria, including probiotic, and allow it to form the concept pharmabiotics. Pharmabiotics, it is the term which was described in 2018 year. It is live strains of commensal bacteria of the human intestine, their components, for example, cell walls, polysaccharides, and ingredients, and other biologically active substances that exhibit pharmacological activity and are safe to use. So, the study of the immunomodulatory properties of individual strains of the human intestinal microbiota tract is a topical area of the research in the world. It has become obvious that the microbiome of a healthy person can serve as a source of obtaining pharmaceuticals for prevention and treatment disease, for example, of the immune systems. So we have some search strategy for pharmaceuticals. We selection some strains of various test systems. We try to find some genes and its products which can cause this effect of this immunomodulatory or anti-oxidative effect in vitro or in future in vivo. So there are many data about how components of lactobacillus bacteria have immunomodulatory activity. So that can be, for example, secreted proteins, some peptides, histamine. If we say about culture fluid, it could be dead surface layer proteins from the dead culture, or polysaccharides or other components, and of course living culture. So there is some scheme from the article from the 2019 year. So this is Lactobacillus fermentum and its potential immunomodulatory properties. So interaction occurs through the binding of lactobacillus to toll-like receptors located on intestinal epithelial cells. Probiotics primarily stimulate innate immunity, and further adaptive immunity is stimulated, and T-B lymphocytes are activated. The advantage of T-regulate lymphocytes and regulatory cells and interleukins are released. And lactobacillus can decrease or increase the expression of Th17 cells and cause the phenotype of microphages to switch from M1 to M2 and to M2 to M1. So my report is about unical strain, Limosi lactobacillus fermentum U21. This strain was selected in the Laboratory of Genetics of Microorganisms in Vavilov Institute from the stress-adopted organism of the astronauts. So the strain was deposited in ICOBM, and we have a patent for this strain. And this strain was selected in 4 different test systems. The first was test system which based on recombinant strain bioluminescence, Escherichia coli, and we showed that this strain can change in the presence of superoxide anion in the medium. So then when we added culture flight and this strain to nematoda in paraquat-induced oxidative stress conditions, we see that we see changes in median life of nematode. And then we selected this strain on 2 test systems on mice and rats, so under paraquat-induced oxidative stress conditions. And we found the preservation of dopaminergic neurons in the brain of mice, and preservation of motor coordination and short-term memory in rats. So then we did whole genome sequencing and tried to find how genes, which genes can cause this immunomodulatory activity, which are known in the literature. So we found there genes which code SPA-C. It is recombinant protein which can adhere to the fetal gut and modulate varying levels of toll-like receptor. We found the cluster of genes CPC which associated this surface polysaccharide production, and we found genes which produce histamine and some specific lactobacillus immunoregulatory gene RCIR. So then we try to test the potential immunomodulatory activity, this very standard protocol. So we take the HP1 cell line macrophages and then induction inflammatory process by LPS. by LPS, and we have LPS in two different concentration because we try to make two stage of inflammatory process. So the start of inflammatory process in the lower concentration of LPS and the maximum inflammatory process in the maximum of concentration LPS. When we added three different compounds, first was leaf cells, it was dead cells as a control, and it was supernatant. And of course we then extract RNA, and as a basic control of related expression was THP1 cell line without these added compounds. So we measuring expression of cytokine genes in THP1 cell line. It was for cytokines, 3 pro-inflammatory and 1 anti-inflammatory. So here you can see the articles which use a similar approach to measure immunomodulatory activity. As you can see in the left, we use 50 nanogram per milliliter LPS, and we found that supernatant here is 3 times when we measure expression. It was 2 hours, 6 hours, and 24 hours after adding the cells. We found that supernatant cause increased expression of interleukin 6, SN in start point of inflammatory at 50 nanogram per milliliter, and the maximum of inflammatory process by 1 microgram per milliliter of LPS. So you can see it here. Then we also found that interleukin 10 genes change their expression. Also, it by the supernatant adding at the start of inflammatory process. So here you can see all the results of our research. We found that interleukin 6 and interleukin 10, but this is very strange that pro-inflammatory and anti-inflammatory cytokines change their expression. But these are results, and then we suggested that there are some components in supernatant that maybe can cause this effect on THP1 cell. And we made a proteomic analysis of the extracellular culture fluid. This is control strain, and this is this is our strain U21, and this is 2 control strains. It's also Lactobacillus fermentum. And we found that there was 2 proteins. One is CLPB. It is secreted proteins, but we didn't find it in other control strains. And we found that Lys-M protein is higher than the other in control strains. So CLPB protein is a component of the CLPB chaperone protease complex that is involved in protein degradation and disaggregation. There is some data in the literature about that this CLPB protein can cause effect on physiological disease in this cycle. And we suggest that maybe it is decay of our immunomodulatory activity. Or also we found Lys-M protein. This protein is more bigger than protein in control strains, and we found that in this our protein is unique serine threonine peptide. it contains the incorporated STP, and by the literature, Liz-M may be responsible for the immunomodulatory activity, and we suggest that maybe this protein cause this immunomodulatory activity, cause this effect of our strain on THP1 cell. So conclusions. We showed that the immunomodulatory activity of the Lactobacillus fermentum U21 strain. One of the possible reasons for the activity may be the unique secreted protein Liz-M with incorporated serine training peptide, and we are trying to prove the activity of Liz-M protein in the immunomodulatory properties in vitro and in vivo. Now we try to have this, to take this protein from the Escherichia coli producent strain, and then I think maybe we have good results in the future. Thank you very much.
Thank you very much.
Waiting for your questions.
Yes, please. Who wants to ask the question? Professor Chikindis, please.
Thank you for a very interesting presentation. My question relates to your conclusion part for the future work. You mentioned, if I am not mistaken, please correct me if I'm wrong, that you will probably try to clone the gene into another host, and then to see if you can observe, similar or whatever effect in the recombinant host. But then again, this recombinant host will have other factors that may influence, so it's not a very clear experiment in proving the activity. It may work. I hope it will work. What about another option? And again, I don't know if this is a right approach. Of course, ideally, that would be to isolate and to see if it will remain biologically active as an isolated protein. But can you make a knockout mutant of, or the mutant with the decreased activity?
We suggest that we try to take the purified protein, as you said, firstly, and then if this protein produce some effect on our test system on THP1 cell, only then we...
Then you don't need anything else, because you have a
yes.
pure protein with activity.
Yes. Yes.
Right.
So it's all.
Thank you.
Thank you. Who else wants to ask the questions? Okay, thank you very much again, and we move further to the presentation. Probiotics and human hygiene, to be presented by Professor Tatiana Khadykina from Ivan Mikhailovich Sechenov, First Moscow State Medical University. You're welcome.
Hello, my name is Tatiana Khadykina. I am an associate professor at the Department of General Hygiene at the Erisman Institute of Public Health at the Sechenov First Moscow Medical University. The topic of our report is probiotics and their influence on human hygiene. Introduction. Within the human body, there are about 40 trillion bacteria, comprising the more than 5,000 microbial taxonomic profiles and living in communities called microbiota. Microbiota could be found in various areas of the body and play an important role in the functioning of our organs, tissues, secretions, metabolic processes, etc. Depending on the condition and interactions of microbiota, physiological and pathophysiological reactions may occur within the human body, sometimes resulting in illnesses. The use of probiotics has an overall positive effect on the body. They can prevent and or help regulate microbiota activity in a way that can deter the manifestations of all illness. Background information. Probiotics are health-promoting bacteria that when administered in an adequate amount to the eukaryotic host, including the microbiota of certain parts of the body, can help prevent or treat disorders within the host and modulate the resident healthy microbiota. Although many health-promoting microorganisms are naturally found within the body's microbiota, the supplemental or necessary addition of probiotics sourced outside the body can be used to aid the well-being. Probiotics most commonly come from Bifidobacterium and Lactobacillus species. Probiotics find their use in various food products, dietary supplements, and medicinal formulations. The objective of the study. This study focuses on how probiotics have a positive effect on microbiota in various hygiene-related parts of the body. Probiotics are tested to see how they could be used to help prevent and/or treat human hygiene-related illnesses that are usually caused by disorders in normal microbiota behavior and the infiltration of bad microorganisms. Throughout the COVID-19 pandemic, it is of paramount importance to learn more about how to improve the health of our populations. The focus on studying probiotics as a way to combat hygiene-related illnesses could be the start of the future of medicine and health. The study's specific objectives. Probiotics use is critically reviewed for the following: gastrointestinal hygiene, food hygiene, oral hygiene, vaginal hygiene, urinary tract hygiene, skin hands hygiene, armpit hygiene. Gastrointestinal hygiene, clinical study. Probiotic use was effective against common gastrointestinal diseases, gastrointestinal disorders, irritable bowel syndrome, allergic diseases, cancers, etc. Exception: regular use of probiotics as a means to prevent allergic diseases is not effective. Proper food hygiene is a critical part in maintaining gastrointestinal health. Probiotics help create a protective barrier for intestines. Oral hygiene, clinical studies. Probiotics could be used as an alternative to antibacterial products, appears to prevent halitosis and reduce bad breath by attacking pathogens. Vaginal hygiene, clinical studies. Probiotics are effective at curtailing the presence of candidiasis. Lactobacilli play an important role in maintaining the pH of the vagina, prevent other bacteria from altering the naturally acidic pH. Urinary tract hygiene. The use of probiotics to treat urinary tract-related infections was not significant enough to determine if it is truly effective. Further research is needed. Skin hands hygiene. The immunomodulatory effect of probiotics further promote the health of the epidermis and health ward of the pathogens. Products with probiotics are found to help restore the microbiota on the skin of hands. Probiotics can be used subsequently with antibacterial substances to keep hands clean and healthy. Armpit hygiene. Clinical studies. Antiperspirant products for underarm odor that contain probiotic elements are becoming popular, but there is no concrete research on the effect of probiotics on the hygiene of armpits. This is diagram discerning the causes and proposed resolutions of microbiota imbalance in various parts of the body. We studied lifestyle, habits, professional activity, scalp and hair health, oral cavity health, underarm health, gastrointestinal health, urogenital health. Discussion. Overall, there is a positive trend in the impact of probiotics on health. The studies conducted and conclusions deduced by other researchers allude to a promising future. Hygiene is an important aspect of health, and it is imperative that more research is done on probiotics to increase their participation in medicine. Conclusion. After analyzing each respective test, the use of probiotics for gastrointestinal, oral, vaginal, and skin hygiene and health is recommended, but with limitations. Probiotics dosage and regularity should be adjusted per individual according to the issue it is trying to address. In the future of medicine and health, Probiotics could be an alternative to antibiotic and antiviral substances. More research should be done in the future in order to further establish the true effectiveness of probiotics on the health of humans. We are grateful to Kelly Fowler and Nikolai Wadzinski for their efforts in aiding her navigation through this program, and to Akina Torisawa for her support throughout this program. Our program was supported by the Ministry of Science and Higher Education of the Russian Federation. We also express our gratitude to the staff of the section of First Moscow University for their support and assistance in this work. Thank you for attention. Hello.
So since this presentation is recorded, so please refer your questions in online services. And we move forward to the next presentation. It is the role of small intestinal microbes for central nervous system inflammation, to be presented by Dr. Hiroshi Ono from Laboratory of Intestinal Ecosystem, Riken Center for Integrative Medical Sciences, Japan. You are welcome.
Hi, my name is Hiroshi Ono. First of all, I would like to thank organizers, especially Dr. Minchi Leon, for providing me this opportunity to share some of our new latest work. So I'm going to talk about the role of small intestinal microbes for central nervous system inflammation, namely multiple sclerosis. As you may know, the cells making our body, the number of cells is about 23 to 30 trillions. In contrast, there are more and more bacterial cells reside in our body, especially in the colon. They say that 40 trillions of gut microbiota harbors in our body. And they affect a lot of the host, us, physiology and pathology, not only gastrointestinal, but also immune diseases, metabolic diseases, and even neuronal diseases. And I'm going to talk about multiple sclerosis today. So let me share some background. So I think you may know hygiene hypothesis, lack of exposure to infectious microorganisms and parasites could affect our disease state, and also dysbiosis. This is newly comes. And this dysbiosis microbiota also affect the susceptibility to kind of immune disorders by suppressing the natural development of the immune system. So not only allergic diseases, but also the autoimmune diseases such as type 1 diabetes and multiple sclerosis is quickly increasing. So this could be attributable to this hygiene hypothesis. So I'm going to talk about multiple sclerosis that is done mostly by Dr. Eiji Miyauchi in my lab. And multiple sclerosis is an autoimmune demyelinating disease. So myelinates covering the nerve fibers in the brain and spinal cord are damaged by autoimmune inflammation at the multiple foci in CNS, so resulting in a variety of neuronal disorders are listed here. And the incidence is quickly increasing, not in Japan as well as worldwide. And this is from the review article, the pathogenesis of multiple sclerosis. First, priming occurs peripherally, in periphery, and somehow those in pathogenic autoimmune T cells evade to, the blood-brain barrier to reach the CNS and then attack the myelin test. So the experimental model of multiple sclerosis, the experimental autoimmune encephalomyelitis, or EAE. In some models, myelin peptide-specific TCR transgenic mice, such as 2D2 mice that we used in this study, sometimes called spontaneous EAE. And another model is the immunization with myelin protein peptides. Most common is the peptide from myelin oligodendrocyte glycoprotein, or MOG, and at the same time pertussis toxin used to permeabilize blood-brain barrier. The consequences like limb tail, and then if EAE more severe, paralysis of hind legs and then front legs, and eventually die. So the involvement of gut microbiota in this EAE comes in several publications. For example, spontaneous EAE in the transgenic mice, the symptoms go on when mice are made germ-free. And also the immunization protocol, also germ-free mice attenuate the pathogenesis. These are the animal models. And in humans as well, the dysbiosis in the gut microbiota of MS patients is reported from Japan as well worldwide. So the gut microbiota could affect the CNS inflammation, but we don't know which kind of bacteria are involved and how. So AG tried different antibiotics, the effect on EAE, and he found that only ampicillin could attenuate very much the onset or pathogenesis of EAE, but not others. And if given orally, and if ampicillin is given intraperitoneally or systemically, it doesn't work. So it means that gut microbiota is involved. And then he next asked, where in the gut this the ampicillin could affect? And he first found that in the small intestine, interferon gamma and IL-17, the pro-inflammatory cytokines are highly induced upon EA induction, and large intestine, small amount, and mesenteric lymph node and Peyer's patch, not very much. And this induction of the inflammatory cytokines are profoundly suppressed in the presence of ampicillin. Yes, the small intestine is a major site for the site of the ampicillin influence. And the Treg cells are known to be involved for the suppression of the EAE and MS. So he next asked whether Treg cells are induced upon ampicillin, but it is not the case. And so in the control without ampicillin, positive EAE case, the number of Treg cells upregulated, but the percentage is not. This is because the Th1 and Th17 autoimmune pathogenic T cells also increased. And this could be in response to this inflammatory responses, because Treg cells are induced upon inflammation. And with ampicillin, no increase in Th17, Th1 cells, so there's no increase in Treg cells. This means that ampicillin is not induces Treg cells to suppress EAE pathogenesis. And he next found that CD4 T cells proliferate in the small intestine in EAE. So this is a normal immunization model, and this is 2D2 transgenic mice. In both cases, the Ki67 positive proliferative cells increased upon EAE induction, or in 2D2 mice in the small intestine. And these CD4 T cell proliferation only occur in intestines, small intestine and large intestine, but not other places, including Peyer's patches and mesenteric lymph node. So intestines, again, is a very important place for interaction of these inflammatory T cells. And this inflammatory T cell proliferation depends on the presence of microbiota. So when microbiota is suppressed with the presence of the 4 cocktail of antibiotics, this proliferative cell numbers go down like this, and Ki67 positive cells goes down. So microbiota is involved. So in this scheme of pathogenic genesis of EAE or MS, we now put small intestine here. So after priming, boost should occur in the small intestine to increase the number of the inflammatory, autoimmune inflammatory T cells. And then those increased cells goes into the CNS to aggravate pathogenesis. So AG next asked which kind of bacteria are involved. So he found only the bacteria that decreases very much, only in the presence of ampicillin, but not other antibiotics. So that is this so-called OTU2. So this is this strain belongs to Aspirotrucaceae bacteria, and very similar to Alobacillum, but 16S identity is 86%, so this is not the same as Alobacillum. Alobacillum related, Aspirotrucaceae. And he could isolate this strain, and if monoassociated in the germ-free mice, only in the small intestinal epithelium, this bacterium adhered tightly to make biofilm-like structure, but this is only in the small intestine, but not in the large intestine or cecum. So they are very have high affinity with the small intestine alone. And this is another way to show the gut microbial composition. And this guy is Aspirotrucaceae that is almost gone in the presence of ampicillin. And this is the largest population in our mouth facility in the small intestine, Lactobacillus. Actually, this is Lactobacillus reuteri. And they are not gone in the presence of ampicillin. So he also isolated this one. Then compared this as a control to see the effect of this in the germ-free mice of the EAE. So he found that Lactobacillus, almost no EAE occurrence, like a germ-free, but this OTU2, Aspirotrucaceae, somehow induces EAE. And then Th17 cells also increases upon administration of this OTU2 Aspirotrucaceae, but not with the Lactobacillus. And this OTU2 Aspirotrucaceae induces IL-17 and interferon gamma production in the small intestine and splenocytes, and also in the small intestine, serum amyloid A and IL-23 cytokine genes are induced with these Aspirotrucaceae. These are known to induce Th17 cells. So OTU2 promote Th17 responses. But the pathogenesis with the OTU2 is not as high as SPF mouse. This suggests that some other bacteria could also affect the pathogenesis of EAE. And when AG co-administer OTU1 and OTU2, Lactobacillus and Aspirotrucaceae, this is the cell bacterial number in the feces. So in the presence or absence of OTU1, OTU2 number is not very much affected, but the pathogenesis is like this. So with OTU1, much higher pathogenesis occurs of VAE compared to the OTU1, OTU2 alone. So OTU1 also could do something, and we thought that antigen mimicry could occur. So when he sought the MOG mimicry peptide in OTU1, there are 3, and OTU2 also have somehow MOG mimicry peptide. So he chose these 2 to see whether they could cross-react with the MOG-specific T cells. And it was the case here. So you could see here in 2D2 cells, so MOG-specific T cells in vitro stimulation with those mimicry peptide or MOG itself. Of course, MOG could strongly induce the proliferation of these 2D2 transgenic T cell, but UVRA from OTU1 weakly but significantly induces the proliferation of these cells, but the AP peptide from OTU2 do not. So OTU1 lactobacillus could provide MOG mimicry peptide to cross-react with MOG-specific T cells. And we further confirmed whether UVRA peptide is responsible. So he knocked out this gene in OTU1, like this, and associate with OTU2 in germ-free mice. So wild-type OTU1 and UVRA knockout OTU1. So UVRA knockout proliferate as well as wild-type OTU1 in the intestinal mouse, but the pathogenesis of VAE is attenuated with knockout OTU1, like this. So this is the final slide. So in the presence of H-Petricacea OTU2, they provide serum amyloid A and IL-23. So the intestinal migrated pathogenic T cells become Th17 in the presence of these Th17 adjuvant activity. So they become pathogenic and then migrate into the central nervous system to cause demyelination. And in the presence of lactobacillus, they provide UVRA MOG mimicry peptide. So cross-reactivity occurs and proliferation of the autoreactive T cells. But this alone did not induce Th17 differentiation, so now pathogenesis occurs. But in combination with this, now proliferated T cells are differentiate into Th17 cells, more Th17 autoreactive cells produced. So more severe pathogenesis occurs. So in conclusion, 2 different small intestinal bacteria act together to be involved in the pathogenesis of VAE. And this could also happen in MS patients. Okay, this is my group, and these are the collaborators, contributors. Eiji Miyauchi is the most important person, but other guys from my lab and from other labs in our institute, and also the collaborators from Okayama University, Professor Morita, and Gunma University, Professor Suzuki, and other people. And these are fundings. So thank you very much for your attention.
This presentation, as well as the next presentation, is also recorded, so please follow the questions to the authors using the mails. So, and we are coming to the next and final presentation for today. It is fasting and fasting mimetics address certain pathways and microflora structure, to be presented by Professor Alexander Hasselberger from University of Vienna. Please.
Hello from Vienna and University of Vienna. It's really a pity that I cannot come to the conference. I really would have liked to do so. I would like to go with you through some aspects of aging, fasting, caloric restrictions and microbiota, fasting mimetics and microbiota. We all realize in the public media that longevity and aging is presently hype, especially driven by economy. And Dr. Sinclair, big companies, Bill Gates, think about if aging is even a disease. But nevertheless, we learned about molecular mechanisms of aging, of fasting and caloric restrictions in this way. The mechanisms of aging were summarized in the so-called hallmarks of aging, and maybe telomeres were the first, mitochondria, DNA instability, DNA repair, and others. But we also see that aging has very personal aspects, and we learned about the archetypes of aging, and that there are groups of people where inflammation and metabolic aspects drive aging. In other people, maybe immunosenescence is of special importance, and some also other pathways. And we also learned that responses, for instance, to diet can be very personally, as you can see here, different glycemic responses to the same diet. Epigenetic mechanisms are of special importance, as these mechanisms, so chromosomes, chromatin, DNA methylation, non-coding RNAs, are really involved in the regulation of most, if not all, of the other hallmarks of aging. We learned that with aging, the methylation of cytosins or CpG islands are changing, and that these changes of hundreds of these CpGs is a good marker for either slow, healthy aging or accelerated aging, and that these markers also tell us about diets and our lifestyle, and this epigenetic clock is maybe now the best marker for aging. We learned that each single step of autophagy is regulated by microRNAs, histones or methylation, and we also know that a number of pharmaceutics and natural compounds like EGCG, quercetin, resveratrol, curcumin enhances autophagy, and rapamycin using the mTOR pathway or spermidine influencing this acetyltransferase here became famous for enhancing autophagy, which might be important in a number of inflammatory, for instance, neuroinflammatory diseases. And also senescence is regulated by the different mechanisms of epigenetic. Senescence became quite important recently, as these cells which cannot divide anymore and which are secreting a number of inflammatory mediators, decide either if they do not develop into tumors anymore, but on the other hand, they make tissues senescent, and this is, of course, a major problem in aging. And we try to get rid of these senescent cells in aging using senolytics. And for economically important rejuvenation, we are presently try hard to look for senolytics, for instance, the combination of quercetin and this kinase inhibitor, or natural compounds like epigallocatechin galate or others. The interaction of epigenetic and mitochondria has a long history because of the antioxidant properties of mostly epigenetically active polyphenols. And for a long time we thought that this is one of the main aspects of anti-aging, but nowadays we learn about the hormesis aspect, that we need a little bit of ROS activity for healthy aging, and also for activating a number of important health-related mechanisms. Histone modification, microRNAs, but also DNA methylation is also known to regulate the length of telomeres by regulating telomerase activity. We looked for the effects of a number of polyphenols, especially EGCG from green tea, on telomerase and telomere lengths, as well as methylation of the regulating factors CMYC and HTAT. And what we interestingly found was that in cancerous cell lines we found inhibited, reduced telomerase activity and telomeres, whereas in normal cell lines, in these fibroblasts for instance, we found increased telomerase activity and telomeres. When we now look again for these hallmarks of aging, we may see that there is one hallmark missing, which is presumably interesting for all of us, and this is the role of microbiota. And it is well known that gut microbiota change in the way of aging, and we also know that gut microbiota is a key control of healthy aging in elderly people. That with aging, a number of really important short-chain fatty acid producing bacteria are decreasing, and with this decrease we are losing some activity of dietary fiber, short-chain fatty acids, and healthy effects transmitted via these G protein coupled receptors here. On the other hand, there is some discussion if butyrate and fibers can improve inflammation in aged mice and in other animals. So this is really an important aspect in healthy aging. Aging is an aspect in most organisms, and in most organisms we also find that caloric restriction and fasting is increasing lifespan. And we learned about the pathways of caloric restriction and fasting, especially here this NAD sirtuin pathway, the AMP, AMPK pathway, amino acids and mTOR, and fatty acids and PPAR pathways. Especially this histone deacetylases, the sirtuins became famous with caloric restriction, and also their targets, the histones, P53, NF-kappa-B, and PGC1 alpha, linked especially to mitochondrial activity. We learned about differences in the sirtuins, the one group SIRT1 in the cytoplasma, SIRT3 in mitochondria, but also some others. But it's not only this histone deacetylases, also DNA methylation is involved or reacts to caloric restriction and fasting, and we can see that caloric restrictions dramatically changes this methylation of cytosines here, and this is of course linked also to the healthy aging as seen and discussed in the epigenetic clock of Forward before. But there is also good evidence that caloric restriction and fasting also alters the microbiota, and with the microbiota the immune system here, like shown here in this publication on T cells, Th17, and the number of microbiota changed. And obviously there is also different changes in caloric restriction and fasting, of course, depending on the timelines. In nutritional research, the mechanisms of epigenetic became especially famous as a big number of nutraceuticals, plant ingredients, additives address mechanisms of epigenetics, sometimes one or more of these epigenetic mechanisms, and for instance for curcumin is known that it affects all three mechanisms of epigenetics. So it was not surprising that scientists looked for possibilities of polyphenols and epigenetically active plant ingredients to mimic the effects of caloric restriction and fasting using some of these compounds or even mixtures of these compounds, especially as for some people caloric restriction and longer times of fasting seem to be too difficult. And so caloric restriction or fasting mimetics became a next hype in this area of longevity. So presently pharmaceuticals and nutraceuticals are screened for effects of inducing SIRT enzymes, and especially looking for the different health-related targets of these different sirtuins, and a number of books are already available claiming a SIRT food diet in this area. At this point here, we need to discuss and clarify that there are of course differences between caloric restriction and fasting depending on the timelines. In our hands, intermittent fasting like caloric restrictions for 16 or 18 hours would not produce ketone bodies in a significant amount, whereas of course fasting and fasting protocols such as 5 or 7 days or longer fastings produce epigenetic active butyrate or beta-hydroxybutyrate quite efficiently. This difference between caloric restriction and really a fasting diet needs to be kept in mind when looking for the consequences of mimetics in this area. And this is a nice graph showing the different effects, for instance, of plant ingredients or natural compounds such as polyphenols, resveratrols, curcumins, quercetin, epigallocatechingalate and others, and of course spermidine, mostly addressing the SIRT pathways, and metformin going this way down, AMPK, and rapamycin addressing the mTOR pathway. Even aspirin is nowadays claimed as a kind of a fasting mimetic, surprisingly. But these nutraceuticals, polyphenols, do not only affect sirtuins and other enzymes. They are also very well known to modify structure of the gut microbiota and also interfere with the production of butyrate, and with these effects on epigenetics. And of course, microbiota have also a special role in the area of the effects of polyphenols, as the metabolization of these polyphenols is decided by microbiota, even mostly personally quite different if metabolites are built which are maybe even biologically active and better bioavailable than others. So polyphenols and natural compounds may act on gut microbiota structure in the way that they can favor the increase of beneficial bacteria, or they may also change the fermentation activity of the microbiota. Not only these natural derived fasting mimetics, polyphenols, are changing gut microbiota structure, also these pharmaceutical compounds like rapamycin or metformin are quite well known to modify microbiota, but of course there is also, like so often, the question of the hen and egg in this area. So as we became interested in the interaction between epigenetics and microbiota, we designed a study analyzing fasting and fasting mimetics and their consequences. In a pre-study in vitro, we looked for these different senolytics, fasting mimetics, listed here: anthocyans, spermidine, resveratrol, epigallocatechin galate, on their effects on senescence and autophagy in pre-adipocytes and adipocytes, using beta-gal and induction of senescence by brom-deoxyuridine. We found that EGCG effectively reduced senescence, looking for P21 as a marker, but also this SASP phenotype, so this inflammatory phenotype, whereas EGCG and most of the polyphenols, resveratrols, anthocyans, and spermidines stimulated SIRT3 as also described in literature. And in the main study, we compared the effects of a fasting protocol that was 7 days Buchinger fasten, which means below 250 calories for 5 days, and we had 20 people recruited in this group. One of them was I myself, and you can see me here in this beautiful fasting setting. And we compared this fasting group with a group of 150 people consuming a fasting mimetic SIRT shot, which was composed of these blueberry extracts, broccoli extract, apple, citrus, nicotinamide, with the ingredients of anthocyans, sulforaphan, fluoridine, quercetin, and some nicotinamide and some others. And this shot was taken for three months with 150 participants. After the 7 days of fasting, we noticed a highly significant increase ketone bodies, which already changed after day 2 and 3. And we also see a significant increased mitochondrial DNA, but also the third shot increased mitochondrial DNA significantly. We analyzed blood spots taken before the fasting and the third shot, and one time in between and at the end of the fasting and the third shot period, and we analyzed expression of microRNAs and mRNAs. What we have seen in the fasting protocol, we have seen really increased CERT1, CERT3, but also FOXO-P21 and DNA repair enzyme MLH1. After the 3 months of the third shot, we have noticed significant increases of CERT1, FOXO, and some tendencies of CERT3. After the fasting, we have seen quite a dramatic increased diversity at the species level, not on the Shannon index. Seen effects on the Firmicute-Bacteroidetes ratio. And we found changes in a number of species, significantly increased was Christensenella. In general, fasting stimulated the group of proteobacteria, whereas the third shot increased actinobacteria. Looking at correlations, we found a quite interesting correlation between telomere length and CERT1, which would make sense as, of course, histone deacetylases are involved in the regulation of telomerases. We also found a correlation with CERT6. We found, as expected, a correlation between the BMI and the ratio of Firmicutes and Bacteroidetes. And very interestingly, we found a correlation between the Bacteroidetes abundance and a microRNA, this microRNA 125, and this microRNA is very well known for effects on cell metabolism, on proliferation and differentiation, so also apoptosis. So this is really a microRNA which is a marker for aging, and this correlation with Bacteroidetes is quite interesting and needs to be explored further. In conclusion and discussion of our study, it was evident that caloric restriction mimetics affect some of the mechanisms activating by fasting protocol, which may be of interest in the area of healthy aging, but scientifically much more interesting is the interaction between the SIRT, maybe even the TOR pathways, and the structure of microbiota, and especially this interaction between, as discussed for this microRNA and microbiota, is or maybe the starting point for further analysis in the interaction between epigenetic mechanisms in aging and fasting and longevity. With this, thank you for interest. I'm looking forward to an exciting discussion and learning from your ideas in this area. I hope to meet again on these two websites, especially the one website here addressing aspects of personalization. Thank you and bye-bye.
Thank you. This presentation is active and open for discussion. If you have questions, please, you are welcome. Please, Professor Chikendas.
Okay.
Good afternoon. Thank you for being with us at this time, and thank you for the very interesting presentation. I have a couple of questions. Question number one, perhaps I missed it, but when you were talking about the fasting experiment, what was the age group? Because we know that the response to fasting is different in different age groups. Can you comment on this matter, please?
Yes, thank you. I think typically the fasting group was between 40 and 60, so we had not too elderly people in the group.
Thank you. And the second question would be, you mentioned the role of microorganisms and how the community is changing, for instance, with the fasting. As for the positive effects on aging, you gave very good examples of what is now termed as phytobiotics, and briefly mentioned some microorganisms. And I wonder if you, from your personal experience in the area, can add anything on the types of microorganisms which may be considered as prominent source of the health-promoting bacteria that may be beneficial in the cases of aging. The reason I'm asking about it is because, well, we're working largely with the spore-forming bacilli, and I came across of just a few papers, literally 2 or 3, reporting on the really well documented by these groups effect of bacilli, spore-forming bacilli, and they were even saying that they're playing an important role as commensals in human microbiota, gut microbiota. What is your take on that? And maybe you can share with us your vision on where to hunt for the best strains or species when it comes to aging. Thank you.
Well, first of all, thank you. This is really a very exciting question and an exciting talk. I certainly need to go into the details of the analysis to look for bacilli, and I would be glad to be in touch with you to discuss this and get exact data. I'm on my mobile phone right now. What I think is important in fasting is the timeline. Of course, after the week of fasting, which makes stress to the gut microbiota, and I think stress is always a cause for species diversity. It would be needed to follow up what is the distribution about the next weeks after this fasting protocol. And this is something which is going on right now, and I would expect that this changes again after this stress phase of fasting itself. For instance, what I would be, of course, eager to know if Christian Zanella, it keeps being enhanced, because that would be a nice argument for healthy effects.
Perhaps the last question for me, and I'm sorry for keeping you busy, but it is really a very exciting topic. Another question, and again, it's your feeling, your opinion. Fasting and the period after fasting, what is your vision on what is happening to the human microbiota? Restoration to the original point or improvement?
Thank you. Very good question. I think the benefits of the fasting week is very essential on the follow-up of the nutrition and the lifestyle. And I would really pick up where Cohen Venema ended, where he said, well, this kind of fibers which then can influence microbiota or specific personal strains. And I think the combination of fasting and prebiotics thereafter would be really a protocol which would be very exciting to study.
Thank you very much. Thank you very much. Thank you very much for the presentation. For presentation, and we are about to finish this this session now. I must, as a co-chair, I must say that I have admired this session and admired the wonderful high quality presentations. They are all high quality presentations and different kinds of interesting spin-offs for neurology, for central nervous system, for immunology and physiology and nutriology. And so every presentations was marvelous, I should say. And I do look forward to the next meetings like that, dedicated to human probiotics, and to get a real pleasure from the presentations and results of very interesting scientific works. Anybody maybe want to say a couple of words concerning your impressions? Anybody wants to say a couple of words concerning impressions on the session? No, the session is over.
Коллеги, трансляцию